Okay, welcome back students. So now we are going to complete the remaining portion of 12th class. As you know, you are not known by your starting, you are always known by your end. Okay, आप अपने आरंभ से नहीं अपने अंजाम से जाते हो. Okay, so now the third part of principle of inheritance, that is your mutation. Okay, so mutation, it is a biological process in which the genetic makeup of the organism goes down. It will totally change. Okay, the genetic makeup of an organism change due to the enhance of some introduction of some mutagens, due to which whole genetic makeup, whole sequence of DNA is changed in that organism. That is the mutation. Okay, so that thing is written here. Mutation is a phenomena which is result in the alteration of DNA sequence and consequently result in changes in the genotype and the phenotype of the organism. You cannot differentiate the phenotype and genotype of the organism. Okay, that is the mutation. In addition to recombination, mutation is another phenomena that leads to variation in DNA. Just like a coin have two faces, heads and tails. Just like that, an organism can be go inside mutation and your evolution. Okay, mutation is fast, evolution is slow. Mutation causes rapid changes in the organism phenotype and genotype. Evolution takes millions of years to do that thing. Mutation is directionless, evolution is always directional. Okay, so next, the relationship between gene and DNA are best understood by mutation studies. As you know, the father who discovered the theory of mutation, who gave the theory of mutation, Hugo de Veris, he tells that the relationship between gene and DNA are best understood by mutation studies. Okay, by the help of mutation, you can study the degree of change of DNA can resist, the degree of resistance a DNA can absorb. Okay, all this thing you can all easily studied inside the mutation. Okay, only those mutations are heritable which occur in germinal cell of an organism, while somatic mutations are not heritable. For example, if my hand having a cancerous growth, okay, and it is because of some mutagens or carcinogens, then this thing will not go in my child. Okay, but if my germ cells, if my testis cells get mutated, so it definitely goes in my progeny. So mutations are inheritable, which happen in germ cell, while the mutations which is happening in somatic cells, they are not inheritable, that remains to that particular organism. Okay, next, mutations mostly harmful. Okay, as I say, mutations are random, they are directionless, so they are harmful. Okay, in some cases we can use mutation in proper way, that is to increase the variety yield in a plant, just like polyploidy of wheat. Okay, next, sometimes they are lethal which lead to death of organism, but sometimes they are beneficial also. The lethal one is the bad thing of your mutation, and the good one is to production of different high yielding varieties of plants. Okay, mostly mutation are recessive and they never eliminate from a population. Okay, it carries with recessive gene. Okay, mutation is carried by your recessive gene, so it will not eliminate by the population, from the population. Okay, next, muton, it is the unit of mutation. As you know, nephron is the functioning unit of your kidney, neuron is the functional unit of your brain. Just like that, muton is the functional unit of mutation. A small part of DNA which undergoes mutation is called muton. Okay, that thing is written here. The smallest part of DNA which undergoes mutation is called muton. Okay, it can be of one nucleotide and it can be of 3 to 4 codons. Okay, next we have causes of mutations. Okay, basically there is three causes of mutation. First one is physical, second one is chemical, and third one is biological. Physicals, your gamma rays, X rays, cosmic rays, alpha rays, beta rays, all these are the causes of mutation in organisms. These are physical sources. Second one is your chemical sources, in which chemicals come in the play. That is your mustard gas, nitrous gas, nicotine, okay, vinyl chloride, vinyl phenyl, all these hydrocarbons, different hydrocarbons, these all are different mutagens who causes mutation in the organism. Okay, so there is written, you have physical mutagen, chemical mutagen, physical UV radiation, alpha, beta, gamma radiations, and X-ray. While your chemical mutagens are mustard gas, phenol, formalin, acetic acid, formic acid, ammonia, etc. There is biological mutagens also, which we call oncogenes or oncogene viruses. Okay, the viruses who causes mutation in the body of organism. They also causes cancer in human and they also cause mutation in the human. Okay. Next we have some words, some theories which is related to the mutation. First we have the mutation theory. The word mutation, it is given by Hugo de Veris. In evolution chapter we studies that theory of mutation. According to that, mutation is directionless, it is random, it can be happen anytime. That is the words of Hugo de Veris. Okay. Next, Hugo de Veris studied mutation in plant Orientha lamarckiana, that is your evening primrose. Okay. Next, Beadle and Totten induced mutation in neurospora by the help of UV rays or X-rays. He convert a wild neurospora into mutant neurospora by the help of UV radiation. Okay. Next, they also give one gene, one enzyme concept. Means a one gene is responsible for production of a particular enzyme. For example, in your lac operon, your Y gene is responsible for the production of beta galactosidase enzyme, while your Z gene is responsible for permease enzyme, and your A gene is responsible for production of your trans-acetylase enzyme. Okay. Now that A gene is not come and produce your beta galactosidase. Your Z gene is not come and produce trans-acetylase. Okay. So the enzyme, particular gene is responsible for the production of a particular enzyme. Okay. That is the theory given by your Beadle and Totten, which we call one gene, one enzyme concept. Okay. Next, MS Swaminathan induced mutation in wheat by the help of gamma rays to obtain a good variety of wheat, which we call Sharbati Sona or Pusha Lera. These all are the varieties of wheat which is produced by mutation, by polyploidy process. Okay, making your wheat plant polyploid. He produced many semi-draft, many drug resistance, many pest resistant varieties of wheat which we now eat daily. That is your Sharbati Sona and Pusha Lerma. Okay. Now next we have mutations type. Okay. So mutation is of two type: point mutation and frame shift mutation. Okay. So first we have gene mutation, which is also called your point mutation, and all these types come under this. Okay. So mutation arises due to the change in single base pair of DNA. This is known as point mutation. A single point. In a single point of a nitrogenous base, the changes occur, then it is called point mutation. For example, you can see here, if I have a codon line, that is AUGCACGAC. Okay. Now if I remove this C from here, okay, now that mutation will occur. That is your point mutation. At a particular point, the mutation is started. The cause of mutation is a particular point in a sequence of DNA. Then it is called point mutation. Okay. Then gene mutation, that is of two types: your substitution point mutation, as I say now, and frame shift mutation, in which whole frame is shifted, due to which mutation occur. Okay. So we have first substitution mutation or point mutation. Okay, it is again of two types, then transition and transversion. See, we have know that in the starting of the molecular basis of inheritance, we studied that in the DNA we have two nitrogenous bases. That is your pyrimidine and purine. Okay, purine are your adenine and guanosine. Pyrimidine is your thymine, uracil, and cytosine. Okay, so in transition, what happened? One purine changes its plates with other purine, and one pyrimidine changes its position with other pyrimidine. Then it is your transition type of mutation. Okay, and if this pyrimidine changes position with your purine, then it is your transversion. Okay, again I am saying the transition is when one purine replaces its position with other purine, or one pyrimidine replaces its position with other pyrimidine, then it is your transition substitute type of mutation. Then we have transversion, in which one purine changes its position with pyrimidine, or pyrimidine changes its position with purine, then it is your transversion type of substitution mutation. Okay. Next we have frame shift mutation. Now the frame will be shifted. Okay. The particular frame, the sequence in which that frame is present, that thing is shifted. Just like when we see train accident. Okay. The train will become derailed because of some that bogie of the trains get detached from the rail. Okay. Same thing will happen in your frame shift mutation or Gibbs mutation. So again, it is of two types, in which we have one addition and other one is deletion. Addition of one or rarely more than one nitrogenous base in the structure of DNA, means only one nitrogenous base is adding in the nitrogenous sequence in the DNA sequence, then it is your addition or insertion type of frameshift mutation. Means like this, see this. If this is your sequence AUGCCA GACTAC, okay, in the middle of that somewhere A will come. Okay, now the sequence will become AUGCCAGAACTAC. Whole sequence is being changed. From here to here, whole sequence of is changed, and now different amino acid will form. So this is your addition or insertion type of frameshift mutation. Second type is your deletion. Now if I delete something from the middle of that sequence, it will become your deletion type of frameshift mutation. See, if it is a sequence AUGCCACCCGGG, okay, now if I remove this C from here, okay, now the sequence become AUGCCACCGGG, okay. Now one nitrogenous base is removed. Due to that removal of one nitrogenous base, again different amino acid will code, and this is your deletion type of frameshift mutation. Okay, due to the frameshift mutation, complete reading of genetic code is changed. It leads to the change in the amino acid after the sequence of addition or deletion of nitrogenous base. Means the total reading of protein, the protein which is going to be formed, it is not going to be formed, some other protein will form in frameshift mutation. So frameshift mutations are more harmful as compared to substitution. In substitution, we are seeing that purine is changing its position with purine, or pyrimidine changing its position with pyrimidine, or pyrimidine is changing its position with purine, or purine changing its position with pyrimidine. So that is not that much dangerous. But if a whole nitrogenous chain, whole that your DNA sequence is changed, then it is more dangerous. So your frameshift mutation is more dangerous than your substitution mutation. Okay, so this is your mutation. Now next is your human genetics. Okay, the study and analysis of genetics character in human is called human genetics. Okay, means we are going to study about human characters, different character, different genes present on different sites of chromosome inside the body, and how they will affect, how will they cause different diseases in human. Okay, so eugenesis, that is improvement of human being by applying principle of genetics. Father of eugenesis is Francis Galton. Okay, so these all are different characters and its dominant and recessive traits. Okay, eye color brown and black, that is dominant, and blue color is recessive. Then earlobes, free earlobes mean that ear pinna, it is attached or detached from your lower region of your neck. Then if it is detached, it is free, then it is a dominant. If it is fused, then it is recessive. Then hair, curly hair, it is dominant character, and straight hair in human, it is recessive character. Then cheek, dimple in the cheek, it is a dominant character, while no dimple in the cheek, it is a recessive character. Next, rolling of tongue, normal roller tongue type of tongue you have, then it is a dominant trait inside the human, while if it is non-roller, it is not rolling inside your mouth, when you closing the mouth, it is an recessive trait. Okay, next we have Rh factor, Rh positive, it is a dominant trait, and Rh negative, it is a recessive trait. Next we have PTC, phenyl thiocarbonate tester, means it is a present on your tongue. If it is dominant, then it is taster, and if it is recessive, it is not tester. Okay, next skin pigmentation, normal color, dominant, and this is albino, that is recessive, means whole body is white. Okay, so this is your human genetics, and these are different characters and their traits. Okay, now we have pedigree analysis or anthropology. Okay, in which we are going to study about the different traits, how the different traits go from one progeny to another progeny by the help of pedigree diagram, pedigree chart. Okay, so pedigree analysis is a process of examination the pattern of inheritance of a genetic trait and disease in a families. Okay, means if your father has some disease, it can be happen to you, and then it can be happen to your child, and it can be present in your grandfather and grandfather. So that thing is studied with the help of pedigree charts, and that study is called pedigree analysis. Okay, so in pedigree chart we have diagrams, different diagrams to show phenotypes and genotypes of particular organism and its ancestors, used in human families to track genetic disease. They can also use for any species and any other heritable traits. used to determine the mode of inheritance of particular disease or trait and to predict the probability of its appearance among the offspring. See, pedigree analysis or pedigree chart does not give you confer information that the disease is coming. It only gives you probability that that disease can be inherited by your offspring. Okay. Next is your pedigree analysis, therefore an important tool in both basis research and genetic counseling of an organism. Before the marriage, people do genetic counseling to see what types of recessive genes and dominant genes are present inside the family tree of the both families which is going to be married. Okay, so these are different symbols used in pedigree analysis. First one is your square, that is male, then circle which is for female, then circle is connected by square with a single line, then it shows marriage, and when circle is connected by square with double line, then it is called consanguineous marriage, means marriage between same relatives. Okay, or relative which is living far away from each other. Okay, means marriage in same caste, we can say that. Okay, next, this is the arrangement of your that pedigree analysis, that it should be, generation should be given in Roman number. That's generation 1, generation 2. Okay, then generation 3, 3i sign. Okay, that Roman numbering. Okay, next, that individual, the babies, that one which is a present left side, it is the this is the oldest one, and the present right side, it is the youngest one. Okay, next one is your identical twins. This is a sign in which two same shape is connected by line and forming a triangle like structure, then it is called identical twins. See, identicals and non-identical twins, what is that? Identical twins are those twins which is developed because of zygote division. Zygote, it divided into two halves and it produced your identical twins. Non-identical twins means two ova, two sperms get fertilized at the same time, but the location is different inside the fallopian tube. So that is your non-identical twins. Identical twins sex are always same. Okay, it either can be male male, it either can be female female. But now your non-identical twins, it can be male male, it can be female male, and it can be female female. Okay, next is your affected male and female. It is being boxed size of design. Okay, next is your affected by history, female and male, in which we have zebra lining. Then carrier, it is center dot on circle, and it is always female, never male. You never see a carrier with square. Okay, always it is in circle. Next we have cutted one. This is decreased female and male, means they are RIP, they are dead. Okay, they are not more in the pedigree, they are dead. It is your decreased female and male progeny. So, okay, then if it is that rhombus shape, means Kaju Katli shape, then it is unknown sex. Okay, so we have different inheritable patterns in the that genetics. First we have autosomal dominant, then we have autosomal recessive, then we have X-linked dominant, then we have X-linked recessive, then we have Y-linked, and at last we have mitochondrial. Okay, so first we have autosomal dominant. Okay, so autosomal dominant means for pedigree involves a dominant allele on non-sex chromosome or autosomes. In dominant autosomal and recessive autosomal, we are talk about only autosome, not sex chromosome. Okay, so pedigree involves a dominant allele on non-sex chromosomes or autosome, then it is your autosomal dominant disorders. Okay, it does not skip the generation. Any type of dominant character disease, it never skip the generation. Okay, further we are going to study about dominant X-linked disorders, in which again we will see that skip of generation is not present. Okay, affected parent can give rise to unaffected child, unaffected parent can give rise only to unaffected child. See this by the help of Punnett square. The line is that affected parents can give rise to unaffected children. Okay, this one, and this. This is your affected, this is your affected, this is normal. Okay, so affected parents can give rise to unaffected child. See, this is your affected female, this is your affected male. Okay, then we get first child, it can be affected, second it is affected, third it is affected, and the fourth one is unaffected, small a recessive. Okay, so this is your unaffected child, and unaffected parents can give rise only to unaffected child. See this thing also. Unaffected parents means small recessive. Okay, small a. Both female and males are small a. Okay, then you will see all the progenies are healthy. Okay, because there is no dominant allele present in that. Okay, males and females affected equally because we are talking about autosome, not sex chromosome. That's why in this males and females are equally affected 50-50. Next, no carrier. Okay, in this category there is no carrier because heterozygous dominant and your homozygous dominant both are diseased. Okay, example Huntington disease, neurofibromatosis, polycystic kidney disease, endochondroplasia, familiar hypercholesterolemia. Okay, so first we have your pedigree chart, okay, in which you will see that no generation is skipped. Okay, see this generation get disease, now next will be get. Okay, now when you see here, this is normal child, this is normal child, they got married and all progeny is same. Okay, so the line which is written here, it is hence proof that affected parents can give rise to unaffected children. See, these both are affected parents and it is giving a healthy child to birth. Okay, and when two unaffected parents get married, then all the children are unaffected. Same thing is written here. Unaffected parents give rise only to unaffected children in your autosomal dominant pedigree. Okay, so for first we have Huntington disease. Okay, it is a neurological disease in which your brain ventricles, the ventricle which is present inside your brain, they get damaged, they get accreted because of some enzymes, some proteins, just like PTT is not synthesized by your chromosomes. Okay, due to that, your ventricle is not developed well and it will cause Huntington disease. Okay, you can see here. See, this is your chromosome number 4 in which you have a gene HTT. Okay. Now if this gene get mutated, now your neurons start to degenerate. And that neurons start to degenerate, it will causes atrophy of basal ganglia inside your brain and the ventricles which is present inside your brain. Due to which involuntary movement to start your skeletal muscles is not under your own control. Okay, it will do shaking, it will start to do twitching. Okay, it will not under your control. Okay, second disease we have neurofibromatosis type 1 NF1. Okay, it is a genetic condition that causes changes in the skin pigmentation and tumor on nerve tissue. Okay, this disease is very dangerous. Okay, and its occurrence rate is very low. Okay, in the 2500 people, only one get affected by this disease. Okay, in this disease what happen, tumorous growth will occur at the endings of nerve, due to which the person get a intense pain. Okay, it is called neurofibromatosis type 1. Okay, that cancerous growth can that tumorous growth can be converted into cancerous if that person goes under the situation, under the control or under the influence of carcinogens. Okay, so this is your neurofibromatosis type 1 NF1. Okay, next one is your PKD, polycystic kidney disease. Okay, in which what happened, your kidney get separated, get influenced and get covered by different type of cyst, due to which the renal function of your kidney is not that much proper and at last your kidney get failed. Okay, so in this what happened, in your body there is PKD genes. Okay, that gene is responsible to induce this disease. If that gene get on and it is not get off, okay, then this disease will happen. This is your normal kidney and this is your polycystic kidney, in which many cysts develop over your kidney. Okay, next disease is your achondroplasia feature. Okay, in which what happened, the bone development does not take place. Okay, your body bones does not develop well, it is just like dwarfism. Okay, but dwarfism, it is an hormonal disorder, but in this the disease is genetic. The bone development is not proper. Okay, that the person who having this disease, they are dwarf, they are bony, they are just people who work in the that circus. Okay, you can see here, this is a man who take part in the Games of Thrones movie. Okay, he was also having this disease. Okay, next one is your familiar hypocholesterolemia. Okay, it is the most common genetic metabolical disorder characterized by marketing elevated LDL sea level from the birth, leading to atherosclerotic cardiovascular disease and premature death. Okay, means your coronary artery is blocked with plaque. And due to that accumulation of plaque inside your artery, you will get heart attack, okay, or cardiac disorder, or different disorders related to the heart. Okay, so this is your familiar hypercholesterolemia, in which level of low density lipid is increased, which is not good for your heart. Okay, next is your autosomal recessive pedigree. Okay, now it is time for small a. Okay, means these and this is your healthy, okay, and this is your diseased. Okay, capital A, small a is normal, and small a is disease. In this autosomal recessive pedigree involve recessive alleles on non-sex chromosome. Okay, skip the generation, it skips the generation. Okay, unaffected parents can give rise to affected children. See how an unaffected parents give to birth to an affected children. Okay, you have two parents, capital A, small a, capital A, small a. Okay, now that they get reading, this is your normal, this is your normal, this is your normal, this is your infected, this is normal but carrier, and this is your normal but carrier. Okay, so this is your autosomal recessive disorder. Okay, males and females affected equally, again because it is an autosomal disorder, it is affecting to your autosome, not your sex chromosome. That's why male and female both are affected same, 50-50%. Okay, presence of carrier, as I said, a and small a, the heterozygous condition, is a carrier in this disease. It does not get disease, but it carries the gene which can cause the disease in the progeny. Okay, cystic fibrosis, sickle cell anemia, Tay-Sachs disease, and phenylketonuria are the example of this autosomal recessive disorders. Okay, and this is the pedigree in which you can see the generation is being skipped. This generation have the disease, now this generation does not have. Then again this generation have the disease. Okay, so autosomal recessive disorder, they skip the generation one by one. Okay, if pedigree in which you will find that all the generations are affected, then it is your autosomal dominant disorder. And if you find a pedigree in which one generation is skipped, then again generation is skipped and get disease, then it is your autosomal recessive disorder. Okay, so first example we have cystic fibrosis. Okay, in this disease what happens, your respiratory tract and GIT tract get filled with mucus. All the glands which is present in your alveoli respiratory tracts and the glands which is present in your respiratory tract had been blocked because of the mucus accumulation inside that, and due to which there is failure in respiratory system and your GIT. Okay, so this is the gene for cystic fibrosis is present on chromosome number 7, in which thick sticky mucus blocks the airway and thick sticky mucus blocks the pancreatic and bile juice, which does not reaches to your target site, due to which you will get diseases of GIT and respiratory system. Okay, so this is your cystic fibrosis. Next one is your sickle cell anemia, which is the most common disease, which is we going to study in this chapter also again. This is the disease of blood circulatory system, in which your blood is changes due to the mutation in the sequencing of beta chain of hemoglobin. The sixth position of beta chain has been replaced by glutamic acid in the valine displaces the glutamic acid. Means in the normal RBC you will find that glutamic acid is present, and in abnormal sickle cell RBC you will find that there is valine is present. Okay, so this is your sickle cell anemia, in which glutamic acid is exchanged with valine, due to which the shape of RBCs changes in low oxygen density or low oxygen tension. When the oxygen in the atmosphere become down, your normal RBC will convert into sickle cells when you have sickle cell anemia. Okay. So next is Tay-Sachs disease. It is a very rare disease Inherited and fatal genetic disease that destroy nerve cells in the brain and spinal cord. Tay-Sachs disease is caused by the deficiency of enzyme hexoaminidase A, which result in the accumulation of GM2 glycosidase. Okay, means in this disease what happen? Your nervous system, your spinal cord, your central nervous system get collapsed, and the person will die due to the neurological disorder. Okay, next one is your phenylketonuria. It is again present in your NCERT. Okay, PKD. Okay, sorry, PKU, in which what happen? The phenylalanine, an amino acid which is prepared inside your body, it will convert into tyrosine with the help of phenylalanine hydrolase is not present. Okay, the enzyme which is your phenylalanine hydroxylase, it is not present inside your body, due to which phenylalanine is not converted into tyrosine, and your phenylalanine start to accumulate inside your body, and it causes neurological disorder, digestive disorder, and multiple organ failures. Okay, so these all are the symptoms of your phenylketonuria. First, mental retardedness. It arrests or it acts on your nervous system. Second, organ damage. Okay, due to the accumulation of phenylalanine inside the organs, it will start to damaging, hurting your organ systems. Next one is unusual posture. Okay, the person who having phenylketonuria, they cannot they cannot go and move straightly, they will move like this. Okay, next is your musty odor from skin and urine. The smell of phenylalanine is just like mustard oil. Okay, so the person who having phenylketonuria, their body smells like mustard oil factory or a mustard oil plant, and their saliva, their excretory organ, their excretory substances like urine, it will smell like mustard. Okay, next one is lighter skin, hair and eye, because this thing also interfere with your melanin. Okay, melanin pigmentation is also interfered by this phenylalanine. If it is present excessively inside the body, then the person skin become lighter in the tone, hair blackness goes dim. Okay, and the eyes does not have that black color if the person have black eyes that much. Okay, delayed mental and social skills, the person cannot recognize things very fastly. Okay, next one, head size is much smaller than normal. The size of the bones which is present inside the head, it is smaller. Next, hyperactivity, because of that neurological disorder, it is the hyperactivity will be present inside the body, and jerking movement of the arms and legs. Okay, that arms and that legs always it will doing jerking movement, it will always in pitching movement. Okay, so that is your phenylketonuria. It is an again autosomal recessive disorder. Okay, this is how this thing will happen. This is your phenylalanine, and it should be converted into tyrosine inside your body. In a healthy person, phenylalanine convert into the tyrosine by the help of enzyme phenylalanine hydroxylase. But if this thing will happen, phenylketonuria, because of the chromosome number 12 mutation, if this thing will happen, this enzyme will not form, and due to which your tyrosine is not formed, and your body will start to accumulating phenylalanine in excess. Okay, next is your sex-linked X-linked dominant disorder. Now we are going toward the sex chromosomes. Autosome done, now sex chromosome. Okay, so involves a dominant allele of X chromosome. Okay, do not skip the generation. Again, it is a dominant disorder, so it does not skip the generation. Females more frequently affected because in this female have XX. Okay, that's why it is affects female more than male. Okay, daughters of affected father will be affected. Okay, daughters of affected father will be affected because in the daughter, the one X chromosome is coming from your father, and that father X chromosome is affected. That's why the daughter is affected in this disease. Never passed on to the father to son, because in son the X chromosome is coming from the mother. Okay, that's why this sex link or X link dominant disorder never comes from father to son. Okay, because father does not give that X chromosome. That X chromosome it comes from your mother side. Okay, example, hypophosphatic retics, ornithine transcarboxylase deficiency, oral facial digit syndrome type 1, and fragile X syndromes. Okay, these all are the example of sex linked or sex X linked dominant disorder. Okay, and this is the pedigree in which you can see it does not skipping the generations. Generation 1 having the disease, generation 2 having the disease, generation 3 have the disease. Okay, first, so example number first is your refractory rickets. Okay, it is a genetic form of rickets which happens in the person who having this chromosomal disorder. Okay, next is your ornithine transcarbamylase deficiency. Okay, you know that ornithine cycle runs inside your liver. it will purify, it will convert your toxic substance ammonia into urea. Okay, so what happened in this disease, that OTC, okay, that enzyme OTC which is responsible for the conversion of carbamyl phosphate into citrulline, it is absent. If this thing is not converted, carbamyl phosphate is not converted into citrulline, okay, so urea formation not take place, due to which what happened, ammonia start to accumulate inside your body and it will cause jaundice or different liver related diseases. Okay, next is your fragile X syndrome. It is just like Down syndrome and it affects more male than female. Okay, next we have orofacial digital syndrome type 1. It is a rare X-linked dominant disorder that affects face, mouth and finger. Okay, in face it affects lip, in mouth it affects your palate, and in finger it will affects your digit. Okay, this is your orofacial digital syndrome type 1. Next we have X-linked recessive pedigree. Okay, in which the trans that skips the generation and the gene of disease is present on recessive allele. Okay, so it involves recessive allele on X chromosome, skip the generation, males frequently affected, son of affected mother will be affected. Okay, because that X which is carrying inside the mother, it will goes to the son. Okay, next, presence of female carrier. In this you will find female carrier. Okay, three genotype will find, in which one is your dominant one, homozygous dominant, which is normal. Second one is your heterozygous, which is a carrier, and third one is your homozygous recessive, which is diseased one. Okay, example of this, hemophilia A, red green color blindness, and Dutchinson muscular dystrophy or muscular dystrophy. So first we have hemophilia. Okay, hemophilia, as that you know, hemophilia is in blood that in which the blood clotting does not place. Your blood will flow very fastly. Normal blood clotting time is 1 to 5 minutes, but in this disease, hemophilia, it will convert into 24 hours. Okay, in that time, whole blood will that flow out from your body, and the blood deficiency will occur inside your body. Hemophilia is of three types: hemophilia A, hemophilia B, and hemophilia C. In hemophilia A, you have the deficiency of that clotting factor number 8. In that hemophilia B, you have the deficiency of clotting factor 9, that is Christmas factor. That's why it is also called Christmas disease, hemophilia B. And in hemophilia C, you have the deficiency of 11th factor of that clotting factor 11. Okay, next is your Dutchinson muscular dystrophy. Okay, it is a muscular disorder in which the muscle cannot perform their adequate functioning. They cannot move by your own will. All the muscular system of your body get collapsed due to the that Dutchinson X chromosome, that Dutchinson gene which is present on the X chromosome has been get mutated in this disease. Okay, initially in the 2 year of age, the disease does not show its symptom, but by the age increases, by the age that moves to 10 years or 15 years, you will see that muscles are not doing its work well. It will start to degenerating. Our famous scientist, that is your Stephen Hawking, having this disease. Okay, now we will see the pedigree of this disease. This is the one pedigree of this disease. This is the second pedigree of this disease, and this is the pedigree of hemophilia. Okay, hemophilia, this is the pedigree of royal family of England. Okay, Victorian Queen of England, that is the pedigree in which you will see that Queen Victoria is the carrier of hemophilia. Okay, and it will transfer inside the family tree, and you can see this guy having this woman having the hemophilia, this one having the hemophilia, this one have the hemophilia, this one have the hemophilia, this one have this have, but they all are carrier. Okay, that does not showing the disease, but they are carrier. Affected one is fully colored one. Okay, that fully blued one squares, that are your colored one, that having the hemophilia. Hemophilia mainly occur in males, less occur in female. Okay, so next is your Y-linked pedigree. Okay, in this you will study that different diseases which is affecting, that occurring inside the male only, not in female, because Y chromosome, it is only present in male. Okay, so it involves alleles on the X chromosome. It do not skip the generation because Y chromosome, it is only present in male. Okay, only males are affected, pass from father to son. Okay, father to son it will go, because the Y chromosome, it is coming from your father. Okay, so example is Hodgkin disease. Then retinitis pigmentosa, Sertoli cell only syndrome, non-obstructive spermatogenic failure. Okay, all these diseases are present inside the male only, not in female. Okay, so first we have Hodgkin lymphoma, also known as Hodgkin disease, is a rare and highly curable cancer that develop inside the lymph system. Okay, the lymph system in the armpits and the legs, this disease will occur. Okay, in which the cancerous growth will occur inside the lymphatic tissues. Okay, second one is your retinitis pigmentosa. In this disease, what happen? The retina of your eye get inflated, get diseased due to the accumulation of different material inside, just like bone spicules or different materials get accumulated inside the eye of human in that disease. Okay, and which you have the symptoms: loss of night vision. In the night you cannot see the light. Okay, loss of peripheral vision, which can lead to tunnel vision, means your eyes can see up to the range. Okay, but in this disease only you can see the straight line. Okay, just this, that's why it is called tunnel vision, straight vision. Okay, next, sensitivity to the bright light. Okay, your eyes get sensitized to the brighter light. If bright light come in front of you, your eyes get sensitized. Okay, next one is loss of color vision. Okay, the color vision, the color you can see by your eyes, that colorful world you can see by your eyes has been lost because of the damage of cone cells. Okay, the person who having this disease, उनकी जिंदगी से रंग उड़ जाते हैं. Okay, they cannot see the color of the world. Okay, so this is your normal eye retina, and this is the attenuated blood vessel, this one. Okay, then pigmentary bone spicules, this one, and then waxy appearance of disc, this one. These three are the causes of this disease in human, and it is a genetic disorder. This thing can also occur physiologically in females also. If they can be affected by some medicines or some infection take place inside the eyes of female, then this disease can also occur in female. But genetically this disease only occur in male. Okay, next one is your Sertoli cell only syndrome, in which your Sertoli cell does not get mature, and due to that immature Sertoli cell, it does not create spermates and sperms. Okay, in which your male become infertile. Next one is your non-obstructed azoospermia, in which testes does not come out from the scrotum and go down inside the scrotum. Okay, means it will present above in the your abdominal cavity, due to which you will does not have the power to produce sperms, and the progeny does not go ahead. Okay, so this is the azoospermia. It is of two type: obstructive and non-obstructive. Obstructive, many sperms throughout the testis, spermal retrieval always successful. Okay, in the obstructive azoospermia, there is little amount of sperms are present, but non-obstructive, that azoospermia, you have two type: partial and total. Total is that condition in which no sperm is enter in the testes. Okay, testis के अंदर कोई भी sperm नहीं बचता है. Okay, next one is your Mendel genetic disorders or Mendelian disorder, chromosomal disorders. Okay, that is your hemophilia, sickle cell anemia, color blindness, phenylketonuria, thalassemia, cystic fibrosis that we studied now. Okay, and chromosomal disorder, that is your Down syndrome, Tunnel syndrome, Klinefelter syndrome, which we have to study. This is done in the that pedigree analysis. I have studied that thing, so we do not have to study again that thing. Okay, so this is all your Mendelian disorders. Okay, so this is your hemophilia, royal disease, which is happen in the Great Britain family. Okay, second one is your color blindness, in which you cannot see color like green and red. Okay, if it is a normal color vision, in which you see green color, yellow color and red color, but a person who is color blind, he will see green color as light green color, yellow is yellow normal, and the red color is just like yellow color. Okay, there is a one ishara, this is called ishara chart, by which that eye doctor recognize that person is color blind or not. If you can see this 74 written by green color, then your eye is normal, and if you cannot see 74 in the green color, means you are color blind. Go to your eye doctor. Okay, next one is your sickle cell anemia. Already I tell that it is disease in which the hemoglobin chain, the beta chain of hemoglobin, the sixth position, it which is present in normal RBC, which is presented by glutamic acid, on the place of that valine will come and the mutation will occur, which is your point mutation. Okay, now your normal RBC, when that level of oxygen decrease in the atmosphere, it will become sickle like Chandrama shape. Okay, next one is your phenylketonuria. Again, I said that in normal person, dietary protein will convert into phenylalanine, and by the help of enzyme phenylalanine hydroxylase, it will convert into tyrosine. and tyrosine help in melanin protein and dopamine synthesis. But in this disease, what will happen? Phenylalanine hydroxylase is not present inside your body. Phenylalanine not convert into tyrosine, and phenylalanine start to accumulate inside the body, and it will convert into phenylpyruvic acid, which will inert, which will affect bad effect on your neural system and the multiple organs. Okay. Next is your thalassemia. Okay, it is an again blood disorder in which your blood get mutated due to the mutation in alpha and beta chain both. Okay, your thalassemia, it is a quantitative problem, and the sickle cell anemia is a qualitative problem. In thalassemia, only the quantity get changed. Okay, alpha, beta, both chains are mutated. Okay, but in sickle cell anemia, qualitative change means on the place of glutamic acid valine change, means the quality of your blood changes. That's why sickle cell anemia is a qualitative problem, while thalassemia is a quantitative problem. Both are bleeding disorders. Okay, so this is your person who having thalassemia. It is of two type: alpha thalassemia, in which alpha globulin chain is being deleted and being mutated, and this is beta thalassemia, in which your beta globin chain is being mutated, and it is controlled by, it is happened due to the mutation in chromosome number 11. Okay, next one is your chromosomal disorder. Okay, in which we are going to study Klinefelter syndrome, Down syndrome, and Turner syndrome. Okay, so first we have Down syndrome, okay, also called Mongolism, okay, in which what has happened, that your normal set of chromosome having 21 two times, but in this disease the 21 chromosome is present three times. That is your triploidy, okay, triploidy, in which 21 chromosome having three sets on the place of two, it is having three. Okay, then this is your Down syndrome. Okay, so it is 41A plus XX or 45A plus XY. It can happen in male, it can happen in female. Okay, in normal male it is 44A plus XX, 44, 46. Okay, 44, 45, 46. Okay, normal. 44, 40, 45 and 46 normal. But in Down syndrome, 45, 46, 47. Okay, 46, 47 chromosomes are present inside the body of the male or female. Then it is your Down syndrome, and it is happening on your 21st chromosome, trisomy of 21st chromosome. Okay, so the features of Down syndrome is that short statured with small round face. Okay, the person who having Down syndrome, they are cute in the facing. Okay, they are very innocent when you see them. Okay, next, broad flat face. Okay, their face is straight. Okay, they do not have any curvature which we have present in our normal person face. Okay, furrowed big tongue, partially open mouth. They are open always, their mouth always remains open. Okay, their saliva dropping out from their mouth. Second, furrowed big tongue. Okay, their tongue is furrowed. Okay, it contains some furcation in the middle of the tongue. Okay, then broad palm with characterized palm, Cimmerian cleats, many loops on the fingertip. Next, retarded physical, physiomotor, and mental development. Their mental development is very less. Okay, they are mental. They are mostly of the persons who having Down syndrome. You can find them in mental asylum. Okay, and at last, that having congenital heart disease, their age is 25. After 25, they may be die because of congenital heart disease. Okay, next one is your Klinefelter syndrome. Okay, it is the presence of additional copy of X chromosome in male. Okay, genetically constitution 44A, this is normal your autosome count, and on the place of normal XY you have one additional X chromosome. Okay, this is your in this disease many female like character will develop inside the male body because of the addition of the additional X chromosome. This is the karyotype of a person who having Klinefelter syndrome, in which you will see that two X chromosomes are there. The normal male having only one X chromosome and one Y chromosome, but so the characters of Klinefelter syndrome is that overall the body is of male, but the character which is present inside the body, it is of female. Okay, development of breast, gynecomastia will happen. Okay, sterile male, they cannot do reproduction, and mentally retarded, they are mentally abnormal, they are mentally very retarded. Okay, they have aggressive nature, they will always ready to for fight. Okay, next one is your Turner syndrome. Okay, Turner syndrome, this will affect in female. Normal female have two X chromosome, but in this disease, what happened? One X chromosome is deleted. There is no presence of one X chromosome. On the place of that, there is nothing. Okay, then this is your Turner syndrome. Okay, this is the absence of one of the X chromosome in the female. Genetic constitution 44A plus XX, that is your normal female. But in this disease, 44A plus XO. Okay, so the features of this is sterile ovaries and rudimentary, means the ovaries are not functional in females. Lack of other secondary sexual characters, the other secondary sexual character which is present inside the female, they are absent. The females are dwarf, they are small in the short heighted ladies they are, and they are mentally retarded. Okay, they also have congenital, that congenital heart disorder, in which they can only live up to the age of 25. So your Down syndrome and Turner syndrome having congenital heart disease, they can die after the age of 25. But in Klinefelter syndrome, you will see there is no congenital heart disease, they are only sterile, and the features of male is disappear on the place of that female features come inside the male. Okay, so the person who having Klinefelter syndrome, they do not die, while the persons who having Down syndrome and Turner syndrome, they will die due to that congenital heart disease. Okay, many other features are there, that is short stature, means that they are dark, low hairline, their hairline is very low. Okay, shield shaped thorax. Okay, normally we have a thin thorax, but in this disease, what happened, the head and the neck ratio, it is same, means there is your head and neck will be going straight down. Okay, next one is widely spaced nipples. Okay, they are not present in the center of the chest, they are dispersed from the center area. Then shortened metacarpal, because in this there is no development of body, short stature person, that's why their metacarpal is very short. Then small fingernails, their fingers nail are very small due to that the not development of the particular growth hormone inside the body. Next we have brown spots in the different parts of the body, they having brown spots. Then characterized facial features, means they have the straight head, that flattened head, just like we have in Down syndrome also. Then folds of skin, they have juria in the different area of their face and their neck. Okay, wrinkles they have, okay, in the form of folds of skin. Next we have constriction of aorta, means they have congenital heart disease. Poor breast development, normally the female have better development of breast, but in the females who having that Turner syndrome, their breast is not developed. Then elbow deformity, as that growth hormone is very less inside the body of that female, so that elbow development having deformities. Okay, and substance of the most important thing which is happening in Turner syndrome is that their ovaries are not functional, they cannot give birth to new ones. Okay, and no menstrual cycle, that if the ovaries are not functional, automatically no menstrual cycle. Okay, so that is your principle of inheritance. Okay, now we take 5 minute break, next we will come with reproduction in human. it's time for human reproduction, the most interesting chapter of biology. Every students know that. Okay. So first we start with male reproductive system. Okay. So male reproductive system, it is divided into two parts. There is your parent testes, accessory ducts, accessory glands, and external genitalia. Okay. In accessory ducts we have retitestes, vasa differentias, epididymis, and vas deferens. Okay. These all are ducts. Okay, these all are pipelines which is present inside male reproductive system. Then we have glands, prostate gland, seminal vesicle, and Cowper glands, or we call also bulbourethral glands. Okay, prostate gland, this is one present. Okay, only one. Then you have seminal vesicle, that is two in number, and Cowper glands, that is again two in number. Okay, one pair, two. Okay, and then we have external genitalia which we call penis. Okay, so scrotum. Okay, it is also your part of external genitalia in which your testis lies. Okay, so the testes are situated outside the abdominal cavity with a pocket called scrotum. Okay, this is a pouch suspended from the groin to has two compartments separated by scrotal septum, each housing one testis. Okay, it is just like a structure. Okay, a bag-like structure in which testes are present. Okay. Next, the scrotum help in maintaining the low temperature of testes. That's why it is present outside. Okay, because for the process of spermatogenesis, you need low temperature, a 2 to 3 temperature, which is less than your body temperature required for the spermatogenesis. As you know, the normal body temperature is 37 degrees Celsius. For the process of spermatogenesis, you need 35 to 34 degrees Celsius temperature. Okay, so that's why testes are present outside the body of your human male body, and it is present in scrotum. Okay, so the scrotum remains connected with the abdominal or pelvic cavity by inguinal canal. Okay, the backside of that you have find that inguinal canal by which your scrotum is connected. Okay, each testis is suspended from the body wall by the spermatic cord, which penetrates into the pelvic cavity through inguinal canal. Okay, this is your scrotum septum, and here somewhere you having find that spermatic cord. Okay, inguinal canal is the route through which the testis originally descend into the scrotum from the pelvic cavity before the birth. Okay, when the male baby in embryonic stage, okay, the testis is not present outside the body, it is present inside the inguinal canal. Okay, now when the that baby boy comes out, you will find that testes will come out from the inguinal canal and it will hang inside the scrotum. Okay, next, each spermatic cord contains the vas deferens, testicular nerve, testicular artery, and testicular veins. The skin of scrotum is separated in the midline by a ridge called raphe. Okay, in the middle of that testes, you will find a line which divides that both parts of scrotum, which called raphe. Okay, next we have testes. Okay, it is your primary sex organ. Okay, where your spermatogenesis process take place. Okay, in adult, each testis is oval in shape with length of about 4 to 5 centimeter and width about 2 to 3 centimeter. A testis is covered by three coats. These are tunica vaginis, tunica albicina, and tunica vasculosa, respectively from outside to inside. So the outermost is your tunica vaginales. It is a partially covered. The testis has parietal and visceral layers. It's having two layers. It is bilayered structure. Okay, next we have tunica albugina. It is a dense white fibrous cord. It is posterior border. It is thickened to form a vertical septum, which we call mediatum testis. Okay, it's cut the testes into the two parts. In the interior border extend inward, forming a septa that divide the testes into a series of internal compartments called testicular lobules, in which your seminiferous tubules are present. Okay, then we have tunica vasculosa. It is the vascular coat lining the testis lobule. Each testis has about 250 compartments called testicular lobule, in which your spermatogenesis process take place. Each lobule contains 1 to 3 highly coiled seminiferous tubule in which sperms are produced, means your spermatogenesis process take place. In the seminiferous tubules of testes open into the vas efferentia through straight tubules called retitestes, and the vas efferentia leaves the testes and open into epididymis located along the posterior surface of each testis. Means that is a pipeline system which is present inside the male reproductive system, which carries sperm from one point to another point. Okay, next part is your epididymis. Okay, Each epididymis consists of a single convoluted tube about 6 meters long, which has three parts. One is head, then body and tail. Functionally, the epididymis is the site of sperm maturation, the process by which sperm acquired motility and ability to fertilize an ovum. In addition, the epididymis store sperm temporarily. Means in your epididymis is the temporary house of sperm where it is stored for some time, which stops for some times. Okay. Next we have vas differentia. Okay, it is again a duct in which connected with your epididymis. So the epididymis lead to vas differentiation to the abdominal and loop over the urinary bladder. Okay, in kidney you have urinary bladder by which this vas differentia it will turn around that ureters of your urinary bladder. Okay, before entering the prostate, the vas deferens dilate to form ampulla that receive duct from seminal vesicle and open into prostatic ureter and ejaculatory duct. Okay, means the ending of your vas differentia it's open into an ampulla which connects to its seminiferous that seminal duct or the seminal vesicle. Okay, functionally vas differentia conveys sperm during the sexual arousal from the epididymis towards the urethra by parasitic contract of its muscular walls. Okay, next we have male accessory glands. Okay, the glands which is responsible for the nutrition of sperm and different material which is present inside the semen, that like fructose, calcium, vitamins which is present in the semen. Okay, which is produced by these glands. So first we have seminiferous vesicle. Okay, seminiferous vesicle are paired glands that lie at the dorsolateral side of base of the urinary bladder. Okay, you can see here this is your seminal vesicles. Okay, it is present both sides, this side and the and that side also. Of the back side of this area you can also see seminal vesicle. Okay, so each seminal vesicle with duct joined ampulla of the vas deferens to form ejaculatory duct. Ampulla of your epididymis with your seminal vesicle forms a junction in area which we call ejaculatory duct. Okay, these glands secrete an alkaline viscous fluid called seminal fluid, rich in fructose, an important nutrient for sperm. This fluid is contained fibrinogen, prostaglandin also. Okay, a majority of volume, about two-thirds of the total volume of seminal plasma is secreted by seminal vesicle. It is a very important gland of male reproductive system. The more than half part of the male reproductive is secreted by seminal vesicle. Next we have bulbourethral gland or Cowper glands. Okay, these are paired bulbourethral glands lied on the either side of the membranous urethra. Okay, the membranes, now we are going towards the urethra. Okay, so this gland are present on the both side of your membranous urethra. Okay, but their ducts empty into the penile urethra. It is present inside near to your membranous urethra, but the secretion of your Cowper glands it's empty inside the penile urethra, means it pours its secretion inside your penile urethra. Okay, during sexual arousal these glands secrete an alkaline fluid in the urethra that protects the passing sperm by neutralizing acid from the urine in the urethra. This secretion of bulbourethral gland also help in the lubrication of the penis. See, the secretion which is coming from your Cowper glands, it's help to make your sperm vital inside the acidic nature of the that reproductive tract, because in male that tract of reproduction and that urination is same. Okay, so make that tract basic, this gland secretes its secretion. Okay, secondly its second function is that it lubricates penis, means it make that intercourse without any friction. The friction which is coming inside the intercourse it will reduce by the help of bulbourethral gland secretion. Okay, next one is your prostate gland. Okay, it is an unpaired gland, lies below the urinary bladder and surrounded the prostatic urethra. Okay, it surrounds your prostatic urethra and it is the largest gland of that male reproductive system. The alkaline milky secretion of this gland make up about one-third of the total volume of seminal vesicle. Okay, the secretion enters the prostatic urethra through several tiny duct. Prostate fluid contains calcium clotting enzyme and profibrinogen. Okay, the white color of your semen it is because of prostate gland. Okay, next one is pathway or transport of sperm in that reproductive system. How your sperm flow inside male reproductive tract, that is the flow chart. Okay, first it is produced in your seminary fluid tubules, then it will go in straight tubules, after that it will go in reta testis, then in your vas efferentia, from vas efferentia it will go in epididymis, then from epididymis it will go in vas deferens, From vas deferens it will go inside the ampulla, then ampulla to educatory duct, then urethra, in which it will first go to the prostatic urethra, then membranous urethra, then penile urethra, and then out from the urethral meatus. Okay, inside female reproductive tract. Next, the semen. Okay, the material in which your sperm travels, the medium in which your sperm travel, that is your semen. So the seminal plasma along with sperm constitute equals to semen. Okay, pH of semen is 7.35 to 7.50, slightly alkaline, because it have to survive in the acidic pH nature of your female reproductive tract. The female reproductive tract pH is acidic. That's why the nature of semen is alkaline, slightly alkaline. Normal sperm count in semen is 20 to 120 million sperm per ml of semen. Okay, the human male educate about 200 to 300 million sperm during coitus, of which of normal fertility is at least 60%, and 40% should be of, no, must be vigorous, means they should be moved very fast. 60% should be of particular size and shape, proper size and shape, and 40% should be of high vigorous, means it should be highly mobile. Okay, next there is some condition related to the sperm. First we have azoospermia. It is a condition in which there is either absence or near absence of sperm in the semen, means your semen does not contain sperm, then that condition is called azoospermia. Then we have asthenospermia, in which that reduced motility of sperm is seen. Okay, means your sperm is not able to move, they do not have that much power to move, their flagella is not that much functional to move, then that condition is called asthenospermia. Next we have teratospermia. In this condition, characterized by presence of sperm with abnormal morphology, means their shape and size is very abnormal, and that term is called in medicine tetrospermia. Okay, so we have three terms. First one is azoospermia, no sperm. Then asthenospermia, means that sperm does not have adequate mobility, and last one is your teratospermia, in which the sperm cannot have, that do not have the proper size and shape. Okay, next we have penis. Okay, penis is the male external genitalia. Okay, penis is an erectile copulatory organ. It is made up of special tissue that help in erection of penis to facilitate insemination. Okay, under the skin of the penis contain three columns of erectile tissue, that is your two cylinder of corpus cavernosa placed dorsally on one cylinder of corpus sporongium along the ventral side. See, it is look like this. This is your two cylinder. Okay, and then you have one cylinder. In the middle of that, here your urethra come out. Okay, that secretion of urethra come out from this area. Okay, you can see here in diagram also. This is your erect penis. Okay, when penis is erect, in erect condition. Okay, so you have dilated dorsal veins which is present upside. Okay, then we have cavus cavernosa filled with blood. Then arteries increase, means when the penis get erect, the arteries will be get higher, means it will be filled with full of blood. Then we have compressed urethra, which is present in your corpus spongiosum, which is present on the ventral side. Okay, and when the penis is flaccid, means it is not in erectile condition, then the corpus spongiosum and urethra, it is opened. Okay, it is little bit open. Okay, next we have histology of seminiferous tubules. Now we are going microscopically and see the seminiferous histology, how the seminiferous tubules look like. Okay, so seminiferous tubule consists of tunia purpurica made up of fibrous connective tissue, well defined basal lamina, and complex germinal epithelium. So the epithelium which is present in your seminiferous tubule, it is germinal epithelium. Okay, it is made up of cuboidal cells. Okay, it is germinal in nature, means germ cells is produced by this epithelium. Okay, not somatic cells. So each seminiferous tubule is lined on the inside by germinal epithelium, which has two types of cells called cuboidal cells or male germ cell or spermatogonium, and columnar shaped cells which is called Sertoli cell or sustentacular cells. Okay, the male germ cells undergo meiotic division, finally leading to sperm formation. Okay, your cuboidal cell, it goes under the meiotic division to come and convert into sperm, while that columnar cells, Sertoli cells, it will provide nutrition to developing sperms. Okay, so Sertoli cell provide nutrition to the germ cell, therefore these cells are also called as nurse cell. Okay, so some other function of Sertoli cell to act basis of blood testis barriers. Okay, to phagocyte the injured dead sperms. Okay, phagocytic action also present of that your Sertoli cells, and the most important function of your Sertoli cell to secrete an androgen binding protein ABP that concentrate testosterone in semifinous tubules, some hormones and some factors which help in the process of spermiogenesis to synthesize estrogen from testosterone. Okay, the region outside the semifinous tubules called interstitial space containing small blood vessel, intestinal cells or Leydig cells. Other immunological component cells are also present. Macrophages, mast cells, these all are also present inside that outside area of your seminiferous tubules. Okay, Leydig cells synthesize and secrete testicular hormone called androgen. Okay, that androgen is secreted by your Leydig cell, and Sertoli cell is responsible for secreting of androgen binding protein which help in maturing, giving nutrition to developing spermatids. Okay, so this is your TS diagram, transfer section of testes, in which we use say developing spermid, then it is your Sertoli cells. Okay, that is your spermatocytes, and this is your spermatozoa, and outside you will see that blood vessel and that Leydig cells. Leydig cells secrete your androgen which will go inside seminiferous tubules and it will bind with your Sertoli cells secreted protein, androgen binding protein. It will give nutrition to your developing spermatids. Okay. Next we have hormones of male reproductive system. What are the different hormones secreted by your male reproductive system? So first we have GnRH, gonadotropin releasing hormone. Okay, GnRH is an hypothalamic hormone. It is secreted by your hypothalamus in brain. Okay, GnR act as the anterior pituitary gland and stimulates secretion of two gonadotropin luteinizing hormone LH and FSH follicle stimulating hormone. These both hormone is responsible for the functioning of Sertoli cell and your Leydig cell, and it is also formed inside the female body also. Okay, so luteinizing hormone, it acts on Leydig cell and stimulates synthesis and secretion of androgen. If your hypothalamus does not secrete that GnRH, and GnRH does not go and act on anterior pituitary, and that LH and G and your FSH does not come out from your anterior pituitary, then your that Leydig cell does not get activated. Okay, the reason of activation of your Leydig cell, it is LH hormone. Okay, next is your FSH, it acts on your Sertoli cell. So now we get 2 points, that LH it acts on your Leydig cell and FSH it acts on your Sertoli cells. Okay, then testosterone is the one of the androgen, it stimulates the process of spermatogenesis. One of the other function of testosterone is descending of testes during the intrauterine time. Okay, when the male baby is present inside the womb of the mother, the testis which is descending down with that inguinal canal, it is because of testosterone. Testosterone is not there, then that testis will not come out from that inguinal canal into the scrotum. Okay, then we have inhibin. Inhibin is secreted in the response of excess spermatogenesis and suppress FSH synthesis. Okay, see, in our body there is different mechanisms which we call negative feedback system and positive feedback system. If the hormone secretion is over, okay, means if it is over the limit, then your negative feedback system is activated and it will go and act on the hypothalamus. Then hypothalamus send the signals to your lower that anterior pituitary, and due to which anterior pituitary will stop the secretion of FSH, and now your Sertoli cell does not get FSH and does not have that power to give nutrition to developing sperm. Okay, so that function is done by inhibin hormone. Okay, and this is your hormone action on male reproductive system. See, hypothalamus is there. Okay, hypothalamus releases GnRH. Okay, now this GnRH acts on your anterior pituitary, and anterior pituitary start to release FSH and LH. FSH act on Sertoli cell and it will start doing spermatogenesis by the help of ABP, androgen binding protein. Now LH, it acts on Leydig cell, and Leydig cell secrete testosterone. Testosterone from Leydig cell and ABP from Sertoli cell come together and do the function of spermatogenesis. Now, if that process of spermatogenesis is sufficient, means sufficient sperm is produced, now your body do not need that much sperm. Okay, then inhibin will come out. Okay, and this inhibin which is coming out from your Sertoli cell, it will act on your anterior pituitary and stop the secretion of FSH. This is your negative feedback system. Okay, means when one hormone is increasing, to control the release of that hormone, your body start negative feedback system, and in the testes, that thing is controlled by inhibin, and inhibin send the signal to your anterior pituitary to stop the secretion of FSH. Okay, this is your male reproductive system, how your hormone acts. Okay, next is your human female reproductive system. Now we will talk about female reproductive system. Okay, so human female reproductive system consists of primary sex organ, that is your ovaries, and secondary sex organ, that is your duct system, and that in which your fallopian tube, uterus, cervix, and vagina come, and external genitalia and mammary glands. All these are come under your human female reproductive system, in which mammary gland is not a organ, it is a secondary sexual character in female. Okay, so paired or ovaries, accessory duct, that is your oviduct, or also called as fallopian tube, uterus, and vagina. Okay, and external genitalia, that is your mons pubis, vestibule, hymen, and clitoris. Okay, so we are going to one by one and see that. So first we have ovary. Okay, ovary is the primary female sex organ that produce female gamete, ovum, and several steroid hormones like estrogen and progesterone. Okay, the ovaries are located on the each side of lower abdomen. Okay, in female it is present on the both side of lower abdomen. Okay, each ovary is about 2 to 4 centimeter in length and connected to the pelvic wall of uterus by ligaments. Okay, then we have female accessory ducts. In the first we have fallopian tube, or that your oviduct. Okay, a fallopian tube extends from the periphery of each ovary to the uterus. It is muscular tube about 10 to 12 centimeter long and situated above the behind the urinary bladder. Means it is present above and behind your urinary bladder in female. Okay, the part of fallopian tube closest to the ovary is funnel shape called infundibulum. Okay, the age of infundibulum possesses finger-like projection called fimbriae that help in collecting the ova during the ovulation. Okay, when ovulation process take place, ovum come out from your ovary and it is collected by finger-like projections called fimbriae. Okay, the infundibulum lead into the wider part of fallopian tube or oviduct called ampulla, and then ampulla last part of the fallopian, that then ampulla vents to your isthmus, which is the last part of your fallopian tube. Okay, this is your that female reproductive system, in which you will see there this is ovary and these finger like projections is your fimbriae, and this is your fallopian tube. Okay, and this part which is near to the ovary is called infundibulum, middle part is called ampulla, and the last part of your fallopian tube is called isthmus, and the area which is present between ampulla and isthmus is called ampulla isthmus junction, where your fertilization take place. Okay, next thing is your uterus. Okay, uterus, it is a cavity which is present in the female reproductive system, a tube which is present inside the female reproductive system where implantation takes place. Okay, so it is an inverted pear shaped muscular structure attached to the pelvic wall and supported by ligaments. Okay, it is attached by the help of ligaments. So the wall of uterus has three layers tissue, the outermost and external layer of thin membranous perimetrium, the middle layer thick layer of smooth muscle called myometrium, and innermost glandular layer called endometrium, which lines your uterine cavity. Okay, so these are your three layers which is present inside the uterus, and their nature. That external one is this thin membranous which is called perimetrium, and function is it is visceral peritoneum, means it is the outside lining. Okay, then the middle one is your muscular layer which is myometrium, and the function it exhibits strong contraction during the delivery of the baby, and it is induced by the help of oxytocin hormone. Okay, next one is your inner layer which is glandular in the nature, means it secretes hormones. It is your endometrium, and its line the uterine cavity undergoes cyclic changes during menstrual cycle. The blood which is coming during the menstrual cycle, it is coming from your endometrium. Okay, then we have vagina. Okay, it is a muscular tube like structure that open outside. Okay, the vagina is partially covered by a membrane called hymen. The cervix and the vagina together constitute as called birth canal. Vagina plus cervix equals to birth canal. Okay, the function of vagina are to receive the male gametes during insemination and serve as a birth canal during parturition. Means when the male penis get inside the female body, it will transfer the sperm inside the female body and it first enter inside the vagina. And second, when the when the baby leaves the mother body, okay, then it's come out to your vagina. Okay, so next is your external genitalia. Okay, so female external genitalia includes mons pubis, labia majora, labia minora, clitoris, and hymen. Okay, mons pubis, it is a cushion like fatty tissue covered by skin and pubis hair. Okay, external genitalia is a part of external genitalia. Second, we have it extend down as a part of fleshy fold of tissue called labia majora and surround the vaginal opening. Okay, here we have labia minora and labia majora, this side area. Okay, next we have labia minora, a paired fold of tissue under the labia majora. It is also surrounded the vaginal opening. See, the majora, it is present the outside, while your minora, it is present inside in the female reproductive tract. Then we have clitoris. Okay, it is a tiny finger-like structure which lies as the upper junction of two labia minora. Above the urethral opening, it contains a pair of corpora cavernosa, cylindrical and electrile tissue. This is your clitoris. It is just like your urethra. Okay, in female. Next we have hymen. It is the membranous that partially covered the vaginal opening. It become ruptured due to vigorous physical activities and during the first coitus. Okay, people says that loss of virginity means when the hymen is broken means you lost the virginity, but it is not true. It can be broken due to physical activities. If you are riding a horse, then this thing can happen. If you play cricket, kabaddi like games in which high tension is applied on the reproductive system, then this can be broken down. Okay, next one is your mammary gland. Okay, human female has pair of functional mammary gland. Mammary gland is also present in male, but it is non-functional. But it is functional in female. Okay, so in human female has pair of functional mammary glands that consist of glandular tissue and variable quantity of fat. Okay, the glandular tissue is divided into 15 to 20 mammary blocks, which each lobe contains cluster of cells called alveoli, which opens into mammary tubules. That is again the tract of milk conduction, how milk move, that your colostrum move in your in the mammary glands. Okay, the mammary gland tubules each loop join to form a mammary duct, then several mammary ducts join to form a wider mammary ampulla that connected to the laciferous duct through which milk come out. Means it is an induced action. Okay, it is an terminus action in which when the lips of baby touches the nipples of mother, then that milk come out. Okay, the milk is actually secreted by the cell of alveoli and is stored in the lumen of alveoli. Okay, and this is the conduction of milk. Okay, so first we have mammary alveoli, then it will transport its solution, its constant inside the mammary tubules. Then from mammary tubules it moves towards the mammary duct. From mammary duct it will goes inside the mammary ampulla, and from mammary ampulla it will go inside the lactiniferous ducts. Okay, and it will come out from nipple to the inside the mother inside the mouth of the baby. Okay, next we have gametogenesis, the second part of this chapter, how the gamete formation take place, how your sperm and ovum formation take place. Okay, so first we have spermatogenesis. Okay, it is the process of formation of spermatozoa in the testis. The process start at the puberty. Okay, when male become adolescence, okay, puberty starts, then spermatogenesis starts. Okay, in testes the immature male germ cell spermatogonia produce sperm by spermatogenesis that begins at the puberty. Okay, at the age of puberty, that spermatogenesis start inside the male body. Spermatogenesis starts at the age of puberty due to the significant increase in the secretion of GnRH. Gonadotropin releasing hormone increases during the time of puberty in the male body, due to which it secretes FSH and LH, and which acts on the Leydig cell and Sertoli cell of testes. Okay, so stages of spermatogenesis, we have 4 stages: stage of population, stage of growth, stage of maturation, and stage of transformation. So first we have multiplication phase. Okay, a sexual maturity, the spermatogonia present on the inside wall of seminiferous tubules multiply by mitotic division and increase in number. Each spermatogonia is diploid and contain 46 chromosomes. In multiplication phase, you only see mitosis. Okay. Some of the continuous stem cell and type A spermatogonia other differentiate into progenitor cell, type B spermatogonia. Now type A spermatogonia serve as a stem cell for spermatic lineage, and where type B spermatogonia as a precursor of sperm, means it will give nutrition to your sperm A. Okay. Next we have growth phase. Okay. Type B spermatogonia differentiate into primary spermatocyte and become enlarged. Still no meiosis, mitosis is going on. Okay. Then we have maturation phase, the most important phase of spermatogenesis, in which meiosis take place. Okay. Some of the spermatogonia type B after growth differentiation called primary spermatocyte periodontially undergo meiosis. Okay. First time meiosis take place. So primary spermatocyte complete the first meiotic division, leading to formation of 2 equal haploid cells called secondary spermatocyte, which have only 23 chromosomes. Okay, now secondary spermatogones again go in second meiosis. Okay, and it will convert into four cells. Okay, so now we have some terms. First we have spermiogenesis. Okay, it is the process by which spermatids become mature spermatozoa. The change taking place during the stages are condensation of nuclear material, formation of acrosome, mitochondria, and filament and tail-like structure to the movement of sperm, and removal of unwanted quantity of cytoplasm. Means unwanted cell organelle, unwanted quantity of cytoplasm from the cell get deleted. Okay. Next we have spermination. Okay. Spermination is the process by which mature sperms are released from the Sertoli cell into the lumen of seminiferous tubules. Okay, they are coming out from the Sertoli cells inside the seminiferous tubules. Then it is called spermination. Okay, so this is your spermatogenesis in summarized way in the form of flow chart. See, this is your 2n structure which is called spermatogonia. It will convert into primary spermatocyte. It will differentiate into primary spermatocyte by the process of mitosis. Now meiosis 1 take place and this primary spermatocyte convert into secondary spermatocyte. Then again meiosis 2 take place. Now this secondary spermatocyte convert into 4 spermate and further the spermate convert into sperm. Okay. Now hormonal control on spermatogenesis. Different hormones come and control the process of spermatogenesis. So spermatogenesis is initiated due to the increase in the secretion of gonadotropin releasing hormone by hypothalamus at the age of puberty. Means when the male child become adolescence, puberty hit to that male child, GnRH secretion increases inside the body. Your hypothalamus start to releasing GnRH in large amount. Now that GnRH acts on your that anterior pituitary, due to which two hormones come out, that is your LH and FSH, Leydig, that your luteinizing hormone and follicle stimulating hormone. Okay, now that LH and FSH will work on, LH will work on Leydig cell and FSH work on your Sertoli cell. Okay, now from your Sertoli cell androgen binding protein come out, and from your Leydig Leydig cell intestinal cells your testosterone come and they will act on your developing spermatozoa. Okay, so structure of spermatozoa. Now we are going to study the structure of sperm. Okay, so Dutch scientist Antoine van Leeuwenhoek discovered human sperm in 1677 using microscope that he had invented. Okay, the sperm is composed of head, neck, middle piece and tail. These are the parts of body parts of sperm. Okay, a plasma membrane envelope the whole body of sperm, on which you get whole part is covered by a plasma membrane. Now we will draw the diagram of sperm. This yellow lining is the plasma membrane which is present all over the sperm. This is the head part, this is your neck part, this is tail. Okay, so that head part having your nucleus and acrosome. Okay, then your neck part, this part, okay, it will contain centrioles. Okay, next you have neck part. Okay, that middle piece part. Okay, it will contain your mitochondria. Okay, and at last you have a tail, filamentous tail, which help in the movement of sperm. Okay, so head part, head contain an elongated compact haploid nucleus, concentrated DNA which is present inside. Okay, the interior end has a cap like structure called acrosome. The acrosome contain enzyme that help in dissolving membrane of ovum from sperm. Then neck, it contains two centrioles which helps in the process of cell division, helps during the cleavage process of zygote. After that zygote formation, the cleavage process take place in which it will helps. Okay, then we have middle piece. Middle piece contain a number of mitochondria that provide energy to the motility of the sperm. The source of energy in sperm, how your sperm can move, that flagella, how that flagella flex, that is because of mitochondria which is present inside the middle piece. Next we have tail. It consists of an axial filament surrounded by plasma membrane to help in sperm to move. in the female reproductive tract towards the female gamete ovum for fertilization. Okay, the motility organ of sperm is your tail. Okay, so this is the structure of human sperm. Okay, in which this is your tail. Okay, then we have middle piece which is contain mitochondria in spiral manner. Then we have neck. In the neck you have two centrioles. Then we have head, in which you have haploid nucleus and acrosome on the head of the that sperm, which help in the penetration to ovum. Okay. Next we have oogenesis. Okay, formation of ovum in female we call oogenesis. So the process of formation of mature female gamete is called oogenesis, which markedly different from spermatogenesis. Oogenesis initiated during the embryonic development stage. Okay, when female baby is present inside the mother body, at that time oogenesis is already started, but it is arrested to a particular step, then it will hit again when the female become pubert. Okay, when the puberty hit to that female, then it again start that oogenesis process again started. Okay, so first we have multiplication phase. Okay, by the time of fetus 25-week old, a couple of million gamete mother cell oogonia are formed by mitosis with each fetal ovary. Both ovaries produce your oogonia by the process of mitosis. Next we have these all are oogonia that she will even produce no more oogonia are formed and added after the birth. Okay, means if a female produces 8 lakh oogonia during the fetal stage, then it remains 8 lakh. It will not get added with other oogonias. Okay, for example. Okay, next we have growth phase, in which oogonia grow into primary oocytes. Okay, and the third stage, the most important stage of oogenesis, is your maturation phase. Okay, in which your primary oocytes will convert into secondary oocyte. Okay, so primary oocyte start to division and enter into prophase 1 of meiotic division and get temporarily arrested to the diplotin stage. Okay, these cells resume their first meiotic division when the female attains puberty. Okay, means when maturation phase start, your primary oocyte will convert into secondary oocyte, but it's temporarily arrested in prophase 1, and when the puberty hits to that female, that arrested secondary oocyte will come out from that arrest. Okay, now primary oocyte complete its first meiotic division. It is unequal division resulting in formation of large haploid secondary oocyte and tiny first polar body. Okay, that arrested primary oocyte will convert into secondary oocyte and polar body when your woman, that female attains puberty. Okay, so secondary oocyte retain bulk of the nutrient rich cytoplasm in primary oocyte. Then secondary oocyte proceed as far as the metaphase stage of second meiotic division. However, it then stop advancing to further. It awaits an arrival of sperm for the completion of secondary meiotic division. When the female is on the metaphase 2, okay, so you have that metaphase of that meiosis second, it will again arrested, and now it will retain its phase in the metaphase 2, or that arrested phase of metaphase, that meiosis second, and when sperm come, it will get contact and it will be de-arrest from that meiosis second metaphase. Okay, so this is the process of oogenesis. Simultaneously, follicle development also take place inside the female body. Okay, so mitosis. From that mitosis, your oogonia convert into primary oocyte, but it is arrested in prophase 1. Okay, now when the child ovary inactive forms the puberty to menopause, okay, it will convert into primary oocyte. Still, it having 2 n haploidy. Okay, now meiosis 1 occur after the time of that puberty, and it will differentiate into secondary oocyte and primary oocyte, but that secondary oocyte is still inside the arrested phase of metaphase second. But if accidentally sperm come and it will fertilize your ova, then it will come out from that arrest and it will convert into the zygote. Okay, that convert into the zygote 2n structure. Okay, simultaneously follicular development also taking place inside the ovary, from which primordial follicle convert into promoidal follicle cells, then promoidal follicle convert into primary follicle grow into growing follicle converts into your mature follicle, and mature follicle convert into mature follicle convert into Graafian follicle, and after Graafian follicle ovulation take place. From that ovulation, if your that ovum get fertilized, then it will convert into corpus luteum. Okay, and corpus luteum is a temporary body, temporary structure which is responsible for secretion of certain hormones like progesterone and estrogen. Okay, so vitamin E is essential for the gametogenesis. Deficiency of vitamin E lead to sterility. Vitamin A is also required for the formation of healthy gametes. These are the two vitamins which is required to the formation for the process, for the good processing of your gametogenesis, that is vitamin E and vitamin A. Okay, now we are going to see the internal structure of ovary, what is present inside the ovary. Okay, so each ovary is covered by an outer thin membrane called germinal epithelium. Again, same thing which we see in testes. It is surrounded by germinal epithelium. Same thing is present inside your ovary. Okay, and an inner white fibrous connective tissue layer tunica albuginea which encloses the ovarian stroma. Okay, the stroma is divided into two zones: peripheral cortex and the medullary region. Okay, the stroma is composed of stromal cell, connective tissue, and blood vessel, etc. Numerous oogonia are found in the stroma of the cortex during intrauterine time. Okay, so this is your primary ovarian follicle. It will convert into secondary ovarian follicle, then it will convert into the tertiary, then tertiary convert into Graafian follicle. Now this Graafian follicle is wait for ovulation. Ovulation take place, ovum come out. Okay, now the structure will convert into a yellow color structure which we call corpus luteum, and it will start to generate, to secrete different hormones like your LH, progesterone, estrogen. Okay, if fertilization does not take place, pregnancy does not take place, then this corpus luteum will convert into corpus albicans. Again, mark my words that if pregnancy fertilization does not take place, corpus luteum converts into corpus albicans, and that we are going to study in that sexual reproduction cycle, in menstrual cycle of female. Okay, so follicular development, it having that stages, different stages. First we have primordial follicle developing primary follicle, then we have primary follicle, then we have secondary follicle, then we have tertiary follicle, after that mature tertiary follicle which we also call Graafian follicle, and then we have the corpus luteum. Okay, then we are going to study that thing in detail. Then we have primary follicle, some cells of stroma become flattened and surrounding as primary oocyte. These flatted cells from ovarian follicle and are therefore called follicular cell. Okay, then we have primary follicle, flatted follicle cell become columnar. Okay, the flatted cells, the chapte cells, they will convert into pillar columnar like cells. Okay, now zona pellucida, membrane of glycoprotein nature appears. Okay, so in your primary follicle first membrane appear, that is your zona pellucida. Okay, primary outside surrounded by a layer of granulosa cells called primary follicle. A puberty, at the puberty only 60,000 to 80,000 primary follicle left in each ovary, means in each ovary it is 80,000, so in both ovary it is your 80 plus 81, 80,000 in one ovary, so in two ovary it will be 1,60,000. Okay, so secondary follicle, as the follicle expand, the stromal cells surrounding the membrane will also become condensed, start forming a covering called theca interna. Okay, it is an theca layer, a cells layer which is called theca interna. Okay, outside the theca interna some fibrous tissue belongs to condense and start forming another covering called theca externa. Means they have two layers, one is theca interna and one is theca externa. First your theca interna form, after that theca externa form. Okay, next we have tertiary follicle. Now secondary follicle soon transform into the tertiary follicle which is characterized by fluid filled cavity called atrium. The fluid of atrium is termed as liquor folliculi. Okay, next we have mature tertiary follicle or Graafian follicle. The tertiary follicle further changes into mature follicle or Graafian follicle, and the primary oocyte complete its first meiotic division when it's convert into Graafian follicle. Okay, due to the accumulation of more fluid, the size of atrium rapidly increase. Okay, that fluid, it is start to increase inside the atrium, and that increase in the number of the fluid, it will make it to rupture at some time. Okay, the projection of granulosa cell covering the oocyte into atrium is called cumulus oporus. Okay, the granulosa cell lying in the closed venicities or xenoplastra become elongated and forms corona radiata. Okay, and a layer is formed which we call corona radiata, and you will see inside a mature ova also. Okay, the Graafian follicle now rupture to release the secondary oocyte ovum from the ovary by the process called ovulation. Okay, and at last we have development of corpus luteum. Once ovulation has occurred, the follicular ball remain as a collapsed sac. This is called corpus hemerangium. Okay, because of blood clots appear on this surface, it is considered as early stage of corpus luteum. Okay, the early stage of corpus luteum is corpus hemerangium, and the late stage of corpus luteum is corpus albicans. Okay, the luteinizing granulosal cell, luteal cell in this collapsed sac began to divide and invade the old antral cavity, thus forming corpus luteum. Okay, Latin yellow body. The corpus luteum Latin word is yellow body. It is yellow because of presence of pigment in luteal cell. Corpus luteum acts as a temporary endocrine structure. The main function of mammalian corpus luteum is to produce progesterone only. Okay, and it will under the influence of LH hormone. Okay, the subsequent phase of corpus luteum depend on whether it fertilization or pregnancy occur or not. Okay, if pregnancy occur, then if fertilization occur, then corpus luteum remains. If pregnancy not occur, your that fertilization not occur, then it will convert into corpus albicans. Okay, so this is your flow chart of that follicular development. Okay, primary follicle, then your secondary follicle, then your tertiary follicle, then your Graafian follicle. Okay, in every stage you will see different layers will develop. In the first stage, your primary follicle granulosa cell layer develop. In secondary follicle, you will see theca interna and theca externa develop. Simultaneously, granulosa layer become more dense and zona pellucida start develop. Okay, in tertiary follicle, you will see atrium filled cavity start to develop in which fluid is filled, and at last in Graafian follicle, that atrium, that cavity is enlarged and it will filled with that atrial fluid. Okay, so in total number of primary follicles, the two ovaries are normal. Young adult woman is about 1,20,000 to 1,60,000, because in the initial you will see that 80,000 ovaries, that ovas or that primary oocytes are there. Okay, in per ovary. So however, most of them undergoes regression and disappear due to death and disposal by phagocytic during the reproductive years of the female. This is referred to as follicular atresia, and this is responsible for the limited number of gamete production in the life of female. See, in female body, the number of gamete, female gametes means ova, it is limited. While in male, the number of male gametes means your sperm, it is unlimited. Okay, so now structure of human egg. Okay, ovum, the structure of ovum. Okay, so previously we studied about structure of sperm, now it is structure of ovum. Okay, so ovum has three membrane: plasma membrane, oolemma, innermost layer. Okay, then we have zona pellucida, outer membrane to plasma, and last one is your corona radiata, which is the outer layer form of follicle cell. Okay, this is your cornica radiata, which is form of that follicular cell. Then you have zona pellucida, then inside zona pellucida you have vetrine membrane, then inside you have cytoplasm. Okay, vetrine space is the space between plasma membrane of oocyte and zona pellucida. This area, this light blue ring area, this is your vetrine space. Human eggs is alecical on the basis of amount of yolk, and isolecical, homolecical on the basis of distribution of yolk. Okay, in the last of this chapter we will going to study about different types of egg also. Okay, so now the step is your fertilization. I divided this thing into two parts. If that ovum get fertilized, what happened? If it is not get, what happened? If it is fertilized, then it is your fertilization take place, then after implantation take place, and if it is not fertilized, then your menstrual cycle run. Okay, so fertilization, the process of fusion of sperm with ovum is called fertilization. Okay, during copulation, coitus, semen is released by the penis into vagina. The motile sperm swims rapidly past the cervix, enter into the uterus, and finally reaches to the ampler region of the fallopian tube, where the ovum is waiting for the fertilization in the arrested metaphase 2 stage. Okay, multiple changes that activate the sperm for the final process of fertilization are collectively called capacitation, and the occur of sperm is coming into the contact with the fluid of the female genital tract. Means when the sperm get in the contact with the female genital tract, it get activated. Okay, the ovum released by the ovary is also transported to the ampler region where fertilization takes place. The site of fertilization in human is ampulla isthmic junction. Okay, fertilization can occur if ovum and sperm are transported simultaneously to the ampulla region. This is the reason why not all copulation lead to fertilization and pregnancy. Okay, because we cannot notify, we cannot give the timing, but particular timing, perfect timing at what that sperm and ovum meet to each other. That's why all copulation does not convert into fertilization and pregnancy. Okay, so fertilization has following main events. Okay, first we have syngamy, union of male and female gamete. Then plasmogamy, intermixing of cytoplasm of male and female gametes. Then karyogamy, fusion of pronuclei, male and female gametes. Then ampemixis, intermingling of chromosome of male and female gametes. Okay, crossing over take place. The secretion of acrosome help in sperm enter into the cytoplasm of ovum through the zona pellucida and plasma membrane. This helps inside to make the sperm go inside the ovum. Okay. So this is the full step in the form of flow chart. So first is movement of sperm towards the secondary oocyte. Now penetration of sperm through corner reducta with the help of hydrolysis enzyme. Okay, after that adhesions of sperm to the sperm receptors, JP3 receptor which is present on the zona pellucida. Okay, then initiation of acrosomal reaction and release of acrosin and zona lysin enzyme which degrade your zona pellucida. Okay, penetration of sperm through zona pellucida with the help of acrosin enzyme, then syngamy take place, means fusion of two plasma membrane. Okay, then phagocytosis sperm by secondary oocyte, during which completion of secondary mitotic division of secondary oocyte to form ovum and secondary body. After that plasmogamy takes place, fusion of plasma, then karyogamy, fusion of nucleus, then ampemixis, and at last we form a zygote. We get a 2N structure which we call zygote, the starting point of new life, that is your zygote. Okay, now if that egg and your that sperm does not meet, then what happens? Then menstrual cycle happens. It is a repeated cycle which happens in every month inside the female body. Okay, so it is a reproductive cycle in the female primates, example monkey, apes and human beings. It is called menstrual cycle. In human female, menstrual cycle is repeated at the average interval of about 28 to 29 days, means it is runs about 1 month. Cyclic menstrual cycle is indicator of normal reproductive phase and external extent between the menarche and mesophase. Menarche means when your menstrual cycle start inside the female body, and menopause means at what, at the state, at the age when that stops inside the female body. Okay, the first menstrual cycle begins at the puberty and it is called menarche, and the menstrual cycle cheese around 50 years of the age, that is termed as menopause. Means when menstrual cycle stops it is called menopause, and when menstrual cycle start it is called menarche. Okay, in this cycle the female body prepares itself for a possible pregnancy, and if the pregnancy does not occur, then the body abort all the preparation in the cyclic manner. Okay, the cycle of events starting from one menstrual till the next one constitute a menstrual cycle. Okay, from one point of menstrual cycle to another point, it is called menstrual cycle. Okay, means from first day to the 28th, when then again next cycle start from the first day to the 28th day, that cycle is called menstrual cycle. Okay, so menstrual cycle have three main phases: menstrual phase or bleeding phase, then follicular phase, oestrogenic phase or polyphase phase or pre-ovulatory phase, then luteal phase or progesterol phase, secretory phase or post-ovulatory phase or fixed phase. Okay, so these are the three phases which we are going to study in detail. Okay, so menstrual phase, the cycle start with this phase, and the menstrual flow, menstruation lasts to 3 to 5 days. Okay, it results due to the breakdown of endometrium lining of uterus and blood vessel along with the unfertilized ovum. Means if in the previous cycle, if that egg is not get fertilized, then this phase start. Okay, if it is fertilized, then this phase not start in the next cycle of menstrual cycle. Okay, then we have follicular phase or proliferatory phase. Okay, in this phase proliferation of ovum take place, means development of ovum take place, development of endometrium take place. Okay, in this phase the primary follicle in the ovary grow and become fully mature Graafian follicle. Okay, the endometrium of uterus is regenerated by proliferating of its cells, means the glandular and the that manners change take place inside the endometrium of the female. Okay, these changes are due to the increased level of pituitary hormone FSH and LH and ovarian hormone, that is estrogen. Listen, the main thing in follicular phase, three hormone acts mainly, that is your FSH, LH and estrogen. Progesterone is present but its role is very less. Okay, this we are going to see in the graph also now after that. Okay, FSH control the follicular phase, it stimulates the growth of follicular and secretion of estrogen by growing follicle. See, FSH acts on the follicle and that follicle start to secrete estrogen. Okay, Both FSH and LH reach their peak level in the middle of the cycle about on 14, that which we call also known as ovulatory phase, or the day when ovulation take place. Means from that Graafian follicle ovum come out, that is happens on the 14th day of menstrual cycle. Okay, then we have ovulatory phase. The peak level of LH, which is also called as LH surge, induce the rupture of mature Graafian follicle and thereby the release of ovum. This process is called ovulation. It is happens on the 14th day of menstrual cycle. Okay, when the level of LH is on the peak, and it is called LH surge. Simultaneously, FSH is also on the peak, which we call FSH peak, and LH we call LH surge. Okay, then luteal phase. Okay, last phase, that is secretory phase. Okay, during this phase the rupture follicle is transferred into corpus luteum. Okay, and it waits that if that your ovum is fertilized or not. If it is fertilized, then it remains just like that corpus luteum and start to producing progesterone hormone. Okay, your progesterone hormone come in the play in luteal phase. Okay, and if it is not get fertilized, the ovum not get fertilized, then your corpus luteum convert into corpus albicans. Okay, so in this secret large quantity of progesterone needed to maintain the endometrium. Now your endometrium become more vascular and more thicker in the size. Okay, the endometrium thicken further and their glands secrete fluid into the uterus. Okay, in the absence of fertilization, corpus luteum degenerate, and this causes degeneration of endometrium leading to menstruation, that will your first to fifth day of next month cycle. Okay, so this is the hormone controlation of in female body. Okay, see, hypothalamus, it will start secret GnRH. Okay, now this GnRH act on anterior pituitary and start the secretion of FSH and LH. Okay, now this FSH go and act on growing follicle, and growing follicle start to secrete two hormones, estrogen and inhibin. Okay, and that LH hormone go and act on your corpus luteum, and now corpus luteum secrete out four hormones, which is your relaxin, inhibin, estrogen and progesterone. Okay. Energy. Come. No. Okay, now menstrual cycle and there are three main phases, that is your completed. Now we will take functions of different ovarian hormones, how different hormones work on that menstrual phase, on the menstrual cycle. Okay, so first we have estrogen. It causes endometrium hyperplasia, means it increases the size of endometrium. It increases the vascularity of endometrium. Uterine arterioles become tortuous, means it will become larger in the size. Okay, it causes myometrium hypertrophy. Ultimately it increases the thickness of uterine wall. Next we have progesterone, also known as anti-abortion hormone or pregnancy hormone. Okay, it increases the secretory nature of endometrium, means it start to induce secretion of different hormones and different material liquids from the your uterus. Okay, it increases the adhesive nature of endometrium, means progesterone is basically doing what? It favoring pregnancy, it making uterus favorable to pregnancy. So all the things is responsible for a good pregnancy. So increase adhesive nature of endometrium because implantation take place in your uterus. Okay, that's why it increases the adhesive nature of endometrium. Then it opposes myometrial contraction, because if myometrial contraction take place, then that ovum will can that fertilized zygote can get outside from that uterus from to vagina and from get out from the female reproductive tract. So it stops myometrial contraction. Okay, it essential for maintenance of endometrium, such as endometrium is necessary for implantation and the fertilized ovum and other events of pregnancy. Okay, that's why progesterone is called anti-abortion hormone or a pregnancy hormone, because it favors pregnancy. Okay, inhibin, it suppress FSH synthesis. Okay, here is your inhibin. It suppresses the FSH hormone to stop growing follicle to produce estrogen. Okay. Next is your only about 400 ova are produced by a human female over the entire span of her reproductive life. Means from your menarche to your menopause, only 400 ova is produced by reproductive system of female. Okay. So this is the graphical representation of menstrual cycle. In the starting, you will see this is your first to fifth day of menstrual cycle, in which you will see the level of FSH is very low and simultaneously LH level is also low. And your progesterone and estrogen level is also low. In comparatively, progesterone level is very low during the menstrual phase of 1 to 5 to 7 days. Okay. Now when follicular phase starts, okay, now the rise of estrogen take place. First rise of estrogen take place, only estrogen is take the rise. Okay, and at the day of 14 means your ovulation day, or that ovulatory phase day, on the 14th day of menstrual cycle, you will see LH surge and FSH peak. Okay, simultaneously you will see that estrogen is also on the peak. Okay, now then that luteal phase start, means ovulation take place, that ovum come out from Graafian follicle and the Graafian follicle turns into corpus luteum. You will see that progesterone rise take place, because your corpus luteum is responsible for the secretion of progesterone. Okay, and simultaneously your estrogen level goes down. If pregnancy take place, then it will rises, that progesterone level rises. If it is not take place, it will goes again down and prepare itself, that uterus prepare itself for next menstrual cycle. Okay, so that is your menstrual cycle. Now you have a zygote. Okay, now embryonic development take place. How that embryonic development take place? There is different phases in embryonic development, which we call cleavage. Okay, so embryonic development, which we call cleavage. Cleavage is also known as segmentation or close mitosis. Cleavage division is rapid and repeated mitotic division. Okay, now mitosis take place. Your zygote divide into 2 cells, then 4 cells, then 8 cells, then 16, then 32, then 64. At this level of division take place inside the then zygote convert into that embryo, and embryo convert into fetus. Okay, so mitosis division start at the zygote, moves through the isthmus of the oviduct called cleavage towards the uterus and forms 2, 4, 8, 16 daughter cell called blastomeres. Okay, now that cleavage in zygote result into multicellular embryo without any changes in overall size. Okay, size does not change. If it is like this, it will remain like this. Okay, the size does not increases, but the number of cell inside that particular ball, it increases. Okay, so first stage is your morula. The embryo with 8 to 16 blastomer is called morula. Okay, now when it is larger, it becomes the number of cell inside that ball like structure increases, then it is called blastocyst, and this blastocyst go and attach to the uterus wall, and with this process is we call implantation. Okay, there we written here. Okay, cell division continue in morula and blastoma become arranged along the periphery, leaving a central cavity called blastocele. The embryo at this stage is called blastocyst. Okay, this is your blastocyst. Okay, and this is the cavity which we call blastocyst or blastocele. Okay, and this is the outermost covering of your blastocyst, which we call tropoblast, and this is your inner cell mass, which will convert into embryo, and this thing will got embedded inside the endometrium of uterus and will go inside and condense inside. Okay, and this is the development of your embryonic development stage. Okay, that is your fertilized egg, it will convert into 2 cell stage, then 4, then 8, then it is your 16 stage which is called morula convert into blastola convert into early blastola, and blastula then convert into gastrula. Okay, next we have implantation, the most important process after fertilization. Implantation, the definition of implantation is attachment of blastocyst to uterus, wall of uterus, is called implantation. Okay, simple definition, that is attachment of blastocyst to uterus wall is called implantation. Okay, so the trophoblast layer get attached to the endometrium. The cell of endometrium divide rapidly and cover the blastocyst. So the blastocyst get embedded inside the endometrium. This process is called implantation. Okay, means if it is a blastocyst and this is the wall of your uterus, it will slowly go inside this and it will embed it inside this, and this process is called implantation. Okay, then we have blastula. It is a stage transformed from blastocyst stage by a process called gastrulation. Okay, means now the cells numbers start to increase inside, means the gastrula stage it will happens after the implantation. Okay, so that thing will increase the number of cell, and now your gastrula will convert into developing embryo. Okay, so extra embryonic membrane. Okay, now your embryo is ready and it having some membrane, some layers. Okay, which we call extra embryonic membranes or extra embryonic layers. First we have chorion, then we have amnion, then yolk sac, and at last we have allantois. Okay, chorion, it is formed by the ectoderm or outer side of mesoderm or inner side. Okay, amnion, it is form of mesoderm or outer side of ectoderm or amniogenic or inside cell. It provides fluid medium, okay, to the developing embryo, which prevents dissociation of embryo and function as a shock absorber. Amnion act as a shock absorber. Chorion, it take parts in the formation of placenta, means chorion will convert into placenta and amnion convert into amniotic fluid. Okay, then we have yolk sac, the form of mesoderm and outer side of endoderm in the inner side. Yolk sac is the first site of hematopoiesis in human ontogeny. Okay, means first time the blood formation take place inside your yolk sac. Okay, then we have allantois. It forms of mesoderm and outer side of endoderm, and it is a protective coupling which is present in the developing embryo. Okay, and this is the diagrammatic representation of extra embryonic membranes. This is your chorion, then amnion, then embryo which is present in the middle, then it is your allantois which is converted into umbilical cord, yolk sac, fetal portion of placenta, maternal portion of placenta will act as an exchange between the both things. Okay, next we have placenta. Okay, placenta again a temporary hormone producing organ present inside the body of female. Okay, it is also a temporary hormone producing unit. Okay, temporary, not a permanent hormone producing unit. Okay, so after implantation, finger like projection appear on the trophoblast called chorion villi, which surrounded by uterus tissue and maternal blood. Okay, this is your placental layer. Okay, which act as a barrier, which act as a support between developing fetus and mother body. Okay, it is a connection between mother and fetus, which we call placenta. Okay, the chronic villi and uterine tissue become integrated with each other and joint form a structural and functional unit between developing embryo and maternal body called placenta. Okay, the placenta is connected to the embryo through an umbilical cord which help in the transport of substances to form the embryo. Okay, this is your umbilical cord. Okay, by which transport of oxygen and nutrients take place between the fetus and mother. Okay, so function of placenta, it acts as a barrier between fetus and mother, supply oxygen, nutrition, etc. from mother to fetus, remove CO2 and excretory waste from the fetus. Okay, act as an endocrine gland for some time and secrete human chorion gonadotropin HCG, human placental lactogen HPL, and estrogen, progesterone, and relaxin. Relaxin is also secreted by ovaries. Okay, the test of pregnancy. Okay, it is because of this hormone. Okay, if in the urine of female HCG is present, then the test of pregnancy get positive. Okay, so these all are the hormones which is released by your placenta, that is human gonadotropin hormone, rescue corpus luteum from degeneration. It will reduce the degeneracy of your corpus luteum. Okay, due to which corpus luteum induce and produce progesterone and estrogen, which maintains pregnancy and prepare that mammary glands for lactation. Okay, next we have relaxin, increase the flexibility of pubis symphysis and help in dilation of uterine cervix during the labor. Okay, as you know, when parturition take place, it will take place by your birth canal, so it should be wider. That's how your baby come out. Okay, and that widerness of birth canal, it is because of this hormone, relaxin hormone. It is also called birth hormone. It is responsible for the birth of a baby. Okay, then we have human placental lactogen HPL and human conjugamin somatotropin HCS, helps in prepare mammary gland for lactation. Okay, and we have CRH gonadotropin releasing hormone, establish the timing of birth and increase the secretion of cortisol inside the female body. Okay, so HCGL HPL relaxin are produced in women only during the pregnancy. Normally in women these hormones are not produced. HCG, HPL, relaxin, these produced during the time of pregnancy only inside the female body. In normal female who is not pregnant, these hormone not present inside that female body. So means the formation of these hormones is the sign of pregnancy. तो मैं कह सकता हूँ कि जब ये तीन हार्मोन बन रहे हैं मतलब वो फीमेल प्रेग्नेंट है. ओके. During pregnancy, the level of other hormones like estrogen, progesterone, cortisol, prolactin, thyroxine, etc. is increased severalfold in the maternal blood. Increased production of these hormones is essential for supporting the fetal growth, metabolic changes in the mother, and maintaining the pregnancy. Okay, these all are metabolic hormones and sex hormone which control the pregnancy inside the female. Okay, to determine the presence of SCG in the urine of suspected woman is the basis of pregnancy test. Okay, if a female contained HCG in urine means that woman is pregnant. Okay, next is changes in embryonic during the pregnancy. Okay, what are the changes take place inside the embryo during the pregnancy? It will divide into different trimester, different weeks. Okay, so after 1 month of fertilization, heart is formed. So first organ formed in a baby is heart. Okay, then end of second month limbs and digits are developed. Okay, then end of 12th week means first trimester completed, then major organs like limbs, external genitalia, organs, etc. are well developed and they are forming inside the body of developing fetus. During the 5th month, first movement of fetus and appearance of hair on the head take place. Okay, and on the age of 24 week means second trimester, body is covered with fine hairs, eyelids separate and eyelashes are formed, and end of 9th month ready for delivery. Okay, this is how the development of fetus takes place inside the female body. This is 1 month old, this is 2 month old fetus, this is 3 month old fetus, this is 4th month old fetus, this is 5 month old fetus, 6 month old fetus, 7th month old fetus, 8 month old fetus, and this is 9th month old fetus ready for delivery, ready for parturition. Okay, next thing is your parturition. Okay, so process of giving birth to young one, it is induced by neuroendocrine mechanism. Relaxin hormone is the main hormone and oxytocin hormone is the main hormone who induce this process. Two hormones, relaxin and oxytocin, is responsible for parturition process. Okay. To signal from the fetus on placenta induced mild uterine contraction, fetal ejection reflex. This causes the relaxation of oxytocin from maternal pituitary gland. Okay. Oxytocin causes strong uterine muscle contraction, which is in turn stimulate further secretion of oxytocin. When that maternal fetus send the stimulus to maternal pituitary start to release oxytocin. Now this oxytocin act on the muscles of uterus and start heavy contraction, due to which the baby start to come out. At that time simultaneously relaxin hormone secrete on via the placenta, and the ovary also facilitate the paturation by providing wider space for the fetus to move out. This relaxin hormone, it will act on pubis symphysis of female and relax pubis symphysis, by which that baby come out. Okay. So after paturation, oxytocin hormone stimulate milk let down by milk ejection reflex. This process is continued, leading to the expulsion of baby out of the uterus through the birth canal. After parturition, the umbilical cord is cut off. The placenta is remnant of umbilical cord are expelled from the maternal body. After parturition, it is called afterbirth. Okay, after birth, that thing means removal of umbilical cord from the female body, from the mother body, is called afterbirth. Okay, so this is the cutting of umbilical cord take place. Okay. Then you have, this is the reflex action. How that reflex action works: baby make that reflex arc, and this reflex send it to the pituitary gland of my mother, due to which oxytocin hormone come out, and that oxytocin hormone act on the uterus of female, due to which heavy contraction take place. Simultaneously, relaxin hormone is also produced by placenta and your female that ovary, which helps in the widening of pubic symphysis and the easy delivery of baby take place. Okay, this is the whole that parturition process in the diagrammatic view. First is onset of labor, then flexion, then internal rotation of head. After the extension, then internal rotation of head and uterus immediately after the birth. Okay. Next we have egg. Okay, classification of egg. Okay, so we have, on the basis of distribution of the egg, three types of egg: isoleucithal egg, teloleucithal egg, and centroleucithal egg. Isoleucithal egg means egg with yolk distribute evenly. Okay, they are also called oligolethical type. Okay, example echinoderm, amphioxus, and mammals. Okay, means on the center, means that yolk is widely distributed, means eventually distributed all over the egg, then it is called isoleucithal egg or homolecithal egg. Then we have teloleucithal egg, egg with end centric yolk distribution. These are polycentric polyleucithal type of egg yolk, heavily laden and uneven. Okay, it is again of three types: slightly teloleucithal, present in fish; moderately teloleucithal, present in amphibians; and pronounced teloleucithal, present in reptiles and birds. Okay, and at last we have centroleucithal egg with yolk in the center middle of the egg you have the yolk, then it is called centroleucithal. This is oligolecithal type of egg, example arthropod and some cylindrita. Okay, this is your isoleucithal. Okay, this is your centroleucithal. In the center of that egg you have the yolk. Okay, this blue part is your yolk, and this is the cytoplasm and the middle of the nucleus. Okay, and nucleus. This is your teloleucithal egg, which is seen in the bird. Okay, in which the yolk is present at the terminus and nucleus is present on other terminus of the egg, and cytoplasm is there. Okay, and this is your isoleucithal egg, in which yolk is present outside, and in the middle you have cytoplasm, and the middle of that cytoplasm you have the nucleus of the egg. Okay, next is classification of egg based on the amount of yolk. So we have two: oligolecithal egg and polylecithal egg. Again, oligolecithal egg is of two types: alecithal and microlecithal. Alecithal means there is no yolk, yolk is very less. Then microlecithal, small amount of yolk is present inside the egg. Then polylecithal egg, mesolecithal and macrolecithal. Mesolecithal means more amount of egg yolk is present, and macrolecithal means large amount of yolk is present. Okay, so oligolecithal, alecithal egg, egg containing no yolk, example eutherians like us. Okay, then microlecithal egg containing very little yolk, example sea urchin, amphioxus, tunicates. Okay, this is alecithal egg and there is no yolk. Okay, and this is microlecithal egg, there is yolk which is scattered inside the egg. Okay, next we have polylecithal egg containing large amount of yolk. Two types: mesolecithal egg and macrolecithal egg. Mesolecithal egg containing moderate amount of yolk, example dipnoi and amphibians. Okay, like this. Okay, this part is only you have the yolk. Okay, and remaining part having that cytoplasm and nucleus. See, small amount of yolk is there. Then this is your mesolecithal, and at last we have macrolecithal egg containing very large amount of yolk, example bony fish, amphibians, reptiles, and birds. The anda of hen. Okay, the egg of hen we eat in the breakfast, it is a type of macrolecithal egg. It contain large amount of yolk, the yellow part of that egg. Okay, which we eat in the breakfast, it is yolk, and it is a type of macrolecithal egg. Okay, so that is your reproductive system, reproductive human reproduction. Now it's for third chapter, that is your reproduction in flowering part, in 5 minutes. Thank you. Okay. Now, sexual reproduction in flowering plants or angiosperms. Okay, reproduction in angiosperm or plants. Okay, so first of all, we have to discuss something related to the flower. Okay, flower, it is a sexual structure present in angiosperm. Okay, so flower have been a great importance of human. It has been aesthetic, ornamental, social, cultural, and religious value. Okay, in religious events, you will use flower. In ornamental purpose, to decorate your house, to decorate an event, you will use flower. So these all are different uses of flower. To give a sorrow feeling, to give love feeling, to tell the love feeling, you will use flower. Okay, so horticulture is the science of cultivation and breeding and marketing of flower. Okay, flower are structurally divided into following different worlds: calyx, corolla, androecium, gynoecium. Calyx, group of sepal usually green in color. Corolla, group of petal brightly colored except some green things. Okay. Androecium, male. Reproductive part of flower consists of stamen having filament and anther. Gynoecium, female. Reproductive part of flower known as pistil and carpel consists of stigma, style and ovary. Now I have a flower for you. You can easily see in this different parts of flower. Okay. So in this flower you will see that these are the petals, okay, which we call corolla. Okay, here we have some green parts, okay, which we call calyx sepal. Okay, then we have androecium. This is your androecium, okay, male part. Okay, this is your androecium male part, in which you will find this is your filament and this is your anther. Okay, this top part is your anther and this is your filament. Okay, next one is your gynoecium, female reproductive part, which is having stigma, style and ovary. Okay, so this is your female reproductive part of the flower, in which this is your stigma, then you have style, this is your style, and inside here you have ovary. Okay, so this is the female reproductive parts of a flower. Okay, so they are mainly the structure of hormonal changes that take place in the formation of the flower. These changes lead to the modification of flower primordium, this arrangement of different whorls of flower. See how flower formation take place. First we see that. Okay, see this. This is a axial bud. Okay, a stem which having axial terminal bud. Okay, now on this bud, nearby some area, you having a leaf. Okay, this leaf start to secrete a flowering hormone and plant hormone which is called phlorogen. Okay, and this phlorogen reach to that apical meristem region and convert that apical meristem into flowering meristem or flowering area. Okay, this is your phlorogen. It will go to the apical meristem region and convert your apical meristem into that flowering meristem. Okay, now this flowering meristem will convert into floral bud and floral bud convert into flower. So formation of flower is structural as well as hormonal changes taking place inside the flower. Okay, so sexual reproduction in flowering plant has been divided into following events: pre-fertilization, in which we are going to study it about structure and the formation of gamete, gametogenesis, that megagametogenesis and microgametogenesis, and fertilization events and post-fertilization event, in which we are going to study different structure, that is your seed, fruit, that endosperm, zygote, embryo, all these things. Okay, so this is your flower, this is your end petal, this is your sepal, this is your gynoecium, in which this is stigma, this is style, this is ovary, and this is your anther, this is your filament, this is your androecium. Okay, so structure of stamen first. Pre-fertilization structure and event, first we have male reproductive organ, that is stamen. Okay, stamen consists of two parts: filament, sterile part which connect anther to the thalamus of the petal of the flower. Then anther, fertile part within the which form the pollen grain. Okay, this is the fertile part. This is the responsible for development of sex organ, sex that pollen grains inside the your flowers. Okay. Fertile part within which the formation of pollen grain take place. Anther usually bilobe. Each lobe has two theca and thus known as dithecous. Each theca has two microsporangia, four in total, and thus typical angiosperm is tetrasporangiate. Okay. See. This is two theca. Okay. In these two theca, you have regions microsporangiums. Okay. So thus typical angiosperm anther is tetrasporangiate 1, 2, 3, 4. 4 sporangium, that's why tetrasporangiate, and 2 theca. 1, 2. 2 theca, then it is called dithecous. Okay. So each microspongin develop and become pollen sac that produce large number of microspore known as pollen grains. Stamen is equivalent to microsporophyll. This is again important line related to the NEET also. Stamen is equivalent to microsporophyll. In the end of the chapter, I will tell you different equivalent structure, different naming of the structure. So anther development is anther is an parenchymatous in nature. The primary parental cell undergoes division to form 3-5 layer of anther wall. Okay, now we're going to see the walls of anther. Okay, so first wall, it is your epidermis, it is the outermost wall. Then we have endothelium, it is the inner second layer of the anther wall layers. It occurs from the radial bands of the tangential wall. This is thickening made up of alpha cellulose with the help of pectin and lignin in some cases. It helps in the dissense of anther and dispersal of the pollen. Again, most important line of your endothelium, that it helps. Again, the most important line of endothelium, that it helps in dissense of anther and dispersal of it helps in dissense in the anther and dispersal of pollen grains. Okay. Then we have middle layer. Middle layer is ranges to 1 to 6, or in some cases several middle layers are present. They provide nutrition and also help in the formation of pollen kit. This is again the important point, that they help in provide nutrition and also help in the formation of pollen kit. Next we have tapetum, that is the innermost layer of anther. It is the innermost layer of anther which act as a nutritive layer. This is a single layer thick. The cells of tapetum possesses dense cytoplasm and generally have more than one nucleus. Okay. The cell of tapetum are initially diploid, but they become polyploid and multinucleated due to the endomitosis and free nuclear division respectively. Okay, tapetum absorb food from middle layer and promote nutrition to the microspore mother cell for developing of microspores. Okay, so this is your four layers. First one is your epidermis, second one is your endothelium, then middle layer, and at last the tapetum. Tapetum is the innermost layer which is important for the nourishment of microsporangiums. Okay, the functions of tapetum. Tapetum provide nutrition to the sporangium tissue, mmc, and pollen mother cells, developing pollen grains, secretion of enzymes and hormones, secretion of sporopollen. What is important word, most important function of your tapetum, that is sporopollen release. Okay, formation of proboscis body. Proboscis body plus sporopollination is equal to hibiscus body. Okay, hibiscus body is mainly made up of a complex substance called sporopollen, which is a polymer of carotenoid. Okay, secretion of pollen kit substance. Pollen kit is made up of lipid and carotenoid. It will help in the pollen dispersal system mechanism. Okay. Next, we have structure of microsporangium. How your microsporangium look like? What is the characteristics of your microsporangium? We will see. It will see in this topic. Okay. So in transverse section, a typical microsporangium appear near circular in outline. When the anther is young, a group of compactly arranged homologous cells called the sporangious tissue occupies the center of each microsporangium. See. This is your microsporangium. ऐसे four होते हैं. Okay, so in the middle you will see there is some cells. Okay, it will do mitosis and meiosis and convert it into your microspore mother cells. Okay, here. So when the anther is young and a group of compactly arranged homogeneous cells called the sporangious tissue occupy the center of the each microsporangium. Four pollen sacs are present in the anther. Inside the pollen sac, microspore pollen grain are formed by the meiotic division of microspore mother cells. See, in this you will see different mitosis, meiosis, okay, that we are going to study. Okay, as the each cell of sporogenous tissue is capable of giving rise to a microspore tetrad, each one is potential pollen mother cell of microspore mother cell. Okay, each microspore tetrad is potential to form a microspore mother cell. Okay, this is your microsporogenesis. Okay, microsporocyte, it will go under meiosis 1 and convert into a meiosis 1 and two nucleus like structure. Then again it will go into meiosis 2nd and you will find 4 haploid cells. Okay, now on that haploid nuclei your that layer of callus will develop, and this is called tetrad. Okay, now that tetrad will dissolve and it will convert into your pollen grain. Okay, callus enzyme will come and dissolve callus outside that, and it will convert into this microspore will convert into your pollen grain. Okay, so this is release microspore. It will become, it contain two cells. Okay, one is vegetative cell and again other one is your generative cell. Okay, see, in this you can easily see. Okay, this is your haploid nuclei, two haploid nuclei. Meiosis 2nd take place, four haploid nuclei you have. Now these 4 haploid nuclei will covered by callus layer, and after that one microspore will take out, and in which you have pollen mitosis 1. Okay, mitosis 1 take place. Okay, from that mitosis 1 you will get a vegetative nuclei and generative nuclei. After that, that mitosis 2nd take place inside the female. There's two chances. 60% plant do mitosis inside the pollen sac, and remaining 40% will do inside the female inside the female body. Okay, we will see that again. Okay, see, structure of pollen grain. This is their pollen grain. Okay, so it having two layers, exine and intine. Exine is the outermost layer which having the covering of sporopollen, the world most hardest substance in plant kingdom. Okay, then we have intine, innermost layer, in which you have two cells. One is generative cell and other one is your generative cell, and other one is your vegetative cell. Okay, so pollen grain are generally spherical, measuring about 25 to 50 micrometer in diameter. It is a prominent two-layer wall. The outer wall layer is thick, rigid, and ornamental, called exine. This layer is formed. It is formed of sporopollen. Okay, sporopollen is one of the most resistant organic material known. It is a non-biodegradable. It can withstand high temperature, strong acid, and alkaline. Number of enzymes that degrade sporopollen is not so far known. Okay, pollen grain are also well preserved as fossil because of the presence of sporopollen. Okay, the inner wall of pollen grain is thin, continuous, soft, and elastic in nature. It is called intine, and it is made up of pectin and cellulose or pectocellulose. Okay, means your intine, it is made up of pectin and cellulose. Okay, the cytoplasm of pollen grain is surrounded by plasma membrane. A few places exine is usually absent. At that places are called germ pore, from where your pollen tubes come out. Okay, then some special points about pollen grains is present in your NCERT. First is your translator apparatus. It is an aspirated calotropis and oxidase family. The pollen grains joined together to form a bag-like pollinium. Pollinium of calotropis is constituent of translator apparatus. Then aeroallergens. Okay, pollen grains of many species which can present in the air causes allergy and bronchial action. Applications are called aeroallergen. Examples: Chenopodium, Parthenium, carrot grass, sorghum, and amaranthus. Hay fever is caused by pollen of ambrosia. Okay. Now pollen tablets and syrup, this is taken by many athletes to increase their efficiency. Okay, pollen grains are rich in nutrients. It become a fashion in recent year. that pollen tablet and food supplement pollen consumption has been claimed to increase the performance of athletes and racing horses. Okay, next is longest pollen is of Zostera seagrass, and viability of pollen grain in some cereals such as rice and wheat. Pollen grain loses viability within 30 minutes of their release. Okay, means after 30 minutes of the release of pollen grain from that anther, it will dead. Okay, in members of Rosinaceae, Leguminosae and Solanaceae, they maintain viability for months. The period for which pollen grain remain viable at high variable and some extent depend on the prevailing temperature and humidity. How is the temperature outside in the nature? What is the humidity of that nature will define the variability of pollen grain. Okay. Then pollen bank. It is a possible to store pollen grain of large number of species for years in a liquid nitrogen minus 196 degrees Celsius. If that particular species get extincted, then from where we will bring the pollen grain, from where we will bring the cell. So we will preserve their eggs, their ovas, their ovules inside pollen that banks in which we put all these things in minus 196 degrees Celsius temperature. Okay, this is your Calotropis, that is your translator apparatus. This is your Parthenium carrot grass. We have also this thing with us. Okay, this is your Parthenium carrot grass, responsible for hay fever and other bronchial disorders. In ecology chapter, we also going to study this thing as an alien species. Okay. Next one is your, this is Zostera, longest pollen grain, and this is your pollen bank, having that pollen grain stored at minus 196 degrees Celsius temperature. Okay. Next is your anther dehiscence. Now your anther become mature, it will preferable to do dehiscence by which pollen grain come out from your pollen sac. That thing is called anther dehiscence. So dehiscence of anther occur due to the endothecium layer. The point where the endothecium layer breaks is known as stomium, this area. Okay, this area of your anther is called stomium, where the breakage of anther take place, dehiscence of anther take place. Okay, in mature anther, only two well layer epidermis endothelium present in the form of outer covering. Okay, the sterile tissue present between the both pollen sacs of either lobe degenerates of both pollen sacs of each anther lobe fused together to form single pollen sac. Means these layer will be fused to each other. These layer will be fused to each other and this become a single pollen sac. This become a single pollen sac. Means when your anther is not mature, at that time you have four pollen sacs. But when your anther become matured, you having only two pollen sac. Pollen sac number one, pollen sac number two. Okay. Therefore, in the mature anther, only two pollen sacs are present. Dehiscence of anther take place during the dry season. Loss of water take place from the cell of endometrium in dry condition. That's why dehiscence of anther take place. Okay, so this is your microgametogenesis or development of male gametophyte. How male gametophyte development? Okay, so it will go in two steps: pre-pollination development and post-pollination development. Means before pollination, what is the development of pollen grain take place, and after pollination, what is the development of pollen grain take place. Okay, same thing we studied previously, same thing is here. Okay, so pre-pollination development. Okay, in the beginning of this process, nucleus of pollen grain divide by unequal mitosis division, resulting of two unequal sized nuclei forming small nucleus which is present near the wall of called dendritic cells, and a large irregular nucleus which is present inside the cytoplasm called tubular nucleus or vegetative nucleus. Okay, larger cell is which larger irregular shaped nucleus present is known as vegetative cell. Okay, this is the nucleus size in which you have seen this cell. It is your large cell which is called vegetative cell, and this is the small cell which is called generative cell. Okay, so when pollen grain is mature, it contains two cell, which is your vegetative cell, another one is generative cell. This stage of pollen grain is called partially developed male gametophyte or mature pollen grain. See how this thing will form. Okay, this is your microspore. Okay, it contains a nucleus. Now this nucleus do division and it will convert into two nuclei. Okay, now one nuclei will go here to the pole, second will go here pole. Okay, after that cytokinesis take place. Okay, formation of that cell wall. a cell membrane. Okay, so this large cell of this cell is called vegetative cell, and this small cell is called generative cell, and this whole structure is called mature pollen grain. Okay, mature pollen grain. This is the mature pollen grain. Okay, so post-pollination development, further development of pollen grain take place on the stigma of carpel after pollination. Okay, so first of the vegetative nucleus enter into the pollen tube and assume terminal tip position. The spindle-shaped generative cell non enter into the pollen tube. Inside the pollen tube, generative cell divide mitocally and forming two non-motile male gamete. Now male gametophyte become three cell structure in which one vegetative cell and two male gametes are present. See now what happened? This two cell stage when go inside the stigma, okay, inside the female reproductive system, this generative cell will do again mitosis, okay, and this is your three cell stage or mature male gametophyte which having three cells. This is your vegetative cell, this is your two gamete cells or gametophytic cell or generative cells which do mitosis inside the female reproductive system of flower. Okay, this is three cell stage represent the mature male gametophyte of angiosperm. Male gametophyte of angiosperm is highly reduced and completely depend on sporocyte. Okay, in bryophyte, in algae, we studied that in biological classifier in the plant kingdom in 11th class that the sporophyte is dependent on gametophyte. But in angiosperm and gymnosperm, your gametophyte is fully dependent on sporophyte. Okay. Next, we have pre-fertilization structure in female, gynoecium. Okay, female reproductive part, gynoecium. It have a single pistil or many than one pistil multicarpally. If a single pistil, then it is called monocarpally. If it have many pistils, mean more than one pistil, then it is called multicarpally. Okay, multicarpally the pistils may be fused together, then it is called syncarpus, and if it is free, then it is called apocarpus. These are the three examples. This is an NCERT pick in which we have hibiscus pistil, which is a single pistil. Okay, then we have multicarpally syncarpus pistil of papaver. Okay, and multicarpally apocarpus gynoecium of michelia. Okay. Next, gynoecium. Gynoecium have three parts. Okay, stigma, that is the landing platform of pollen grain. Then style, elongated slender part beside the stigma, which is connects the stigma with ovary, and ovary basal bulge part where ovule is present. Okay. Gynoecium inside ovary is the ovarian cavity locule in which placenta is located. Arising from the placenta are ovules, megasporangium. The number of ovules in an ovary may be one, just like in beet, paddy, and mango, and too many in papaya, watermelon. These ovules in future converts into seed, and the ovary in future convert into fruit. Okay, so the part of mango you eat is ovary. When the flower, the mango is in flower stage. Okay, and the seed which you throw out, it is an ovule. Okay, so structure of megasporangium, ovule. Okay, ovule is attached to the placenta by stalk called funicle. Junction between the body of ovicle and funicle is called hilum. Each ovule has 1 to 2 protective envelopes which called integuments, except the tip where the small opening which is called micropyle is present. Opposite the micropyle end, it is your chalazal end. See, I will draw here the diagram of your ovule. Okay. So this is your chalazal end. This is your micropylar end. These two are integuments. This is outer one, this is inner one. Okay. Then this is your funicle stalk. Okay. And this is the area where your funicle is get attached with ovule, which is called hilum. Okay, and this area particular this area is called nucellus, and this is the site where your megasporogenesis starts. Okay, same thing is here, same diagram is here. Okay, next we have enclosed within the integument there is a mass of cell called nucellus. Okay, here in the cell contain reserved food material. Inside the nucellus is the embryo sac, female gametophyte, and ovules generally have a single embryo sac formed from a megaspore. Okay, now megasporogenesis, how your megaspore is formed. It is the formation of megaspore from megaspore mother cell. Okay, ovule generally differentiate a single MMC into micropylar regions of the nucellus. It is the large cell containing dense cytoplasm and the proponent nucleus. MMC undergoes meiosis to produce 4 megaspore. Now I will show you how that 4 megaspore will produce. Okay. See, this is your ovule, in which we have nucellus. Okay, now nucellus have two types of cells. Okay, these two type of cells are called macroprogenitor cells or progenitor cell and archegonial cells. Okay, now this archegonial cell will differentiate into two. Okay, and forms your ovule. Okay, now what happened in your ovule? Meiosis take place. Okay, meiosis 1, meiosis 2, that's how. Okay, and that's how four different haploid cells will develop. Now these three cells send its nutrition to this one, and this alone megaspore will develop. Okay, that archegonial cell convert into megaspore mother cell, and megaspore mother cell will convert into megaspore, 4 megaspore, by the help of process of meiosis. Okay, now this megaspore cell undergoes in mitotic divisions inside that. Okay, this megaspore cell undergoes in mitosis division. Okay, let's see that thing. Okay, now first we have, I will draw here. This is your functional megaspore. Okay. Now this functional will go into karyokinesis 1. Okay, and form two nuclei. Now these two nuclei again do karyokinesis, which we call karyokinesis 2. Okay, now we have 4 nucleus in a cell. Now these 4 nuclei will move towards the pole of the cell. Okay, and again do karyokinesis, that is your karyokinesis 3. Okay, okay, now two cells from that all that 8 cell structure, 8 nucleus structure will move toward the center. Okay, 1 2 3, 1 2 3, and these two cells, these two nucleus will come in the center. Okay, now cytokinesis will take, cell membrane will be formation take place. Okay, now we will see that different cells will develop. Okay, so at last we have embryo sac, in which you have 1 2 3 4 5 6 7 cell and 8 nucleus. 1 2 3 4 5 6 7, these are the 7 cells. 1 2 3 4 5 6 7 and 8 nucleus. 1 2 3 4 5 6 7 8. Okay, again see, 1 1 2 3 4 5 6 7, these are the 7 cells, and 1 2 3 4 5 6 7 8, this is 8 nucleus. So at last we have 7 celled 8 nucleated structure, which we call female mature female gametophyte. Okay. Okay, so this is your female gametophyte in which you have 1, 2, 3, 4, 5, 6, 7. 7 cell and 8 nucleus. 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleus. Okay, this is your NCERT diagram. And at last you have mature embryo sac. Again you can see 1, 2, 3. Okay, these three are called antipodal cell and it is present at chalazal end, and there you have three cells which is called, two are called synergid and one is your egg cell. Okay, in which it is present on micropylar end, and there is in the synergid we have filiform apparatus which is responsible for the pollen tube movement towards your sphere, that filiform apparatus. And in the center we have two polar nuclei. The cell is called central cell. Hence we have a structure which is called 7-celled 8-nucleated structure. Okay, here. Okay. Next we have pollination. Okay. Now pollination is the process of transfer of pollen grain from anther to stigma. Okay. So it is a transfer of pollen grain from anther to stigma of a pistil. Okay. So pollination, it is of three types. Okay. Basically two types, then the two types have again three types. Okay. Autogamy, self-pollination, genetogamy and xenogamy. Okay. So first we have autogamy, which is also called self-pollination. Okay. See, if a flower having both anther and your stigma, then it will transfer that pollen grain from that anther to stigma, then it is called autogamy, self-pollination. A single flower, just like this, you having a flower. In this flower, you having stigma and anther, both the thing, then this type of pollination is called self-pollination. They are pollinating self. Okay. Self-pollination again, it is of two types, chesmogamous and clistogamous. Okay, so plants like Viola, common pansy, Oxalis communalia produces two types of flower. One is chesmogamous flower and other one is clistogamous flower. Okay, chesmogamous flower are open flower and clistogamous flower are closed flower. Inside that already self-pollination take place. In this cross pollination or your genetogamy or xenotogamy will take place. In this your autogamy take place. Okay, autogamy, chesmogamous flower, they are similar to the flower of other species with exposed anther and stigma. This is your chesmogamous flower. Then clistogamous flower, they do not open at all. Anther and stigma lie close to each other. They are autogamous in nature, means autogamy take place in them. When anther dices in the flower bud, pollen grain come in contact with stigma for pollination. Clistogamous flower produce assured seed set event. In the absence of pollinator, clistogamous lead to inbreeding depression. Inbreeding depression mean when you do selfing between the same family, then you will face inbreeding depression in which recessive character will come out. Okay, so this is your clistogamous flower. Next is your genetogamy, means a plant having both flower, male and female, but that flower, male and female flower is not present in a single flower. Then it is called genetogamy. Means if I am in this plant, okay, and here your female flower is there and here male flower is there, then from this male flower to this female flower, when pollen grain is go, then it is called genetogamy. Okay, means flower are present on a same plant, both male and female flower are present in same plant, but they are not present in a single bulb. Okay, then it is your genetogamy. It is a transfer of pollen grain from the anther to the stigma of another flower of same plant. It is a functional cross pollination involving pollinating agent, but it is genetically similar to autogamy. Ecologically, it is cross pollination, but genetically, it is self pollination, because plant is the same, na body is same, only the flower is different. Either female है, either male है, male से female के पास चला गया pollen grain, but the body is of your on a single thing. Okay, that's why genetically it is autogamy, but ecologically and functionally it is cross pollination. Okay, and last type is your genogamy, in which both flower are present on different flower, that different plant. Male plant is different, female plant is different. Then it is called genogamy. Okay, it is a transfer of pollen grain from anther to the stigma of different plants. See, This is plant A, this is plant B, both having that female flower on different parts. Okay, this having male flower, this having female flower, and it will go from here to here. Body is different, flower is different, then it is xenogamy. Okay, now agents of pollination, we have different agents which causes pollination in the nature. Okay, so abiotic one is by wind and water, and biotic one is insect, bird, bat, primates, aerobial rodents, reptiles, etc. Okay, so abiotic agents pollination by wind is called anemophily. Okay, more common abiotic agent, most common abiotic agent, it is not more, it is most. Okay, wind pollinated flower often have single ovule. Okay, in each ovary, and numerous pollen packed flower packed into an inflorescence. When you see a wind pollinated flower, you will see an inflorescence. Okay, example, corn cob, the tassels are the stigma and the style which wave in the wind to trap pollen grains. Okay, wind pollination is quite common in grass family, poaceae family. Okay, then abiotic wind way of effective pollination, flower produces large amount of pollen. Now there is no pollinating agent, you are having a abiotic pollinating agent which you have, it is not in your control. Okay, so you have to produce large amount of pollen कि कुछ ना कुछ तो जाके pollinate होए, कोई ना कोई तो जाके pollinate होए उससे. Okay, then pollen grain are lighter and non sticky because you have to make them fly in the air, so it should be lighter and non sticky. Okay, they often possesses well exposed stamen for easy dispersal of pollen into wind's current. Okay, their stamen are well exposed, they are fluttering in the air, they are moving in the air freely. Okay, large feathery stigma to trap airborne pollen grain. Now in their stigma is large. Okay, large platform, and they are feathery in nature because they have to capture these pollen grain which is thundering in the air. Okay, abiotic agent, water. Okay, pollination by water is called hydrophily. Okay, it is quite rare, it is limited to about 30 genera, mostly in monopod. Okay, in monopod you will see this thing. Okay, example, Vallisneria hydrelia, fresh water, and Juicera marine seagrass. But in lower plants, water is regular mode of transport for the male gamete, just like in pteridophytes, bryophytes, and algae. Okay, so abiotic water, अब इसमें क्या होता है, जो Vallisneria होता है, female flower reaches the surface of water by long stalk, and the male flower or pollen grain are released onto the surface of water. They are carried by wave current, water currents, and reach the female flower. In seagrass, female flower remains submerged into the water. Pollen grains are long and ribbon-like. They are carried inside the water and reaches to the stigma. Okay. The pollen grain of the most of the water pollinated species have mucilagous covering to protect them from wetting. Okay. If they do not have mucilagous covering, they can easily sink inside the water. Not all aquatic plants use hydrophily. For example, your water hyacinth and water lily, these. Okay, these do not use hydrophily. Okay, they mostly use insect or anemophily. Okay, the flower emerges above the level of water for entomophily or anemophily. Entomophily, that is insect pollination, animal pollination, and your anemophily is your wind pollination. Okay, so wind and water pollinated flowers are not very colorful and do not produce nectar. Okay, next we have zoophily. Okay, entomophily by insect, ornithophily by birds, chiroptophily by bat, malacophily by snail, and ophiophily by snake. Okay, and melitophily by bee, psycholophily by butterfly, canthenophily by beetles, cephenophily by wasp, and palenophily by moths. Okay, so these are different. Zoophilus means pollination by animal. Okay, biotic factor. So biotic agents, animals used in majority of flowering plants. Example, bees, butterfly, flies, beetle, wasp, ants, moth, birds, bats, primates, rodents, reptiles, etc. These all do pollination. Okay, and the pollination by insect is called entomophily. Okay, particular bee is the most common type of entomophilic agent. Okay, often flower of animal pollinated plants and especially adapted for a particular species of animal. Okay, features of insect pollinated flower, that is most important line. Large, colorful, fragment, and rich in nectar. Because how that flower, the cost of the pollination in anemophilous flower is nectar. Insect come in the greed of nectar, the pollens get attached to their body, and when they go in the next flower to take the nectar, they will accidentally do pollination. Okay, so nectar and pollen grain are the floral rewards for pollination. Small flower from fluorescence to make them visible. The flower pollinated by flies and beetles secrete foul odors to attract these animals. For example, Rafflesia and Ammophilus, these all are the largest and widest flower on earth, and they will produce smell like rotten meat and rotten egg to attract fly and beetles. Okay, the pollen grains are generally sticky in nature. Okay, because they have to attach to the body of that pollinating agent, that's why it is sticky in nature. Okay, this is your Amorphilus, world longest flower. Okay, and its smell is just like a rotten meat. Okay, this is yucca plant, and its pollinating agent is only a one plant in one insect, which is your proboscis moth. Okay, so Amorphophilus, it is the tallest flower of 6 feet in height, and it is not contain any scent, any good smell. It having smell of rotten meat. Okay, and the pollinating agent of this flower is that flies. Okay, dead flies. Okay, castor flies that flies on dead bodies. Okay, then the moth species and the plant yucca cannot complete their life cycle without each other. The moth deposits its egg in the locule of the ovary. The flower get pollinated by moth. The larva come out of the egg and seed start to developing. Okay, this is the example of mutualism. Okay, if one is removed, then other will be get extincted. Okay, many insects consume pollen or nectar without bringing about pollination. Then they are called pollen robbers or nectar robbers, like bumblebee. Okay. Next, pollination by snake is called orthenophily. They are found in Michelia and sandal. Red sandal, लाल चंदन का pollination करता है सांप. Okay. Then malecophily is referred to as the pollination by bat, snail perform pollination. Ariscania, snake or cobra plant which has the septa mimicking a snake hood. Some arum, lilies and lemma and ovulus, these all do pollination by the help of snails. Okay, then you have chiroptophily, in which the pollination is done by bat. Okay, so next is your artificial hybridization. Then means inside your house you can perform that. Artificial hybridization means you can perform genogamy. Okay, so the process artificial hybridization is best way to produce commercial proof. It helps in breeding to obtain plant with desirable characters. Okay, Mendel also do this hybridization to produce different traits of pea plant. Okay, in the flower is unisexual pistillate, then emasculation is not done, but bagging is done before the stigma become receptive and again after pollination. Okay, so this is the steps of your artificial hybridization. That is, first we do emasculation, after that artificial pollination, and at last we do bagging. In bagging we write the time and date of that thing, which is also called tagging. Okay, so then outbreeding devices. Okay, certain devices in plant ensure that cell pollination does not take place, because cell pollination brings inbreeding depression. So we have to avoid that inbreeding depression. Then we use outbreeding devices. Okay, so first we have unisexual flower. Okay, Diosis condition prevents cell pollination. Example, papaya, means female plant different, male plant different. Then it is called dioecious condition. Okay, then dichogamy. Protogamy, the pistil mature before the anther. Example, wheat, protandry. The anther mature before the pistil. Example, ivy. Okay, dichogamy, in which we have two conditions. One is protogamy, in which that gynoecium mature first and anther mature less. Automatically fertilization stops. Okay, and protoandry, in which the anther mature first and gynoecium mature late. Automatically pollination does not take place. Then herocogamy, the sapiential separation of anther and stigma. Example, oxalis. Okay, and self impeccability, genetical mechanism that prevention self pollination. Example, petunia. Okay, next is pollen pistil interaction. Okay, now your pollen is landed on stigma, and now it is will converted into pollen tube. It will transport its gametes by pollen tube inside the stigma. Okay, that thing is called pollen pistil interaction, in which phyllopium apparatus give the major role. Okay, this is your pollen pistil interaction. Okay, in which pollen grain sits on the stigma and now pollen tube come out. In that pollen tube you have that one generative cell which will do mitosis and convert into two germ cells, that is gametic cell or your generative cells. Now that thing reaches to your ovary, embryo sac, and enter towards the micropyle and inside your embryo sac. Okay, now double fertilization. Okay, after that fertilization take place. Now your both gametes, the male gametes and female gametes get in the contact, and that thing is your double fertilization. After entering one of the synergid, the pollen tube releases the two male gametes into the cytoplasm of synergid. One male gamete moves toward the egg cell and fuse with the nucellus, which we call syngamy, and forms zygote. Okay, and the other male gamete moves towards the. Okay, so double fertilization means in this you have two steps. One is syngamy and other one is your triple fusion. In syngamy, one generative cell will get fused with your egg cell and forms zygote. Okay, while other is triple fusion, in which one generative cell get fused with your two polar nuclei and forms a 3N structure, which we call perisperm or perinucleus. Okay, which is the process is called triple fusion, and when both the things is going simultaneously, then it is called double fertilization. Okay, next, that Strasburger in 1884 in Monotropa give that the term syngamy. Okay, the second male gamete fuses with two polar nuclei and the secondary nucleus formed by the fusion of two central cells called nuclei. The secondary fertilization most of the plant involves three nuclei, and it is called triple fusion. Means your two polar nuclei fuses with one that generative nucleus, and form a 3N structure, which we call primary endosperm pen, which is your precursor of your pre-nuclear endosperm, which is precursor of pre-endosperm. Okay, since two type of fusion, syngamy and triple fusion, is taking place, that's why it is called double fertilization. It is an event unique to flowering plant, and it happens only in angiosperm. Okay, next, the central cell after triple fusion become primary endosperm cell pack. Okay, and develop into endosperm. Okay, primary endosperm nucleus, it will convert into primary endosperm cell, and then primary endosperm cell convert into endosperm. Okay, zygote develop into embryo and antipodal cell, and remaining synergid degenerate after fertilization, and this time zygote obtain food from degenerating synergid and antipodal cell. Means when your antipodal cell and synergid get degenerate, at that time they will transferring their nutrition to developing embryo. Okay, and they will degenerate. So next we have post-fertilization events: endosperm development, embryo development, ovule into seed, and ovary into the seed. These are the four post-fertilization events. Okay. So first we have endosperm. How your endosperm is formed. Okay. So endosperm development precedes embryo development. The peck divide repeatedly and form a triploid endosperm tissue, which we call endosperm. Okay, the cell of this tissue is filled with reserved food material and are used for nutrition of developing embryo. It is highly nutritive structure which will provide nutrition to developing embryo. Okay, so the endosperm is a following type on the basis of development. First is nuclear endosperm or free nuclear endosperm, which we see in coconut. Okay, the coconut water or tender coconut is free nuclear endosperm. Next we have cellular endosperm. This type of endosperm is found in member of Gamopetalae family. Okay, during this the development, each division primary endosperm nucleus is followed by cytokinesis, so endosperm remains cellular from the beginning. Okay, in cellular endosperm you will find different cells are present, but in nuclear endosperm you will find that all the nucleus are fused and form a single cell like structure, which is liquid in nature, which we drink in coconut water, which we drink in the form of coconut water. Okay, so this is your tender coconut water is free nuclear endosperm made up of thousands of nuclei, and the surrounding white kernel is the cellular endosperm. This watery fluid is your free nuclear endosperm, and this white part, okay, which we eat in coconut, it is cellular endosperm. Okay, next is your development of embryo. Okay, now your development of embryo take place, in which the process of development of embryo from zygote is called embryogenesis or embryology, Embryo develop at the micropylar end of embryo sac where the zygote is situated. Okay, means at this site, okay, micropylar site, your embryo development take place mainly. Okay. So dicotyledon embryo, the zygote divide transversely to form two cells. The cells that lie toward the micropylar is known as the basal or suspensor cell. The other cell forms toward the chalazal end is known as apical or embryonal cell or embryonal axis cell. Okay, the basal cell divide transversely and form two cells suspensor. The embryonal cell divide vertically to form embryonal cell. Means this type of division take place. See. This type of development take place inside the embryo development in dicotyledon embryo. Okay, the two embryonal cells further divided to form four embryo cells known as quadrant stage. Okay, the two suspensor cells divide by transfer division and form a long filamentous-like structure called suspensor. Suspensor push the development embryo into endosperm of nutrition for nutrition. Okay, the micropylar send the suspensor swell up and known as posterior cell. The cell of suspension near to the embryonal cell is known hypophysis. Okay, this is your development stages of embryo development in dicot. Okay, you can easily see this is your globular stage, it will develop upward, this is your suspensor, and there is your heart shaped embryo, it will convert into cotyledons. Okay, next we have monocotyledon embryo. The zygote divides by transfer division and forms into two the cells. The upper part cell toward the chalazal end is known as embryonal cell again, while the lower cell toward the micropyle is called basal cell. Same thing. The basal cell increases in size and form vesicular suspensor. Okay. Then the embryonal cell divide to form a terminal cell known as cotyledon and the lower cell known as the embryonal cell. Same thing, the same pattern which is present in your dicot is present in monocot. The difference is the structures. The transfer division in embryonal axil give rise to the cells. One of the give rise to plumule initial, other give to radical initially. Plumule initiate divides to form a plumule while radical initiate divide to form radical. The embryo of monocot possesses only one cotyledon. It have only one cotyledon. Okay, but in dicot you have dicotyledon. That's why it is called dicot. Okay, in Gramineae the cotyledon is known as subtulum, that is present on the lateral side. This is your subtulum. Okay, it acts like a shield. Okay, the embryo axis has a radical and root cap enclosed by sheet called cauloprisa there, and the epicotyle have plumule which covered by polyptiles here. Okay. Next is fruit. Okay, your ovary turns into the fruit, the edible part. Okay, so in some there's two types of fruit. One is false fruit, and another one is true fruit. False fruit means the fruit develop other than ovary, then it is called false fruit. For example, your apple, in which the fruit is developed from thalamus. Then we have another example of false fruit, that is strawberry, in which that is developed by acne and receptacle. Okay, so example of that fruit is your mango, tomato. False fruit is a fruit in which other parts of flower also take part in formation of fruit along with ovary. Example, strawberry, apple, cashew, fleshy thalamus of these fruits are eaten. Okay, in fruit the wall of ovary develop into pericarp. Some fruit develop without fertilization. Such fruit is known as parthenocarpic fruit. In the process is called parthenogenesis or parthenocarpy. These fruit do not produce seed. Example, banana, citrus. Some parthenocarpic fruits are developed artificially by the application of auxin and gibberellin like hormones. Okay. Next we have seed. Okay. The ovule in the ovary develop into seed. These seeds are basically of two types. One is your albuminous or endospermic seed. Another one is non-albinous or non-endospermic seed. Here one thing is very important that you have to remember, that example. Most of the monocot seed, example wheat, rice, cotton, barley and maize etc. are endospermic. Endospermic seed के example होते हैं monocot seed. But there is one monocot seed which is the orchid seed. It is a albuminous, non-endospermic seed. Okay, means it does not have endosperm. Albuminous or endospermic seed are those seed in which endosperm remains preserved, and these belongs to monocot family. But there is one monocot which is called orchid. It is non-endospermic in nature. Okay. Next we have non-albuminous or non-endospermic seed in which endosperm is consumed during the development of seed, and the examples are most of the dicot. But there is one exception, which is your castor. It is an dicot, but its seed is endospermic in nature. And this thing I ask in NEET exam mainly, in which it will ask that which one of the following is your endospermic seed, and they will give in option wheat, rice, coconut, and orchid. And orchid is your non-endospermic seed. Okay. So this is your morphology of seed. Seed coat. The outer covering of seed coat is called testa, and inner coat is called tegmen. While canal, it have three parts: perisperm, endosperm, embryo. Embryo having two parts: embryonal axis and cotyledon. Cotyledon will have two parts: plumule and radicle. Plumule give to that shoot, and radicle give convert into root. Okay, this is your monocot seed, and this is your dicot seed. Okay, so monocot seed versus dicot seed, that is a difference between that definition. Monocot seed is defined as seed that consists of single embryo leaf or cotyledon. Dicot defined as seed that consists of two embryonic leaves. Okay, number of cotyledon: monocotyledon single cotyledon, dicotyledon two cotyledon. Okay, the cotyledon in monocot seed are thin and small, while the cotyledon dicot is fleshy and stores food material. Then endosperm, the endosperm is present, which stores the large amount of food for embryo. It is in monocot seed, while in endosperm, that in dicot seed, endosperm is reduced and even absent. Okay, plumule in monocot occur terminally, in this occur laterally. Okay, coloprisa in monocot, coloprisa is present around radicle. In monocot seed, coloprisa is absent in dicot seed. Then coleptile is present in monocot, coleptile is absent in dicot. Shape and size, shape of size of monocot variable, while shape and size of dicot are variable, but these are usually more symmetrical and large in size. Okay. So next is your asexual reproduction in that plant. Means no sex is developing, but progeny is going ahead. Okay, then it is called apomixis. Apomixis is the asexual development of seed and embryo without fertilization. Apomixis will produce offspring generally identical to parental copy. These are the copies of their parents. Okay, except for non-recurrent apomixis. This term is derived from Greek word away from and mixing up. Okay, type of apomixis: diplospory, apospory. Diplospory, the embryo sac developed from the megaspore mother cell through direct mitotic division. This is called sac is diploid. Example: Parthenium, Tarchecum, Membranum of Asteraceae. Then apospory, formation of gametophyte directly from sporophyte. Means directly from sporophyte, gametophyte is developing without meiosis. Then it is called apospory. Okay, then adventitious embryony. In this method, embryo is formed from any diploid cell of nucellus or cell of integument. Then it is called adventitious embryony. Okay, application of apomixis: development of pure line you can get, maintaining of purity, heterosis conservation, easy hybrid seed production. Okay, next is your parthenogenesis: formation of embryo from unfertilized egg. Then it is called parthenogenesis. Then apogamy, development of sporophyte from gametophyte without the fusion of gamete. Then it is called apogamy. Then polyembryony, it is defined as occurrence of two or more embryo in one ovule. It was found in the seeds of orange by Leeuwenhoek. Polyembryony is of two types: true polyembryony, false one. True, the embryo are formed in the same embryo sac, then they are formed from embryo or endosperm and cell of the embryo sac except the egg. And false, the embryo arises from different embryo sac in the same ovule. These can develop from the cell of the nucellus or the integuments. Okay, importance of polyembryony: produce plant with great vigor, produce seedling of parental type, nuclear seedling produce roots that are better. And last we have parthenocarpy, that is formation of root from unfertilized ovary is known as parthenocarpy. It in some angiosperm, fruit is formed from the ovary without fertilization, which is known as parthenocarpic fruit. Okay, so we have some table related to the ploidy level. So zygote, okay, 2n, embryo 2n, radicle 2n, plumule 2n, cotyledon 2n, nucellus perisperm 2n, integument seed coat 2n, microspore mother cell mmc 2n, megaspore mother cell 2n, ovary wall, fruit wall 2n, carpel 2n, sepal petal 2n, stamen anther 2n, leaf, root, stem all are 2n. This is list for your diploid. Okay, now we have some haploid and tetraploid. So microspore, pollen grain N, tube cell, generative cell N, male gamete, female gamete N, megaspore N, embryo sac N, synergid N, antipodal N, egg cell N, polar nuclei N plus N. It is not 2N, it is N plus N. Then secondary nucleus 2N, primary endosperm nucleus 3N, endosperm 3N, and alluring layer 3N. Alluring layer is the outermost covering of endosperm. Okay, then we have some pre and post things. Okay, so pre ovary, post it will call fruit, ovule to convert into seed, ovary wall it will convert into pericarp, integument it will convert into seed coat, outer integument it will convert into testa, internal integument it will convert into tegmen, nucellus it will convert into degenerate in sometime present in the form of perisperm. Then synergid and antipodal it get degenerate. Hilum of ovule it will convert into hilum of seed. Funicle of ovule, stalk of the seed. Mycopyle of ovule it will convert into mycopyle of seed, and chalazar of ovule it will convert into chalazar of seed. Okay, then some relative terms. Stamen it is called microsporophyll, also carpel it is also called megasporophyll. Pollen sac it is called microsporangium. Ovule it is called integumented megasporangium. Then egg or ovum it is called female gamete. Then sperm or male gamete it is called microgamete. Then germinating pollen grain it is called male gametophyte, and embryo sac this is called female gametophyte. Okay, so it is the end of the chapter, that is sexual reproduction in flowering plant. Now in 15 minutes, after 15 minutes we are going to start your last unit of 12th class, that is ecology. Thank you. Okay, now it is the last chapter of 12th class, that is your organism and environment, the last unit of 12th class, ecology, which is organism and environment. Okay, so in this chapter we are going to study, we're going to discuss about ecological level of study and different ecological points of earth. Okay, so ecology, it is a study of interaction among organisms and the abiotic forces and biotic forces to that organism. For example, in this room I am present in simultaneously air, water, sunlight, temperature is also present. That is your abiotic forces, and some insects, microorganisms are also present, that is interacting with me. So this study of interaction between abiotic and biotic forces is called ecology. Okay, so the term ecology was coined by and described by E. Haeckel, father of ecology is Ratter, father of Indian ecology is Professor Ramdev Mishra, and the first of all term ecology was employed for study by plant as by Baum. Okay, so this is the ecological hierarchy. So organism, two population, two community, two ecosystem, then biome, then biosphere. As we going from organism to biosphere, the size increases and complexity increases. Okay, ecology is concerned with four levels: biological organization, that is organism, population, community, and biomes. Okay, physiological ecology at organism level, in this study of adaptation of an organism to environment in the term of survival and reproduction. Okay, so how that different biomes form. Okay, on earth, how that different biomes are formed. The reason of formation of different biomes is seasons, different seasons: winter season, summer season, autumn season, spring season. And it is when your earth revolve around the sun, this thing will happen. When the earth is towards the sun, at that time spring and summer take place, and the same part which is away to the sun, at that time winter and autumn take place. Okay, so that's how your biome formation take place on earth. Okay, so we have different type of major biomes: desert, grassland, arctic and alpine tundra, coniferous forest, temperate forest, tropical forest. In India we have tropical rainforest, deciduous forest, desert forest, and sea coasts. Okay, tropical rainforest, it is present in Meghalaya, that seven sister parts of India. Deciduous forest is present in Madhya Pradesh, Maharashtra, Jharkhand, Chhattisgarh, some part of Karnataka, some part of Gujarat, some part of UP, some part of Bihar. Okay, desert, that is your Thar desert in Rajasthan. Okay, and coastal sea coast, that is your western coast and eastern coast, okay, of India. Then we have niche. Okay, niche is a particular area in which the organism live happily and do R value increases. Its R value increases. That area is called niche. Okay, a place where organism live and range of conditions can tolerate. Diversity of resources it can utilize, have a distinct function role in that area. An organism live, eat, work in this conformable environment called niche. Niche is the area where organism increases its R value. R value is reproductive value. Okay, then we have major abiotic factors. First we have temperature. Okay, temperature is the major abiotic force which control the growth of an organism. Okay, in earth we have phases different temperature in different areas, and different organisms are live in that different temperature range. Okay, unique habitats such as thermal spring and deep sea hydrothermal vents have the average temperature more of 100 degrees Celsius. In that temperature some microbes live, just like your thermoacidophils. Okay, temperature play an important role in the geographical distribution of organism and also their physiological function influenced by it. Throughout, gradual global warming affect organism in both survival and distribution also. Okay, as we move from equator to poles, temperature decreases, and ecosystem, the greenery, the organism which is present in particular ecosystem from equator to poles decreases. Okay, so on the basis of temperature, we have two types of organisms. First is eurothermal and second one is clinothermals. Eurothermal, these can tolerate wide range of temperature and widely distributed on earth. Example, mammals like human beings, leopard, dog, monkey, cow. These can easily tolerate any type of temperature. Then we have stenothermal. These tolerate only a very range of temperature and are restricted to a particular distribution area. We have two types of stenothermals: thermophilic, who will like to live in high temperature ranges, and cryophilic organism that will live in cool temperatures. Okay, then we have second abiotic factor, that is water. Okay, so as you know, water is the main source from where that life exists, the life come on earth. Okay, so on the basis of salt toleration, we have two types of organisms: urohaline and stenohaline. Urohaline, these organisms which can adapt themselves to a wide range of salinity for survival and able to perform their due physiological functions in the same range of salinity. Okay, then stenothermal, these organisms which can tolerate narrow range of salinity for survival and also perform their due physiological function in this narrow range of salinities. Okay, then we have light. Okay, as you know, sun is the ultimate source of energy. The sunlight which is coming on earth, it is taken by your plants to do photosynthesis to produce food. Okay, but in deep oceans where sunlight cannot penetrate, in that areas the source of energy is hydrothermal vents and geothermal vents. The energy which is coming out from that sources is being used to do metabolic activities on the organism which finds there. Okay, then we have soil. Okay, the nature and property of soil is given place independent on the climate, weathering process, sedimentary soil and soil deposition. How that soil form, that is we call weathering. Breakdown of large rock to convert into small particles of soil, that process is called weathering, and it is very important for formation of soil. Okay, properties of soil like composition, soil particle size and their aggregation determine its water holding capacity. Which type of vegetation will give suitable for any area dependent on the physical and chemical property of the soil, such as particular size, porosity, pH and mineral composition. Okay, next we have some response to that adaptation, response to abiotic factors. First we have regulators. These organisms can maintain homeostasis by physiological and behavioral way. For example, mammals and birds. Means if outer temperature is hot, then their body become cool, and if the outer temperature is colder, then their body become hot. That type of organism which can control their body temperature according to the outside, then they are called regulators. Okay, then we have partial regulator. They can do that thing of regulation at a limit. After some limit, they will have to escape from that area. Then they are called partial regulator, just like lizards in desert, which can tolerate a little bit of temperature, but when the temperature exceeds that cannot regulate by their body, they will escape from that area. They will start burrowing inside the soil. Okay, then we have conformers. 99% of animal or nearly all plants cannot maintain their constant body temperature. If outside this hot, then their body temperature will increase. If outside is cold, then their body temperature goes down. So these type of organisms are called conformers, and 99% of animals and nearly all plants cannot maintain their body temperature. Okay, next we have migration. Okay, so some organisms start do escaping that unfavorable condition and go to some favorable areas to live. That process is called migration, and mostly migration is done by birds. There is some animals also which do migration, just like wild beasts in Africa. If the drought season will occur in some part of Africa, then that wild beasts do a major migration to reach out a fresh area where your high water supply is there and greenery is there. In the birds, we have two birds, Arctic tern and Siberian crane. They will known for their famous migration. When in the Siberia, in Russia, winter started, then these birds start to migrate towards India in the Bharatpur bird sanctuary. And in the term of height, your Siberian can have the longest migration in terms of height, and in terms of distance, Arctic tern have the longest distance range of migration. Okay, then we are suspend. Some organism cannot run away from that particular ecosystem. So they will stay there and stop the metabolical activities of their body, and they will known as suspends. Okay, in bacteria, in microorganism, they will start to produce endospore. Okay, in higher plants, their seed start to become dormant, and when the outer environment become favorable for germination, they come out from dormancy and do germination. Then we have animals. In animals, it's like deer. They will do hibernation. They go in winter sleep, that we call hibernation. In animals like fish and frogs, they will go in estivation, that is your summer sleep. Okay, and some protistants like diatoms, dinoflagellates, they will go in diapause stage, in which they stop all the metabolical activities, and when outer environment become favorable, they start to do their work. Okay, adaptations. Okay, so different organism develop different adaptations, just like the organism which is living in winter in cool area, in the very minus degree temperature areas like Antarctica and Arctic, they will develop different physiological and morphological character inside their body. That thing is called adaptation. So first we have morphological adaptation. Okay, in animals, temperature affect the absolute size of the animal and this its body size. So we have two rules. One is Bergman rule, another one is Allen rules. Okay, in Bergman rules we say that when the temperature become cooler, then the body size of organism increases. Okay, while in Allen rules we will say that the external body parts which is in contact with outer environment, they will increase their size in warm condition to expose out, to give out all the heat which is present inside the body. While the organism which is living in cold region, they have smaller body parts, outer parts, which make them to stop to release of that heat energy outside in the environment. We have a classical example of Allen rule, that is Arctic fox, normal red fox and phoenix fox. Arctic fox, this live in polar region. Its outer body parts like your ears and forelimbs, they are shorter. The shorter the outer body part, the less heat will come out from the body. So they preserve their heat by having that smaller outer part. While that red fox in the tropical region, they have normal size of ear and legs, so they do not require that much of heat loss or heat gain. Okay, while this phoenix fox which is present in desert, they have large body, that external body parts just like here and legs, to make that heat loss take place. If that heat loss not take place from their body, then there is chance of developing of heat stroke inside the body. So these all are morphological adaptations. Now we have some physiological adaptations. Okay, physiological adaptation means that organism will develop some physiology inside their body by which they can adapt to a particular regions. Okay, for example, in polar sea, aquatic mammals like seal having thick layer of fat blubber which make them feel safe and warm in cold climate. Okay, then we have kangaroo rat in your North American desert, which can withstand in the drought condition even by not drinking water in a week. Okay, how can you live without water? See, they will eat nut. Okay, that nut contained fat, and due to the oxidation of fat inside their body, they store water, and that water they utilize. Plus they will do a urination, that they will excrete out very concentrated urine to save the water. Okay, secondly, in some organism which is living in deep water, they will develop a special anti-freezing protein by which their body does not get freeze in deep seas where temperature can be minus 10 or less than minus 10 degrees Celsius. Okay, so these are our physiological adaptations. Now the second part of this chapter is population. Okay, so population, again, this is your ecological biological hierarchy in which you will see that proton, electron, neutrons comes together and they form atom. Many atoms comes together, they form molecule. Molecules comes together, they form biomolecule or macromolecule. These macromolecule aggregate together and forms organelles. Now these organelles will make a cell, and cell convert into tissue. Tissue together forms organs. Many organs comes together form organ system, and many organ system in an organism form an organism. Then organism, many organisms comes together of same species and forms a population. Then that all the living species in a particular area forms community, and functional unit of ecology, that is ecosystem in which many communities live. Okay, and many ecosystems comes together, they form biome, and many biomes comes together, they start to form biosphere, and their biosphere is your earth. Biomes means your stratosphere, lithosphere, hydrosphere. Okay, so now we have demography. Demography is the study of population. Okay, the scientific study of population is called demography. Population is defined as the total number of individual of species present in a particular area at a given time. Okay, again the definition: population is defined as a total number of individual present in a particular area in a particular time. Okay, so the population have specific character different from character of an individual. Okay, population have different characters, a single individual have different characters. That is a difference between a population and an organism. Okay, so we are going to see the characters of population. So first is your population density, which we call R value. Okay, then we have natality, then we have mortality, then we have population growth rate, and at last we have age sex distribution of a population. Okay, so first we have population density. Population density or population size, which is denoted by n. Okay, so population density is the particular population present in a per square kilometer area which is living, reproducing there, increasing their R value there. That is called population density. In terms of population, West Bengal is the highest populated state of India. Okay, so example, population density of Siberian crane in Bharatpur wetland is in year is more than 10. It's million for chlamydomonas in a pond. Okay, it is different. The value is different for different organism. As you know that flamingo is in Siberian crane is in multicellular large organism, while your chlamydomonas is a microscopic organism. So its number is in million. And while your Bharatpur National Park Siberian cranes are on fingers. Numbers are on fingers. Okay, next, total number is difficult. Sometimes what happen, total number of an organism is difficult to count, so we do relative population density. Means we, just like for example, we catch 10 fishes from a pond, so the population density of that pond is 10 fish. If we caught 1000 fishes from that pond, so the population density is 1000. So every time population density, relative population density changes. Okay, in some cases indirect estimate of population size is performed. Example, tiger census in national parks and tiger reserve based on the pug mark and fecal pellets. Okay, the number of pug mark you will search, you will see it will accumulate in data and will say the population density of particular national park and tiger reserve. Okay, next is your birth rate. Okay, second character of a population is birth rate or fertility or natality. Means number of birth per capita is called birth rate. Okay, to find the birth rate, we have a formula is B equals to total number of birth upon initial population. So birth rate varies from region to region. If the area is very good in food resources, having high shelter value, having high reproductive value, then birth rate is very high. If a particular area has high disease rate, means the diseases are spreading very fastly, then the birth rate is very low, death rate increases. Okay, then we have death rate, mortality. Okay, so the death is defined as the total number of death in a population with respect to total number of individual of a population in a year. The formula is D equals to total number of death upon initial population. Okay, so there we have one question for that. If 4 individuals in a laboratory population of 40 fruit fly died during the week, the death rate is calculated as. Now we will going to solve that. Okay, so the question is saying 4 individuals in a laboratory population of 40 fruit fly died during a week. Okay, so how many individuals are died? 4. So total number of death is equal to 4 upon the number of population, that total population of fruit fly, that is 40. Okay, so we get the death rate is equal to 0.4%. Okay, so there is death, that is the sign of death rate. Okay, the tag on a leg in which we will mark the death time, death day, and all other things. Okay, next we have growth rate. Okay, how the growth is taking place in a particular region. Okay, that is called growth rate. Okay, we have two factors for basic processes that controls growth rate. First one is natality, that is your birth rate. Then mortality, that is your death rate. Then immigration, number of individual of same species that have come into the habitat. They are come inside the area. It is called immigration. Then we have, it is a immigration, that is the number of individual population who left the habitat, means they go out from that habitat for a while. Okay, they are not coming back, they are lost, they are going outside from that particular habitat, that is called immigration. Okay, so for finding your growth rate, we have a formula, that is n t plus 1 equals to nt. Nt is the time at a given time, the population density, plus b plus I, that is birth rate, plus immigration minus death rate plus immigration. Okay, that is the formula to find the growth rate of a particular population. Okay, in this diagram you will see that this is plus sign, which is showing birth rate, and immigration, which is increasing the population density, and then you have death rate and immigration, which makes population density low. Okay, then we have age sex structures. Okay, it is a graphical representation to represent age of a particular population. What type of age people lives in that particular population, we will define that by the help of age sex studies. Okay, or age sex pyramids. Okay, so we have three types of age. First is pre-reproductive, which is less than 15 year. Then we have reproductive, that is 15 to 44 year, means adult. Then we have post-reproductive, that is more than 45 year, means old age people, senior citizens. Okay, so on the basis of this, we have three pyramids, three graphical representation of population. First one is your expanding, second one is stable, and third one is declining. So expanding is when your that higher number of pre-reproductive individual, moderate number of reproductive individual, and fewer post-reproductive individual will from young population, it shows rapid growth. This is your expanding. Okay, the example of expanding population is India, China, Pakistan, Sri Lanka, Nepal. These countries come under expanding population. Means when you draw the pyramid, age sex pyramid of these countries, you will find it is an expanding pyramid. Okay, then we have stable population. Okay, an equal number of pre-reproductive and reproductive individual with constant mature population or stable population. Okay, you will see this type of pyramid in developed countries like UK, Russia, USA, New Zealand, and Australia. Okay, then we have declining population. Okay, fewer number of pre-reproductive individual as compared to reproductive one will make population age, it should be negative growth. It is seen in Japan. Okay, in Japan there is less number of pre-reproductive and more number of post-reproductive people. Then it is called declining population, which we can easily see in Japan country. Okay, Japan is developed country, but still they facing population loss. Okay, because of some genetic disorders and some that environmental effect. Okay, next we have population growth models or curves. Okay, these curves represent growth of population, how it is taking. Okay, so on the basis of that thing, we have two curves. One is your exponential curve, and second one is logistic curve. Exponential curve is the curve which is stretches out during that environmental that all the environment having proper food, unlimited shelter, and unlimited reproduction. It will, that exponential growth you will see in the case of microorganism, because they can eat unlimited food, they can do unlimited reproduction, and they have unlimited shelter. Okay, exponential growth is not seen in real world. Okay, in multicellular organism you cannot see exponential growth, because one time they will die, their reproductive rate is not that much faster to do reproduction. Okay, so on the basis of that, we have a graph which is called exponential growth curve. Okay, it is a J-shaped curve in which you will see the population size increasing by time increase. Okay, when time going ahead, the population size increasing and producing a J-shaped curve, which is called exponential curve. Okay, or exponential growth. Okay, second type of growth is the logistic growth. This growth is having some logic, that's why it is a logical growth, logistic growth curve. It is also called sigmoid growth curve or S-shaped growth curve, Pearl Hostel growth curve. Okay, and the formula for this curve is dn by dt equals to rn k minus n upon k. Okay, so this is the formula for this curve, in which you will see that there is three phases. First one is lag phase, then lock phase, then stationary phase. Okay, see, lag phase, lock phase, stationary phase. In this phase, there is some hindrance, some you can say some problems are there to developing a particular population, but after facing that problems, after crossing that hard time, they show exponential phase, means they have rapid growth. Okay, and after some time when the food material get less, then the dispensity of food take place. At that time the graph become stationary. Okay, and this growth curve show the reality of life. Okay, first in the life you will show, you will feel, you will face some hardness. After some time when the hard time passes, you will get opportunities, you will get different good times in which you will grow rapidly. And at the time of a end of, means in your old age, you will see a stationary phase where you want to stop. Okay, it also show a growth of a human also. This lag phase represent the childhood, log phase represent your adulthood, and the stationary phase represent your old age. Okay, so this logistic growth is a real curve which is present in nature, while your exponential growth, okay, dn by dt, is not a real thing. It is an imaginary thing, it is an imaginary graph. Food is not always unlimited. Shelter is not always unlimited. Reproduction is not always unlimited. So that exponential growth is not a realistic growth, while your S growth or your logistic growth is a real growth in a particular organism life. Okay, so this is the diagram of your NCERT, in which we have both the type of growth on a scale. Then this is A1 is your exponential curve, in which the formula is equal to dn by dt equals to Rn, while your S curve or your sigmoid or your one Herschel curve, in which we have the formula dn by dt equals to Rn k minus n upon k. Okay, next we have life history variation. Okay, as we study that thing on evolutionary scale, we will see that different organisms produce different evolutionary skills, different evolutionary titles that they can live in the particular regions. Okay, so some organisms breed only once in their lifetime, they only require once time breeding. For example, your bamboo tree, okay, it will do flowering in span of 150 years, after will it will die. Okay, while other breed many times, just like most of the birds and mammals. Okay, some produces large number of small size of offspring because they have fear that their predators can easily eat them. They do not have security to save their offspring, so they produce large number of offspring कि कुछ तो बचेगा, कोई तो बचेगा जो हमारी प्रोजेनी को आगे चलाएगा. Okay, and some other while having produce a small number of large size offspring because they know we can do better parenting, we can save our offspring, so they will produce small amount of large size of progenies. For example, birds and mammals. Okay, all these things are produced due to the life history variation, the variation which is coming inside the organism during the evolutionary scale time. Okay, so these facts indicate that life history trait of organism have evolved due to the limited abiotic and biotic component of the habitat. When you get limited abiotic and biotic things, means that limited temperature, limited air, limited water, limited soil, limited sunlight, and biotic limited food, limited reproduction, limited shelter, then you will show this type of things inside your life. You will show this type of offspring limits inside your life. Okay, so we have some examples here. This is your bamboo tree which reproduce in 150 year of time. This is your salmon fish, it is again reproduce once in the time, then it will die. Okay, then you have brancals, she cells, we reproduce at a very high rate because they have afraid that they can easily eaten, they can easily killed by their predators. Okay, so now next part of this chapter is population interaction. Okay, population interaction means when two different population interacting with each other and showing what type of interaction they have. Either it is positive, either it is positive negative, or either it is positive zero. Then we studied that thing in population interaction. Okay, so population interaction is an interaction between two biological communities. Okay, so on the basis of we have different type of interaction. First we have mutualism, in which both the species are having good side, means both are dependent on each other, one cannot live without other. Okay, second one is competition minus minus, means there is loss of both species taking place. Then predation, in which one species is a positive side and one species is on negative side. Then parasitism, again one is on positive side and again is in negative side. Then commensalism, one is on positive side and other does not have any positive thing and does not having a good negative thing also. Okay, amensalism, one is having negative side and other having neither negative or either positive side. Okay, so first we have mutualism. Okay, so it is an interspecific intersection in which both the interacting species are benefited. Means in this relationship both species are benefited. There is no red flag. Okay, so the following are the examples in this chapter. You have to remember only examples. Okay, so first is lichen. Okay, it is a mutual relationship between algae and fungi, in which algae is your that phycobiont and the fungi is mycobiont. Algae is preparing food for fungi while fungi giving shelter to algae. Then we have mycorrhizal association, okay, in which a glomalous genus fungus is living inside the root of higher trees, in which the glomalous fungus providing phosphorus, nitrogen like nutrients and making the plant safe from pathogenic attacks, while on the place of that plant giving them nutrition and shelter. Okay, both species are interconnected with each other. If one species is deleted, then there is adverse effect will happen on other species definitely. Next we have fig tree and wasp. Okay, so I have a fig fruit in which I can show you how there is a mutualistic relationship taking place between this fruit and that wasp. Okay, so you can see here, this is the how fig wasp works. Okay, so this is your fig fruit and inflorescence. Okay, so in this from this area female wasp get entered. Okay, and it lay egg inside that. Okay, so when that female wasp flies from one fig flower to another fig flower, it will do pollination, and simultaneously when that egg, okay, that egg hatches, the larva come out, it will come out from that area. Okay, because it is the entrance and it is the exit. So when it is come out, then it will go another fig, then it will do pollination. Okay, that's how both the species is interconnected with each other. Fig plant is dependent on wasp for pollination, for making its generation go ahead, while your wasp is dependent on this fruit for nutrition and making its egg a suitable environment. Okay, next example is sexual deceit by Mediterranean orchid. Okay, this is a classical example of mutualism again, in which one species gets its sexual desire done and other species get pollination. Okay, so this is your Mediterranean orchid plant. Okay, and this is a Mediterranean wasp. Okay, so it is male wasp. Okay, so what happened? This flower look like female wasp. Okay, so this insect does not distinguish between a female insect and this flower. He thinks it is a female insect, so it will sit on it and start doing copulation, but it is not a successful copulation because it is a pseudo copulation. Now when he knows that it is not a female wasp, it will go and another plant, okay, and start doing the same thing. Okay, so both the species is benefited. Flower, it get pollinated. Wasp, its sexual desire has been fulfilled. Okay, either it is a pseudo, a false desire, but it is going to be filled. Okay, so mutualism, it is a relationship in which both the species get benefited. Okay, we can have both more examples just like honeybee and your flower, lichen, mycorrhizal root association, your butterfly and flower, in which that butterfly eating nectar from your flower and the pollination of flower is done by your butterfly. Then we have most classical example of mutualism in which one species extincted, then other one that definitely get extinct. That is yucca plant and proboscis moth. This is proboscis moth and this is yucca plant. This plant having only one pollinating agent, that is your proboscis moth. If this thing will get extincted, then there is no pollination of this plant will take place and their progeny does not go in next generation. Okay, and if this plant will die, this plant will get extincted, then proboscis moth does not have a place, does not have a shelter to live, because it is the only shelter for proboscis moth. Okay, so next relationship is your commensalism. Okay, commensalism means one is in positive and other one does not have any bad thing and does not have any good thing. Okay, so commensalism is defined as the interspecific interaction where one species is benefited while other species is neither benefited nor harmed. Okay, not harmed, not benefited, both things is not happening to zero species, while positive species getting benefit. Okay, example we have orchid. Okay, this is orchid growing on a mango tree branch. Okay, this relationship is called epiphyte, in which this plant is growing over the stem of mango, getting sunlight properly, and in this mango does not getting any harm and simultaneously does not getting any profit. Okay, then it is an example of commensalism. Next we have wild buffalo and that agret birds. Okay, this thing when that buffalo move in the wild, it will make holes in the soil, and when it makes the hole, the insect which is living in the soil, it will come out. So they will in the they will in the eye of that insect and they will eat that insect. They will getting the food in the result of that, in the form of that buffalo not getting any profit and not getting any loss. Okay, so it is an also example of commensalism. Next example is your brancals and whale. Okay, these are the blankers, these are the mollusks. Okay, they get attached to the whale skin and they will move towards the different ecosystem, because when whale do migration, it will move different areas of oceans, so they will get free transportation. They can easily save from their predators, because the predators of this organism does not come near to the whale. Okay, and on the other hand, the whale does not get any harm and does not get any profit from these brancals. Okay, so this is their commensalism example. Fourth example of commensalism is your that clownfish and sea anemone. You see this organism in the movie Finding Nemo, and you see in that first part that this is the house of Nemo. Okay, that is sea anemone. Okay, so this fish lives inside this house. Okay, so the fish get a house, but this anemone does not get any advancement, any profit, any loss. It is only living there. Okay, next is your proto-cooperation. It is a type of mutualism, but the difference between proto-cooperation and mutualism is that in mutualism, both species are dependent on each other totally. If the absence of one species take place, then other species will definitely having adverse effect. But in proto-cooperation, if the species which is connected to each other, if they remove from each other, that there is no any effect will coming on other species or both species. Okay, for example, we can give mutualism is a relationship between husband and wife. Okay, and proto cooperation is a relationship between boyfriend and girlfriend. Okay, if they get detached from each other, there is no loss taking place between the both. Okay, while if the relationship between our husband and wife get affected, then there is major different social acts will take place, different fighting take place between them if they having that bad relationship between them. Okay, same example is here, your proto cooperation. We have different example. First is your hermit crab and sea anemone. Okay, so this sea anemone sits on the back of the hermit crab and room inside the ocean bed. Okay, so in this, that sea anemone getting free transportation system, while your hermit crab have a very dangerous appearance. When this thing will sit on that, its predators escape away from him. Okay, but if this thing get detached from the hermit crab, it does not having any defect, it does not cause any damage to both species. It can live independently, it can live independently, they are not totally dependent on each other. Second example is your crocodile and flesh eating birds, small flesh eating birds. Okay, so this bird do what? It eat the meat which is accumulated between the teeth of the crocodile. Okay, so it getting food and crocodile getting free dental care. Okay, but if this thing will not come and eat the food, then that there is no effect on the teeth of the crocodile take place. So it is again the example of proto cooperation. Third example of proto cooperation is your rhino and yellow red bird, big bird. Okay, in which this bird eats the ticks and mites which is present on side that rhino, and that rhino get a body cleansing system. Okay, means the body get freely cleansed, the ticks mites get easily cleaned by this bird, and the bird getting food. Okay, if we remove one species from that, it will not get any effect on other species. Okay, if we remove that rhino, then this bird can eat seed also. Okay, and if we remove this bird, then that rhino can live with that we can say that with the mites or that the tricks on the body. Okay, it does not cause any major damage to rhino. Okay, next one is your competition minus minus relationship in which both species get bad effect. Okay, it is the process in which fitness of one species are valued significantly lower in the presence of other species. Interspecific competition is a potent force of organic evolution. If there is a competition, we get one winner and one loser. But in biological world, that winner after some time can lose the fight also, because the competitor is lost. Due to that loss of the competitor, he does not have the wish of competition, it will lose the competition. Okay, so both the species in a negative side. So competition occur in abundant resources also, and it will occur in low resources also. Okay. For example, the interference competition, the feeding efficiency of one species reduced due to the presence of other species, even if the resource is abundant. For example, the Galapagos tortoise and goat. Okay, goat having very high grazing efficiency, while Galapagos tortoise have very low grazing efficiency. Okay, food is unlimited. There is unlimited food on that Galapagos island, but the grazing efficiency of goat is very high, and grazing efficiency of your tortoise is very less. So after that species of tortoise is extincted from that Galapagos island because of that overgrazing by goat. Okay, this is the classical example of that thing. This is your Galapagos tortoise, and this is your Galapagos. Okay, so a Bengal tortoise in the Galapagos island become extinct within a decade after goat were introduced on the island due to the greater browsing efficiency of goat. It will eat. It is an herbivorous tortoise. हाँ क्या हुआ? So next example of competition is resource partitioning. Okay, how can you avoid competition? We are going to see that. Okay, so Gauss competitive exclusive principle and resource partitioning. Okay, it states that two closely related species competing for the same resource cannot coexist independently, and the competitive inferior one will be eliminated eventually. Okay, this may be true in limited resources but not otherwise. Means if food resource is limited, so the one who having higher grazing efficiency, higher eating ability, it will survive, while the low eating ability one will always extend from that particular reason. Okay, species facing competition may evolve mechanism for coexistence rather than exclusion. Example, resource partnering. Now uh, some high IQ species, or some that we can say some quick mind species will develop some measure some different uh, processes, okay, to do and partitioning their resources. Okay, that is resource partnering example, in which you will see there is species of birds. Okay, they will occupy different area to eat. Okay, just like this species only eat the top part of the tree, this species will part eat with the middle part, this species will only eat the top central part, this will eat middle central part, and this species will eat only lower part. This species is not going to eat this top part. Okay, this phenomena is called resource partnering. Okay, in which particular species partner their resources, or they will change their feeding time. For example, this bird will come and eat in the morning time, early morning time. This bird will come and eat that particular resource inside the late morning time. This will come in afternoon. This will come in that early evening. This will come at late evening. So they will divide their eating times or their eating areas to do not go inside a competition. Okay, this thing is called resource partitioning. Okay, next we have competitive release. Okay, this is again an experiment done by Cornell's. Okay, and we call it Cornell field experiment. Okay, on the rocky sea coast of Scotland there is two branchial species, Balanus larger and the Cathameles smaller. Balanus is your Gunda and Cathameles is a common man. Okay, so Balanus dominate intertidal area and exclude Cathameles. When Cornell's experimentally removed Balanus, Cathameles colonized the intertidal zone. Means if you remove the superior species from a particular area, you will find that the inferior species will increase their number there because there is no competition taking place. Okay, so this is called competitive release. You are releasing the competition, you are removing the competition. Okay, and its classical example is your Brankel's sea coast, is a Scotland sea tidal intertidal region experiment. Okay, then we have parasitism. Okay, in which one organism is of positive side and other organism is on negative side. The positive side one is called parasite and negative side one is called host. Okay, so first we have to see what are the character a parasite have. Okay, what are different features a parasite can be having to become a parasite. First thing, it should have a cuticle lining, thick cuticle lining to make itself safe from the enzymes and acids of host body. Second thing, it do not have digestive tract, because if the digestive tract present, then it will do not work that much. Okay, why that parasite want digestive tract? If it is directly taking nutrition from the host, then it do not require digestive tract. Third thing, it should have hooks and suckers to take and to attach to that particular host. And the fourth thing is that the reproductive value of that organism is very high. Then that organism is called a parasite. Okay, so we have some classical example of parasites. Okay, so we have two types of parasite, ectoparasite and endoparasite. Ectoparasite, which is present outside the body of the host, and endoparasite, which live inside the body of the host. Okay, so ectoparasite examples are हेड लाइस ऑफ ह्यूमन. ओके, जो बालों में जू होती हैं वो. ओके, then ticks on dog skin. Okay, these are the ticks which is present on the dog skin. Okay, then marine copepods on fish. Okay, these are the marine copepods which is present near to the gills of fish and suck all the dissolved oxygens. Okay, and cascuta on hedge plant. This is called cascuta. This yellow one is cascuta. Okay, it is in Hindi it is called amar bale. Okay, it never dies and it is the major cause of many ecosystem destruction because it will suck all the plants nutrition, and when that plant dies after some time it will also die because it is not getting nutrition because its host is dead. Okay, then we have endoparasites. They take shelter within the body of the host, means they live inside the host body. Okay, then we have example just like liver fluke, it lives in the liver. Okay, then we have the tapeworm. Okay, it lives inside the small intestine of human. Okay, these have different host also, just like blood fluke, it is having the that liver fluke, it having host like snail, fish and human, it having pork and human. Okay, then we have broad parasitism. Okay, it is a phenomena in which one bird species lays its egg in the nest of another bird species and foreign egg are hatched as well as chicks are fed by the host bird. Okay, the classical example of broad parasitism is cuckoo bird and crow. Okay, crow makes nest. Okay, and cuckoo bird do not want to make a nest, so it will go and lay egg inside the nest of crow, and the egg resembles same to each other. Okay, see, this is all are the eggs of crow and this is the egg of cuckoo bird. Okay, the color, the appearance is same, sometimes the shape changes but the color is same, and that mother crow bird does not distinguish between its own egg and its that foreign egg, and it start doing that incubating that eggs, and give when that egg get hatched, it will start to feed them. Okay, this is again an example in which the shape is same but color is different. Still, the crow does not distinguish between either it is my egg or egg of other bird. Okay. Next that relationship is your predation. Okay. Why is it blue? This shell color is blue because of the deposition of copper and other substances. Okay, that is blue. Every egg of different birds having different color. Just like hen egg, it is a white color. Some hens can produce black color egg also. Okay, so different eggs have different colors. Okay, and it is on the embryonic development. During the embryonic development of that bird, what type of pigment is present inside that egg, it will come in the form of that eggshell, calcium composition. Okay, then predation. Okay, it is an interspecific interaction where one animal kill and other animal eat that animal. Okay, that one who kill, we call them predator, and the one who is being killed is called prey. Okay, in this image you can see there, this is the tiger which is in predator, this is the deer which is prey. Then again this is the snake, it is in predator, and the frog is the prey. Okay, in this you can see the finch is there and it is eating an insect. The insect is the prey and the finch is an predator. Okay, predation is an important part of food chain. Okay, so next is predator play an important following rules in an ecosystem. Okay, predator always control the number of prey. If prey number does not controlled by predators, then it will definitely destroy the particular ecosystem. Predator is a very important part of an ecosystem management. Okay, predator is very clever in their mindset because they know how to control the population of prey in a particular environment. Okay, so they act as a channel for energy transfer to highest trophic levels. Second, they keep the population of herbivore under the control, which otherwise can reach very high population density and disturb the balance of the ecosystem. That is the main role of predator in an ecosystem, that it control, it manage the counts of herbivores, their prey in a particular ecosystem. If they do not control that thing, then the ecosystem will be destroyed one day. Okay, then species diversity in a community is maintained by reducing the intensity of competition among the competing prey species. Also, they control the competition between particular two species by decreasing or increasing their number in a particular area in a particular ecosystem. Okay, next the prickly pear cactus of example is the prickly pear cactus introduced into Australia in early 1920 caused epoch in spreading rapidly into the millions of hectares of drylands. Okay, this is a prickly pear cactus, and this is the predator of this cactus, and it eats this cactus. Okay, but when this prickly pear cactus introduced in that particular area, it does not have its natural predator, so it will rapidly grow in that area. But when after that ecologists, the biologists living there, they know that there is one moth who is the predator of this cactus. It will introduce them and it will eat them. It will make control the growth of prickly pear cactus in Australia. Okay. Next we have organism of prey species have also evolved various defense mechanisms to save them. Okay. Now prey also develops different mechanisms to make safe from predators. Okay, which we call camouflages mimicry. Okay, they will hide inside the environment. They will make them to be hidden inside the environment. They make them look like the environment to hide in that environment. That thing is camouflage or mimicry. They will act like other top predators. They will make design of their body to other top predators to make the predators make them away from their action. Okay, so we have camouflage, cryptic coloration observed in certain insect species and frogs to avoid detection by their predators. In this image, you will see Vetinus mossy frog. Okay, it will develop a body design just like a moss plant. And when it will do camouflage, you cannot differentiate either it is a frog or a moss plant. Okay, then we have leaf mantis. Okay, this mantis develop body design just like leaf, it having the venation which look like leaf. Okay, one more example I have, that is stick insect. Okay, it look like a dry stick, it sits on dry stick of a tree and it look like dry string, that is your stick plant. All these are the example of camouflage. Some animals are highly dispersable to predators, so they save from predation. The monarch butterfly species accumulates a chemical by feeding on poisonous weed calotropis during the caterpillar stage. Okay, so we have a calotropis plant. This is your calotropis plant, mother. Okay, the leaves of this plant is very poisonous. It contains a milky secretion. See, See, this is a milky secretion which is coming out from your calotropis plant. It is very poisonous in nature. It is an glycosidic. Okay, it causes heart attack if we drink this thing in very high concentration. So the monarch butterfly caterpillar feed on this thing. It will not die. It drink this thing. It eat this thing. Now when the predator will come and eat your that monarch butterfly, it feels very distasteful. It does not look like the taste that bird need. Okay, so these are your images of camouflage, and this is your monarch butterfly caterpillar, and this is the that calotropis plant. Okay, next is plant cannot hide. Now we are talking about plant. Okay, plant cannot hide them from their predators, means herbivores. They cannot run from their predators. As you know, deer can run from tiger, but that plants, trees, they cannot run from herbivores. So in their body they develop different defense mechanism, just like thorns, okay, spines, to make the tongue of that herbivore pinched. Okay, this bougainvillea, okay, all these cactus, they develop acacia, cactus, bougainvillea, they develop spines in their stems. Now no herbivore come and eat them. Okay, some plants like calotropis produce highly poisonous glycoside, that is cardiac poison. Nicotine, statamine, optimum, quinine, etc. are the chemical produced by plants of their defense against herbivores. Okay, again this is the example. This is the glycoside which is produced by this plant, and this plant is not eaten by herbivores because it causes heart attack. Okay, to a particular species of animal. Then we have one more example of predation, that is star pisaster is generally found in the rocky intertidal area. It is an example of predation and it feeds on various species of invertebrates. But in a particular experiment, we remove this starfish from that area, and after some days, we see that 10 species of invertebrate get extincted from there, because this pisaster starfish controlling the habitat there, and we remove this top predator from there, due to which 10 species has been extincted because of that over exploitation of resources. They overexploit the resources ended, now they do not have the resource, and then they will be extincted. Okay, so predators are very important for a particular ecosystem to control the number of prey, to control the number of their food. Okay, if they increase in number, they can cause havoc, they will eat all the plants, they will destroy that ecosystem. So predators have very important role in controlling particular ecosystem. Okay, next we have amensalism. Okay, in this relationship, one is in minus, another one neither in profit neither in loss. Okay, the classical example of amensalism is algal bloom, in which your algae is on zero side, and the food at that plant which is living in that water body, the animals which is living in that water body, are on negative side. See, algae do not have any thing to that plants or animals. They do not want to eat that plants and animals, but accidentally it will grow that much inside that water, due to which sunlight cannot go inside the water. Okay, now sunlight do not go inside the water, so the water, the plants, the hydrophyte which is present inside the water, they do not get synthesize the food. Okay, and they do not do photosynthesis, though they will start to die. Okay, and now the organism which is on that aquatic plant, they will do not get food, they will also start to die. Secondly, this algae eat all the oxygen. Secondly, this algae eat all the oxygen which is present inside the water, due to which the water become poisonous, and it also release some toxins inside the water which will also cause the toxicity of the water. Okay, so in which algae is on zero side and your aquatic habitat, aquatic plants, aquatic animals on negative side. This phenomena is called algal bloom. Okay, algal bloom, in which algae grow extensively in a water body, making the BOD decrease. Okay, sorry, making the BOD increase, due to which the water habitat start to die. Okay, so this is your first chapter of ecology, that is population and organism. Now we are going towards the second chapter of ecology, that is ecosystem. Right? Right? One. Now second chapter of ecology unit, that is your ecosystem. Okay, now we will see different ecosystem which exist on earth and on India, because we are living in India and NCET is an Indian book, so we have to study that Indian ecosystem also. Okay, so ecosystem two types: terrestrial ecosystem, aquatic ecosystem. Terrestrial ecosystem then again divided into three parts: forest ecosystem, grassland ecosystem, desert ecosystem. Then your aquatic ecosystem, it is of two types. One is your freshwater ecosystem, in which river, pond, lake come. Okay, and saltwater ecosystem in your sea, oceans, and that lagoons come. Okay, ocean can be of different size also, or that ecosystem can be also of different size also. Thus, like mega ecosystem like oceans and seas. Then macro ecosystem like forest, smaller than oceans. Okay, then micro ecosystem like pond, smaller than your forest. Then nano ecosystem, a raindrop. Okay, and you know that our mouth, your body, is an also ecosystem in which many bacteria lives. Okay, so ecosystem components. Now ecosystem having two components: one is your biotic component, in which we have producer, consumer, decomposer. Producer, plants who produce the food; consumer, who consume the producers. It is of three types: herbivores, primary carnivores, and secondary carnivores. Herbivores are also called primary consumer. Your primary carnivores are called secondary consumer, and secondary carnivores are called tertiary consumers. Okay, and last we have decomposer, who clean all the mess which is done by these two. Okay, then we have abiotic forces, which is air, water, light, sunlight, temperature, soil. Solar radiation equals to sunlight. Okay, temperature and rain. These all are your abiotic components. Okay, so ecosystem, it is the structure and functional unit of a ecological hierarchy or a particular ecology. Okay, so the interaction of biotic and abiotic component result in physical structure characteristic of each type of ecosystem. So a two important structural feature of an ecosystem is species composition. What type of species is living, how many type of species is living in a particular ecosystem, that is the first component. And second one is your stratification, the level of species which is living in a particular area, just like canopy of trees in an Amazon rainforest. Okay, you will see higher species, long trees, which is making the shade of the Amazon rainforest. Then we have canopies, then we have shrubs, then we have shrub herbs, then we have land dwellers. Okay, that is we call stratification. Okay, this is the thing. Okay, stratification: canopy, sub canopy, then shrub layer, then ground layer, then understory layer. Okay, these all are stratification in forest. Okay, same thing we will see in deep ponds also. We have three regions in pond: one is littoral zone, one is sublittoral zone, and other one is profundal zone. Littoral zone is that zone in which sunlight can easily come penetrate the water. Then sublittoral zone means 50 to 40% light is only is coming, and then we have profundal zone, no light is coming. The energy source in that area is the geothermal vents or that hydrothermal vents, the energy of your earth, the core of your earth. Okay, then we have major functionals of an ecosystem. The four major function of an ecosystem comprises productivity, decomposition, energy flow, and nutrient cycle. These four things runs your ecosystem. If one thing will get neglected, deleted, then your ecosystem get collapsed. Okay. In your NCERT, productivity is there, decomposition is there, energy flow is there, nutrient cycle is now deleted. So we are not going to study nutrient cycle because it is deleted. We are going to study productivity, decomposition and energy flow only. Okay. So first we have productivity. Okay. Productivity means the producing capacity of an ecosystem of the material. Okay. The biomass which is produced by a particular organism at a particular level is called productivity. Okay. So the rate of biomass production is called productivity, is expressed in gram per year or kilocalorie per meter square. Okay. So amount of biomass produced per unit area over a time period by plant during photosynthesis is called primary productivity. Then it is divided into two: gross primary productivity and net primary productivity. Gross primary productivity, it is the rate of production of organic matter during photosynthesis. It is also called total photosynthesis. C. We have primary productivity. Primary productivity means the productivity done by your plants. Then primary productivity is of two types: gross primary productivity and net primary productivity. Okay. So gross primary productivity is the productivity which is done by doing photosynthesis. All the food which is prepared by your plant by photosynthesis is called gross primary productivity. Now we have net primary productivity. Net primary productivity is gross primary productivity minus respiratory lost. Okay. By doing metabolic functions of your body, the thing you are doing metabolically, the respiration, the energy losses which is taking place inside your body, when you minus that thing from gross primary productivity, you get net primary productivity. Okay. And you can see here I written that net primary productivity equals to gross primary productivity minus respiratory loss, or you can say that gross primary productivity equals to net primary productivity plus respiratory loss. Okay. So primary productivity varies in different ecosystem because it depends on the plant species inhabiting that area. Okay. If a plant has just like C3, C4, you know, in 11th class you studied about C3 and C4 plants. C3 plants are less productive while your C4 plant is highly productive. Okay. They can even do photosynthesis in low CO2 concentration. Then but C3 plant required a minimum CO2 for doing photosynthesis. Okay. So different type of plant species inhabiting a particular area define the productivity of that area. Okay. Then environmental factors means temperature, sunlight, water, air, all these abiotic forces, this also influence productivity of a particular area. Then availability of nutrients means your soil, what type of nutrient that soil contain. It is high in nitrogen, it is high in phosphorus, it is high in iron, magnesium. What type of nutrients that soil contain, it also defines the primary productivity of that particular ecosystem. Then photosynthetic capacity of plant, again I am saying C3 and C4 plants. C4 plants do higher productivity, having high efficiency of photosynthesis, while your C3 plant having low efficiency to do photosynthesis. Okay. So these all are the primary the productivities variations that depends on a particular ecosystem. Okay. Next we have secondary productivity. It is the productivity which is done by consumers. Okay. Means I eat food, now the food which is going inside my body, it is my secondary productivity. Okay. So it is the rate of formation of new organic matter by consumer. I am consumer, the plant I am eating, it is producer. Okay. Plant is producer, I am consumer. So I am eating that plant and the energy which is producing by my body is called secondary productivity. The biomass which is producing by my body by eating that plant, it is secondary productivity. Okay. So the rate of storage of organic matter not used by heterotrophs is your net consumer productivity. Net consumer productivity is equal to net primary productivity minus that respiratory loss. Okay. Or heat loss. Okay. That energy used by heterotrophs. Means I am walking here. So at that time I am walking, my muscles are moving. Okay. So my muscles require energy. So that energy coming where from where? When I eat the food, I get the energy. So it is utilizing by muscles and it will going to move. So that thing is your NCP, net consumer productivity. Okay. So annual net primary productivity of whole biosphere is 170 billion ton. Okay, whole earth, the net primary productivity of your whole earth is 170 billion ton. Okay, on of this 70% of surface, the productivity of ocean is only 55 billion ton. Okay, you know that earth having two sides. One is watery side, where your ocean, lakes, rivers, seas is present, that occupies 70% area of your earth. Other one is land, which occupies only 30% area. Okay, its area is more, this area is less, but this 70% only produce 55 billion ton food for our earth. Okay, the remaining one out of this 170, you can do this calculation here. 175 the remaining 115 billion ton food, it is produced by your 30% land. Okay, see the power of land, because the plant which is present on land, they have higher power to do photosynthesis. So your land, which is 30% only of your total earth, having 115 billion ton product primary productivity, while the water which is present 70% on your earth, it having only 55 billion ton of productivity. Okay, the reason is because of the plants, the difference between the plants which is present in the ocean, the hydrophytes and the land plants. Okay, so this is the diagrammatic relation, the diagonal representation of primary productivity and secondary productivity. Gross primary productivity, the energy produced by your leaves, and it is net primary productivity, which is used to produce fruit by your plant, and the 100 joule which is lost by in the form of heat, in the form of respiratory loss. Now that 200 joule which is produced by your plant, it is eaten by insect. Okay, now in insect, that thing is net secondary productivity. Okay, and in this net secondary productivity, you will see 100 joule will go in production of fecus material, 33 joule of energy is being stored inside the tissue, and the remaining 67 joule of energy is lost in respiration process. Okay, that's how we manage the productivity on earth. Next we have decomposition. Okay, second factor which influence ecosystem, which control your ecosystems. Okay, so we have decomposition. Okay, so it is the breakdown of complex organic matter by decomposer into a smaller organic matters. Means your macromolecules are converted into micromolecules. Okay, it is largely an oxygenic required process. It is an aerobic process. It is not anaerobic, it is aerobic process. Okay, raw material for decomposition is called detritus. These three images are of detritus. Okay, leaves, bark, flower, these all are dead plant remains. It is an detritus. Then dead remains of animals, this is detritus. Then fecal material of animals, this is detritus. Okay, next steps in decomposition process. So first we have fragmentation, then leaching, then catabolism, then humidification, then mineralization. Okay, these are the 5 steps of decomposition. So fragmentation. Large particles of detritus break down into small particles by detrivodes like earthworm. Earthworm, as you know, it is a farmer friend. It will break down the large particles in small form. Okay. Then we have leaching. Now the soluble inorganic matter which is present inside your detritus is being leached out inside the soil and will preserve inside the soil. Then we have catabolism. Okay, it is the fungal and bacterial enzyme converting complex organic compound into simple compounds. Okay, means your organic compounds which is very complex in the size, it will be converted into simple by the help of enzymes of bacteria and fungus, that is your catabolism. Okay, then we have humidification. Okay, in which dark colored amorphous substance humus is formed. Okay, and at last we have mineralization, in which release of mineral by degradation of humus microbes help in this process. Means your humus get degraded and it will convert its mineral, it will send its mineral inside the soil. This process is called mineralization. Okay, this is the diagrammatic representation of decomposition in your NCERT of terrestrial ecosystem. Okay, next factor influencing decomposition. Okay, so we have first chemical composition of detritus. How your detritus look? What is your detritus is made up of? So decomposition is slow in detritus rich in lignin and chitin, because on these substances like lignin and chitin, degradation process goes slowly. Okay, because of the composition, the component which is present inside your lignin and chitin, it will slow down the process of lignin form, that slow down the process of decomposition. Next, it is quicker if the detritus is rich in nitrogen and water soluble substances like sugar. Okay, sugar, it is very a simple matter, simple molecule which can easily degraded by your microbes. Okay, so if the detritus made up of nitrogenous compound or sugar, then it will easily decompose. But if it is made up of chitin or lignin, water resistant substances, then it will slowly degrade. Okay, then climatic factor, temperature and soil moisture. Okay, warm and moist environment favor decomposition. Okay, if the condition in the atmosphere is warm and it is moist, then the decomposition process is very fast. Okay, and if the temperature is low and an aerobics inhibit decomposition, resulting in build up of organic matter. Okay, means if the temperature is anaerobic and if the temperature is very low, then the process of decomposition becomes slow, because as you know, in the starting of decomposition we say that it is a highly oxygen required process, and anaerobic condition is that condition in which you are not providing proper oxygen to the environment. Okay, so these are the factors which influence the decomposition. Then energy flow. Okay, third component of your ecosystem. Okay, so sun is the only source of energy. It is the ultimate source of energy on earth, other than deep sea hydrothermal vents and your sulfur vents which is present in the deep ocean, because at that reason sunlight does not reaches, so the energy source is deep thermal vents. Okay, on the incident of solar radiation, less than 50% is photosynthetic active, which we call PAR. Okay, 100% sunlight which is coming from the sun, out of that 100%, only 50% is PAR, photosynthetic active radiation, which can be used in photosynthesis. Okay, plant and photosynthetic and chemosynthetic bacteria fix solar radiation energy to make food. Plant capture only 2 to 10% of PAR. Now, from this 50%, only 2 to 10% is used by plants. See, 100% sun for other 50 rupees पता नहीं कहाँ जा रहे हैं. Okay, now from that 50 rupees you are utilizing only 2 to 10% on you, that is the plants. Okay, so 100% energy is coming from your sun. From that 100% energy, 50% is your photosynthetic active radiation. Now from that 50% PAR, you only plants only using 2 to 10% for photosynthesis. Okay, that is again the important point. So this energy sustains the entire living world. So this 2 to 10% energy, this 2 to 10% energy running the world. Okay, so ecosystem obeys second law of thermodynamics. They need to constant supply of energy to synthesize molecules. It helps in the contractile the entropy. Okay, in physics you studied second law of thermodynamics, which is also applied here also. Okay, then the flow of energy from the sun to produce and then consume is a directional and keeping with the first law of thermodynamics. Energy neither be created nor be destroyed. It will transfer from one form to another form. Okay, so energy flow, we have two things. First is your producers. Okay, they are the organism that synthesize food, plants, basically plants. Then consumer, heterotroph, these are the animals that directly or indirectly dependent on plant for food. They include primary consumer, herbivores, feed on plant, example insect, birds, mammals and mollusks. Then we have secondary consumer, which we call primary carnivore, feed on herbivores, example frog, fox, man, etc. Then we have tertiary consumer, which we call secondary carnivore feed on primary carnivore, example tiger, lion. Okay, so this is your producer plant. Okay, this is your lion, that is your tiger, that is your tertiary consumer or secondary carnivore. Then we have secondary consumer, primary carnivore feed on herbivore, example frog, fox, man. Okay, then we have primary consumer herbivore feed on plant, example insect, birds, mammals, mollusks. Okay, these all are having food which we call plants. Okay, then these things are eaten by your primary carnivore or secondary consumer, that is your human, fox. Okay, then these are thing is eaten by a tertiary consumer or secondary carnivore feed on primary carnivore, example frog, that is your tiger, lion. Okay, tiger, lion, tertiary consumer, they are always top predators. Okay, next we have food chain. Okay, it is a component of energy flow in which food in the form of energy transferring from one trophic level to another trophic level, means it is a chain in which one organism is eating another organism. Okay, so it is a series of group of organism dependent on other for food. This is a relationship constituent of chain, hence it is called food chain. There is two food chain which runs on the land as grazing food chain and detritus food chain. Grazing food chain means you are eating plant. The first point, the starting point of grazing food chain is plant. And detritus food chain, okay, which is also called saprophytic food chain, which having the starting point which we call detritus. Okay, in detritus food chain, plant never be the starting point, while in your grazing food chain, plants is the starting point. You can see here, grass, it is eaten by grasshopper, then grasshopper eaten by frog, frog eaten by snake, snake eaten by hawk, and when hawk dies, it is eaten by decomposers. Okay, same thing you can see here. This is an aquatic food chain. We have phytoplanktons, plant, the small plants which is present in the water. It is eaten by your zooplankton. Then zooplankton are eaten by small fishes. Small fishes is eaten by your mackerel, small medium sized fishes. Then mackerel is eaten by tuna, large fish, and then it is eaten by large shark, top predator of aquatic ecosystem. Okay, so we have grazing food chain. Okay, in the begin of this, producer that capture the solar energy and through photosynthesis feed the energy into the food chain. Okay, in the grazing food chain, as I said previously, plant is on top. Plant is the first one. Okay, in the food chain, there is unidirectional flow of energy from sun to producer and then to the various consumer. Okay, sun se energy I, it is taken by your producer. Producer gives that energy to primary consumer. Primary consumer gives that energy to secondary. Secondary to tertiary. Tertiary to top consumer. No coming back. Only one direction. Okay, single way, no double way. Okay, usually there is maximum 4 or 5 trophic level in a food chain. If it crosses 5 trophic level, then that food chain is unstable. Okay. Next, detritus food chain or saprophytic food chain. Okay, so this food chain begins with decomposition of dead organic matter or decomposer. As I said previously, detritus food chain starts from detritive. Okay, detritus, dead matter. Okay, in this food chain, primary consumers are bacteria and fungi because the dead organic matter is eaten by your bacteria and fungi. Okay, dead organic matter, then bacteria and fungi. Bacteria and fungi eaten by some small microorganisms or eukaryotic cells. Then it is eaten by insects. Then insects eaten by birds, and all this food chain runs. Okay, not in detritus food chain, energy flows rather very slow, yet magnitude of energy is great. This is the very important point of this topic. That is, in detritus food chain, energy flow is rather very slow, yet magnitude of energy is great. Okay, and the vice versa will happen inside your grazing food chain. In grazing food chain, energy flow is very fast, but the magnitude of energy is slow. I am talking about grazing food chain. In detritus food chain, the energy flow is very slow because the process of decomposition is not a fast process. It takes some days, some months, some years to decompose a body. It is depend on the size of the body. If it is a body of rat, it takes some days. If it is body of a tiger, of an elephant, then it will take some months. If it is a body of whale, then it will take then 12 to 1 year to decompose. Okay, so that's why the energy flow in this is very slow, but the magnitude of energy, the energy which is coming which is crossing from one trophic level to another trophic level is very high, and the vice versa in your grazing food chain, that food energy is very fast, the flow of energy is very fast, but the magnitude of energy is very low. Okay, in mangrove vegetation this food chain goes up to the big organisms. Okay, till the tiger it will go. Okay, see, the dead mangrove leaves eaten by bacteria and fungi, then it is eaten by aphid, mollusk, crab and nematode, then it is eaten by small fishes, and small fishes is eaten by tiger. Okay, that is your mangrove food chain, which is a type of detritus food chain. Okay, next is your trophic level. Okay, so we studied that different organisms are sitting in different places. This different places in which different organisms are seated, this is called trophic level. T1, T2, T3, T4, just like that. Okay, so in your trophic level, producers occupy the first trophic level, T1. Okay, then your primary consumer, T2, secondary consumer, T3, tertiary consumer, T4, and your top consumer, top predator, T5. Okay, then we have some important points regarding to your food chains, that the aquatic ecosystem GFC is a major conduct of energy flow. Okay, in terrestrial ecosystem, a much amount of energy flow through DFC than GFC. Okay, DFC through more energy than your GFC in terrestrial ecosystem, land ecosystem. Okay, DFC may be connected with GFC at some level. Okay, then some organism of DFC are prey to the GFC animals. Okay, when you are studying intermixing of that food chain, food webs, you will see that some organism of DFC are prey to the GFC animal. We studied in this also. Okay, that DFC organism is eaten by the animals which is present in your GFC also. Okay. Okay, and this is your food web. When many food chains comes together and intermingle to each other, then it is called food web. Food chain really does not exist in an ecosystem. It is an imaginary flow of energy. Really in an ecosystem, food web exists. Okay, again I am saying the line, it is very important line, that really food chain do not exist in nature. In really sense, food web exist in the nature. Okay, next we have ecological pyramids. Okay, now this food chain does not giving us adequate idea, so we do a graphical representation of food chain in the form of food web, pyramid. Okay, so on the basis of that we have three pyramids: pyramid of number, pyramid of biomass, and pyramid of energy. Okay, so pyramid of number says you number of particular organism in a particular trophic level. Okay, just like T1, T2, T3, T4, different animals are present here, and different number of these organisms are present in different trophic levels. That is your pyramid of number. Then we have pyramid of biomass. Pyramid of biomass tells you the weight, the dry weight, the flesh weight of that particular organism which is present in a particular trophic level. Then we have pyramid of energy. Okay, pyramid of energy, it is the only pyramid which is always upright. It is never inverted. Okay, pyramid of energy tells you that energy flow of a particular food chain, of from one trophic to another trophic level, the flow of energy, it is given by a pyramid of energy. Okay, so we are going to study that pyramid of number. In this type of pyramid, the number of individual organisms is various trophic level is shown. These pyramid are mostly upright because number of producer T1 is maximum and number of herbivores, carnivores decrease towards the apex of the success trophic level, such as grassland ecosystem and aquatic ecosystem. You will see here plants are many in number. Then we have primary consumer which is less than plants, then secondary consumer which is less than primary consumer, then tertiary consumer which is less than secondary consumer, and at last we have a single top predator. Okay, so when we going upward in this pyramid, the number of individual is decreasing, that is your upright. Okay, second example, this is an ecosystem of aquatic ecosystem pyramid of number, in which you will see the number of phytoplanktons is very high. It is a microscopic organism. Then we have zooplankton, again its number is very high, but it is less than phytoplankton, it is again microscopic organism. Then above that we have fishes, small fishes, which having more less number than your zooplankton. Then we go more ahead, more upside to the tropic level, then we show large fishes that having less number than your small fishes. And at the top of the pyramid, we have top predator like shark, which is only one in number. Okay, so this is your pyramid of number, which tells you a particular constituency, particular number of organism in a particular tropic level. Okay. Next we have pyramid. This is a pyramid of number in inverted in tree, because tree is single. Okay, a single tree contains many ecosystems. So that ecosystem of tree, the pyramid when we draw in ecosystem of tree, it is inverted. See, a single tree is there, number one, single. Okay, only one tree is there. Then on that single tree, many birds lives. Okay, then on many birds we have different bugs, and on different bugs we have different fleas. Okay, so this is the pyramid of tree ecosystem, which is inverted. Okay, next we have pyramid of biomass. Okay, it will tell you the weight of the organism which is present in a particular tropic level. Okay, so we have first pyramid of biomass for terrestrial ecosystem, in which we see the producer. The weight of producer is 1,000 kg. Then when we go to the herbivores, its weight become 100 kg. Then we go to the primary carnivore, it become 10 kg. And when top carnivore, it will become 1 kg. So the pyramid of biomass for terrestrial ecosystem, it is upright. Okay, then we come to the pyramid of aquatic ecosystem. We will see that thing is inverted. Okay, now tables become round. Okay, turn around. Okay, so it will see that give you producers. They are large in number, but they are microscopic. So what is the weight? There is no weight for microscopic. They are in grams. Okay, so that pyramid of biomass in aquatic ecosystem, it will become inverted, because the weight of producer is very less, while the weight of carnivore is very high. Okay, so this is your pyramid of biomass. Okay, then we have pyramid of energy. Okay, this pyramid tells you the movement of energy from one tropic level to another tropic level, which is also we call 10% law. Okay, in which 10% of energy is traveling from one tropic level to another tropic level, in which there is no loss of energy and simultaneously no gain of energy take place. Okay, so this is your pyramid of energy, which is always upright. Okay, next we have some limitations of ecological pyramid. First, it does not take into account the level same species belonging to two or more tropic level. Okay, sometimes what happened, two species can occupy different level, means some species can be primary consumer and can be same time that species can be a secondary consumer. So this will not give a brief account on a particular species which is present in different tropic levels. Okay, then it assumes a simple food chain, something that almost never exists. As I said previously, simple food chain does not exist in nature. In nature, only food web exists. Okay, next one, it does not accumulate food web. Okay, because it is a single flow of energy, single flow of biomass, single flow of number, then it does not give any account of food web. Okay, and at last, saprophytes are not given any praise. Okay, the detritus food chain is not coming inside your ecological pyramids. Okay, so these all are the limitation of food, that that is the limitations of your ecological pyramid. So from that, your second chapter of ecology is over. Now the last chapter of 12th class, that is your biodiversity and its conservation, we are going to start. हाफ नहीं ज़्यादा भी लग सकता 45 मिनट. Okay, so last chapter of 12th class, biodiversity and conservation. In this chapter, we are going to see different animals, the tricks to consume them, to conserve them. Okay, different habitats, different type of species at different levels. Okay, so let's get started. So topic to be discussed in this chapter: level of biodiversity, means at what level biodiversity exists in a particular ecosystem. Number of species on Earth, where we have some analytical data in which we'll see that number of species present on Earth. Then patterns of biodiversity, loss of biodiversity and biodiversity conservation. Okay. So first definition. Okay. What is the definition of biodiversity? So biodiversity is the diversity of biological organization ranging from cellular microbes to large biomes. Okay. Or biodiversity is defined as the totality of gene, species and ecosystem in a given region. Okay. Edward Wilson popularized the term biodiversity, and the term biodiversity was coined by W.G. Rosen in 1985. Okay. Next, level of biodiversity. So we have three levels of biodiversity. First is genetic diversity, second is species diversity, and third one is ecological diversity. Genetic on the genetical basis, on the micro basis. Then species, now we will go into some higher level. We are going to differentiate between, compare between two different species. Genetic diversity means we are in a single species, but the genetical variety that a single species carries, we are going to discuss in genetic diversity. Species diversity, two different species we are going to compare. Okay. And ecological diversity, in which we are going to discuss on a large level on a ecosystem, on a particular ecosystem, how many species are present in a ecosystem. Okay. So first we have genetic diversity. Diversity shown by a single species at a genetic level. For example, strain of rice, mangoes, different varieties of mango grow in India. Just like in the north we have Dasheri, in the Rajasthan we have Sadabahar, in Gujarat we have Pairi and Vanraj, in the Maharashtra we have Kesar and Inflanzo, in Karnataka we have Totapuri, Malgoa, Manchur, Raspuri, in Kerala we have Chandrakara, Muvavandan, in your Tamil Nadu we have Sindhura, Lumani, in in UP we have Langla, Ratul, in Bihar we have Jaradu, Churusa, in the West Bengal we have Himsar, Malla. That different types of variety of mango India have, that thing is called genetic diversity. In the rice we have 50,000 different strains of rice in India. Okay. In India we have 50,000 different strains of rice. Okay. This is again one of, this is rasparin. It is a medicinal plant. Okay. So different species of rasparin contain different composition of rasparin inside their body. Okay. Then we have species diversity. Now we are going to discuss about the diversity on species level. Now we comprise the species, different species to different species we are going to compare. Okay. So diversity at species level, example, Western Ghat have greater amphibian species than Eastern Ghat. This is your Western Ghat, in which your Gujarat part, some part of Gujarat part, Maharashtra coastal part, Karnataka coastal part, that Kerala coastal part will come. In this part, you will have more diverse species of amphibians than the Eastern Ghat part. Okay, and these all are the different amphibian species which is seen in western part of India, Western Ghats of India. Okay. Next we have ecological diversities. Okay, diversity at ecological system, ecosystem level. Okay, just like for India. Okay, in India we have different ecosystems. Okay, just like desert, Thar desert. Then we have rainforest in Meghalaya, Nagaland. Okay, and some parts of Karnataka, Kerala, Tamil Nadu, you will see rainforests. Okay, mangroves in West Bengal, Eustachians in West Bengal. Okay, coral reef in Lakshadweep and Andaman Nicobar Islands, wetlands in Jharkhand and Chhattisgarh, alpine meadows in Uttarakhand and your Himachal Pradesh, and some part of Bihar and your some part of Maharashtra and Gujarat also. Okay, so these all are the ecological diversity which is seen in India. Okay. Now global biodiversity. Okay, now we are going towards the world. Okay, towards the earth. Okay, so according to IUCN 2004, more than 1.5 million species described so far. Okay, till now IUCN, International Union of Conservation of Nature, tell you that 1.5 million species described so far. Okay, so according to the Robert Mayer global estimate, about 7 million species would have on Earth. He considered the species to be discovered in tropics. Only 22% of total species has been recorded so far. Means 22% of species is recorded too far. Means remaining there is 88% of species is 78 species percentage of species is remaining to be discovered on Earth. Okay. In animals are more diverse, about 70% than plant, including plant and fungi. Okay, on Earth 70% is your animals and 22% is occupied by your plant and fungi. Okay, among animals, insects are the most species rich group. In 11th class you studied that arthropoda is the world largest phylum. Okay, and second largest phylum is your mollusca. Okay, out of every 10 animals, 7 are insect. Okay, number of fungi species is more than the combined total species of fishes, amphibian, reptiles and mammals on earth. Okay, and this is the diagram from your NCERT which tells you different composition of different major taxas. Okay, in plants you will see that fungi and angiosperm occupy the whole pie chart. Then we have a small amount of fern, mosses, algae and lichens on earth. Then we will go towards the invertebrate. Then you will see that more over the invertebrate chart is occupied by insecta. Okay, then we have other animals, means other invertebrates. Then we have crustaceans, means water insects, and at last we have the mollusk, which is third most in invertebrate pie chart. Okay, and at last we have vertebrate, in will we see that half of the pie chart is occupied by Pisces, then half of the pie chart is occupied by mammal, bird, reptile and amphibian, and this you will see that reptile is the most. After that we have birds, then we have amphibians, and at last we have mammals, which is the less in the number. Okay, and the line either this written, number of fungi species is more than the combined total species of fishes, amphibians, reptiles and mammals. You can compare this thing. Okay, next one is biodiversity in India. Okay, what is the biodiversity level in India? What is the different species which is present in India? Okay, so India has only 2.4% of world land area, but having 8.1% of species diversity. That much species diversity India has. World only gave 2.4% land to India. On that 2.4% land of India, having world's 8.1% species diversity in that 2.4% land of India. Okay, India is one of the 12th mega diversity country, nearly 45,000 species of plant and twice as many of animals. If 45,000 species of plant is there and is saying twice as many of animal, means 90,000 species of animals are present in India. Okay, if we estimate Mayer's global estimate, only 22% of the total species has been recorded so far. Applying this proportion to Indian diversity figure, we estimate that there is probably more than 1 lakh plant species and more than 3 lakh species of animal yet to be discovered in India. Okay, and these all are major different ecosystem presence in India, that is Darjeeling tea fields, Konkan coast, that is your Nokan Kalinjan Fall, Cherrapunji, and this is Deodar Forest of Himachal Pradesh. Okay, see the vast biodiversity of India. Different biomes occur in India. Okay. Next we have patterns of biodiversity. Okay, we have different patterns, that different features which affects the patterns of biodiversity on Earth. First is latitudinal gradient. This thing says when you move from equator to poles, the biodiversity decreases, and when you move from poles towards the equator, biodiversity increases because temperature increases. That's why the hospitality to that particular organism is increases when you going from poles towards the equator. Okay, same thing is written here. Species diversity decreases from equator to poles. Okay, tropics latitude range 23.5 degree north to 23.5 degree south have more species than temperate or polar region. This area and this area come under your tropic. Then this area and this from here and this from here. I will draw this on this. Okay, the red line which is I am marking, this area till this area, it will come under your tropic region. Okay, now next color, this green line, this area and this area, means this and this come under temperate forest region. Okay, and the third one which is from blue, this and this is your south pole and north pole. Okay, so example of latitude gradient, we can see that Colombia near equator about 1400 species are present. Then in India in tropics more than 1200 species are present. Then we will go to the Europe 41 degree north, more up towards the pole, 105 species are present. And Greenland 71 degree north means towards the poles. We have 56 species of bird we can see. So now we are going to from the equator towards the pole. The number of species is decreasing. Okay. Tropical forest region like Ecuador has up to 10 times of vascular plant species as compared to the temperate forest region like Midwest of Asia. This will be your example of animal and this one the example of plant. Means in Ecuador you will find 10 times more vascular plants while in your forest region from comparing to the Midwest region of your USA. Okay, because that area is near to your pole region, we are going towards the pole. That's why the number of plant is decreasing, while the Ecuador, which is near to the tropical rainforest, it having more species of plant. Okay, next one, this is some species count which is present inside the Amazon rainforest. Okay, Amazon rainforest have more than 40,000 species of plant, 3,000 species of fish, 1,300 species of bird, 4 to 7 species of mammals, 4 to 7 species of amphibians, 378 species of reptiles, and more than 1,25,000 species of invertebrates. Okay, that is the data of only Amazon rainforest, Brazil. Okay, this is a tropical Amazon rainforest is python. Okay, that is your anaconda. Okay, now next is your species area relationship. Okay, it is again a that graphical relationship in which we will draw two different graphs. One is your parabola and another one your straight line, which says s equals to 2 to the power z. Okay, where s equals to species richness, a equals to area, and c is the y-intercept. Okay, and z is the slope of the line, regression of coefficient. Okay, so on a logarithm scale the relationship is of straight line, then the equation will be log s equals to log c plus z log a. Okay, you have only remember you have to only remember this formula. Okay, log s equals to log c plus z log a. Okay, and it is given by Alexander von Humboldt. Okay, so generally for a small area the z value is 0.1 to 0.2, where for larger area it has become 0.2 to 1.2, and if it is a mega biome, mega ecosystem, then the z value become 1.15. And you know the z value, that z is the slope of the line, means this. Okay, the slope it is line carrying. Okay, next one is your importance of species diversity. What is the importance? What make different species in a particular ecosystem? The diversity of ecosystem, the diversity of species in ecosystem make it more better. It is given by your Tillman long-term ecosystem experiment. Okay, according to David Tillman, plot with more species show less year-to-year variation in total biomass. Okay, if a particular species have more, if a particular land have more number of species, then the productivity does not fluctuate. Okay, if in the first year it is 1,000, then in the next year it will be 1,000. Okay, but if that increased diversity contribute to higher productivity. Okay, if you increase the species in the particular ecosystem, it will increases the productivity of that ecosystem. Okay, because you add new species, that new species will produce more efficiently more food than the previous species. Okay, so it is essential for ecosystem health and survival of human race. Species richness and diversity are essential for ecosystem health as well as survival of human race on the earth. The more species is there, the more material we get. The more herbs, shrubs, trees we have to find, the more animals are present in the ecosystem, they will maintain the balance of ecosystem, and that balance of ecosystem is good for us. If it is not maintained, then it will cause different disaster, different cataclysms. Okay. Next is your river popper hypothesis. Okay, it is an analogy used to understand the importance of biodiversity. It is proposed by Stanford ecologist Paul Enrich. Okay, according to Paul Enrich, there is one plane. Plane equals to, I will write here, you can see easily. Okay, plane equals to ecosystem. Okay, rivet, that screws, rivet of chair, the chair in the aeroplane, equals to normal species, and the rivet of engine or wings equals to keystone species. If you remove rivet of engine and wing, then plane will collapse. Okay, same thing. If you remove keystone species from a particular ecosystem, your ecosystem will collapse. But if you remove rivet of chair, it does not affect your plane. So same way, if you remove some normal species from a particular ecosystem, it does not going to affect a ecosystem. Okay, that is the hypothesis of Paul and Rich River Popper hypothesis. Okay, see, this is your Paul and Rich, in which rivet equals to species, flight, that is your plane, equals to ecosystem, and the rivets which is present on your wings and the engine equals to keystone species. If you delete this thing, this ecosystem will collapse definitely. But if you remove the rivet from the seats, from the some inner part of your plane, which is not that much essential, then it will not going to affect your ecosystem. Okay, and this is known as rivet Popper hypothesis by Paul and Rich. Okay, now next is your I read book data. Okay, red data book. Okay, it is a book launched by IUCN in which they mention the name of different endangered species which going to be extinct soon from the earth. Okay, so IUCN Red List 2004 says that 784 species, out of which 338 are vertebrates, 359 are invertebrates, and 87 of plants were extincted in last 500 years due to overpoaching, due to that mass extinction, due to the habitat loss. Different causes are there. We are going to study further. Okay, example: dodo from Mauritius, quagga from Africa, thylacine from Australia, Steeler sea cow from Russia, and three subspecies of tiger: Bali, Java, and Caspian. These all are extincted from Earth now. You cannot see them in nature. Okay, they are not present in nature. They are extincted. They are rest in peace for whole life. 27 species disappear in the last 20 years. More than 15,500 species face the threat of extinction. Means these are endangered species. Okay. 12% of birds, 23% of mammals, 32% of amphibians, and 31% of gymnosperm species face the threat of extinction from Earth because of us. Okay, the current species extinction rate is 100 to 1000 times faster than the pre-human time. Means when we are not human, when we are that Australopithecus, Ramapithecus, Homo habilis, Homo erectus, at that time there is rate of extinction is very low or nil. But when we become human, Homo sapiens sapien, the type of extinction it is 1000 times faster. Okay, because of our need. When need turns into the greed, it will spoil everything. Okay, so impact of loss of biodiversity. First, decline in plant production. Okay, plant production decline, drought will happen. Okay, next, environmental contribution such as drought. Okay, automatically plant production will reduce, then automatically drought will happen. Okay, then increased variability in ecosystem process such as plant productivity, water use, and pest and disease cycle. All these things will affect it, and it will affect on the population of humans. We are the only organism on earth which hit X on our own leg. Okay, so these are the all the extinct animals from the earth. Okay, this is your dodo, this is Steeler sea cow, this is quagga. Okay, this is your extincted tiger species. In this and in your NCERT only three is given: Bali, Javan, Caspian. But there is more: Nagdong tiger, Logden tiger, Trillian tiger, Boranian tiger, Western tiger. These all tiger species are extincted from the earth. They are not present now on earth. On earth now only Siberian tiger, Royal Bengal tiger. These remaining. Okay. Next, this is your thylacine. Okay, Tasmanian devil cat. Okay, or Tasmanian wolf. Okay, this image is been clicked when color camera is not invented. Okay, at that time this species extincted from the earth. उस टाइम तक कलरफुल कैमरा बना ही नहीं था, इसकी फोटो ही नहीं है अपने पास एक भी कलर वाली. ओके, और इसका तो यह हाल है कि उस टाइम तक कैमरा ही नहीं बना था जब ये एक्सटिंक्ट हो गया था. ओके, इसका तो स्केलेटन ही बचा है अपने पास. सेम फ्रॉम क्वागा, दिस एनिमल इज आल्सो एक्सटिंक्टेड एट दैट टाइम व्हेन देयर इज नो इन्वेंशन ऑफ कलरफुल कैमरा. That type of overpoaching we do. Okay, Then we have the causes of this loss of biodiversity, the four evil quadrates: habitat loss and fragmentation, co-extinction, over-exploitation, alien species invasion. Okay, so first we have habitat loss and fragmentation. Nowadays, we are listening many cases of jaguar, leopard entering inside the cities. Tiger, lions, these top carnivores are entering inside your cities. Okay, they are not entering inside your cities. You are entered inside their home. Okay, because you cut down all the forest, and that due to the cut down. Pause करना. Hello? So the most important cause of habitat loss and fragmentation is the most devilish quadrant of these all four is your habitat loss and fragmentation. Okay, means you are cutting down the home of animals and where they will live. They will invade to your cities. Okay, so example, tropical rainforest lost from 14% to 6% every day. Thousands of hectares of rainforest are being lost within an hour. Okay, now we are recording this video. At that time, 14 hectare, more than 14% hectare of land has been cleared down in Amazon rainforest. Okay, the Amazon rainforest been cut down for cultivating soya bean, for conservation of grassland for cattle. Okay, we are cutting down Amazon rainforest, the lungs of earth, for the food of cattle, for the food of for the production of soya bean like crops. Okay, that how rude we are. Fragmentation badly affect animal requiring large territories and migrating animals. Okay. Next one is your that over exploitation. Okay. Human have always depended on nature for food and shelter. That is the proven thing. But when need turns into the greed, it leads to over exploitation of natural resources. Okay, many species extinct in the last 500 years, just like steeler sea cow, passenger pigeon, were due to the over exploitation by human. We kill them, we eat them. For that thing, these species are extended from the earth. Some species like now recent extinction, that is double horn rhino from Africa. Okay, we kill that rhino for the horn to make some medicines. We extend that thing. Okay, that animal. Presently, many marine fishes population around the world are over harvested, endangering the continued existence of some commercial important species. Okay, then we have alien species invasion. Now we are introducing some superior species inside a ecosystem which will start to inhibit that area and destroying that ecosystem. Okay, I have one example with me, that is your Parthenium grass. Okay, carrot grass or Congress grass. Okay, this is a very dangerous alien species which is introduced in India during the Green Revolution type, when India is on भुखमरी. ओके, जब इंडिया में भुखमरी चली थी 1947 के बाद, तो ये आ गई थी इंडिया में बाय अमेरिका, बिकॉज एट दैट टाइम वी डू नॉट हैव दैट मच wheat to give our country, so we take that wheat from America. But at that time when the wheat is coming with America, from America, at that time in the that bags of wheat, this grass seeds will also come. Okay, and that seeds start to that spread all over the country, and it get the pure ecosystem to grow here in India, and it will become havoc in India. This grass does not have any good use. It is not used as a burning fire. Okay, fire, that firewood we cannot use as a firewood. Okay, second thing, the reproductive rate of this plant, it will one seed, one flower of this plant can produce 50,000 seeds. Okay, means this plant have very high R value. Next thing, this plant release a chemical which is called parthenia, which does not allow to grow different plant species near to this. Okay, if this plant is landed here, grown here, now in this particular area another species will not grow because of that chemical parthenia. Fifth one, this plant causes an allergic reaction in human that we call hay fever. The pollen of this plant is having some that allergic reaction to particular peoples, in which it cause that hay fever like conditions. Okay, so this is the one invasive species, alien species, which is introduced in India. Second one we have, that is lantana. Okay, that is your second invasive species, invasive weed species, which is grown in India, which is lantana. Okay, it is again does not have any good use, and it is a weed like plant which is being grown everywhere, itself grown everywhere. Okay, and it does not have any use in India. Okay, so these all are invasive species. One more example is there, water hyacinth, Jalkumbi, Echinoria, or terror of Bengal. Okay, this plant come in India with Britishers during the British rule in India. Okay, this plant look very good in appearance, but it is a very high productive power plant which can increase its productivity, increase its number in a years in India. Okay, it will introduced by Britishers in India, but it occupy all the water ecosystem in India in within 10 years. Okay, so these all are invasive weed species. Okay, next we have example of Nile perch introduced into the lake of Victoria, East Africa, cause extinction of more than 200 species of chilchid fish in the lake. Because Nile perch is a top predator, it is a very high predator fish, large in size. It will eat more than 200 species of chilchid fish in the lake. Okay. Third example of alien species invasion. This is illegal introduction of African cuttlefish, that is your Clarius, for aquaculture, is posing a threat for the indigenous catfish in our rivers. Okay, this African catfish Clarius, it is not should be cultured in India. It should not be pet in India for either pet purpose or for food purpose because of its high eating efficiency. It eat indigenous species of catfish because it is a total carnivore. Okay, and it eat indigenous cuttlefish species more than indigenous cuttlefish has been extincted from India. That's why. Okay, so this is the Nile perch. See how much large it is, and it eats all these small fishes. And more than 200 species of this fish, Chilchid fish, has been extincted from the Great Victorian Lake. Okay, next we have weed. This is your carrot grass, Congress grass, or Parthenium. This is your lantana, and this is your water hyacinth, Jalkumbi, Econia, or terror of Bengal. Okay, and this is your African catfish. See this much long it is. It can easily eat this Indian catfish. Okay, last one is your co-extinction. Okay, means if one species get extincted, then other one definitely get extincted. That is co-extinction. So when a species become extinct, the species associated with it is also extinction, co-extinction. Okay, example, extinction of parasite when host died, parasite will die. From where it will take the nutrition? Host giving the nutrition to parasite. If the host is died, then automatically the parasite will be extincted. Okay, next one is a co-evolved plant pollinator mutualism. Extinction of one causes extinction of other. The best example of this line is yucca plant. I will write here. Okay, co-extinction, that is your yucca plant and probinus moth. Bumblebee and flowering plants like sunflower. Okay, if these plants get extincted, then automatically their pollinator get extincted. Okay, if pollinator get extincted, then definitely that plant species get eliminated because there is no one who do pollination for these. Okay, so now what are the reasons to conserve all these things? Okay, on the basis of that we have three things: narrow utilization, broad utilization, and ethical arguments. Okay, so narrow utilization. Okay, we humans are narrow minded. Okay, put profit in anything, we will make to save that thing. Same thing here. If a plant species start giving fruit to us, drinks to us, or fodder to us, we start to save that thing. Many medicinal plants are saved till the date they are not get extended, because we know we have the profit from that. If that thing get extended, it will hit our profit. Okay, so that thing is called narrow utilizing arguments. Okay, human drive economical benefit from nature such as food, firewood, fiber, construction material, industrial product, tannin, lubricant, dye, resin, perfume. All these things are extracted out from plants. So we humans consume. Best example, red sandalwood. Okay, red sandalwood, it is sale in ton in crores. Okay, so we preserve that thing, we do not want to extend that thing, so that thing come under narrow equivalent arguments. Okay, so more than 25% of drugs are derived from plants, as you know. Okay, 25,000 species of plant have medicinal value. So all these 25,000 species are preserved by us human, because we are narrow utilizing arguments. Okay, next we have broadly utilized arguments. Okay, broadly the biodiversity play a major role in major ecosystem services. Example, Amazon forest produce 24% of total oxygen in earth atmosphere. Pollination through bees, bumblebees, birds, and birds aesthetic pleasure, or these are the by broadly utilized, means it is for everyone. Okay, either it is animal, either it is plant, either it is human, it is for everyone. Okay, you can see in this image the beauty of these images. Okay, for the beautiness of the ecology, the beautiness of the nature, we should preserve that nature. Okay, then this is the image is clicked by me from Kuru. Okay, next is ethical arguments. If someone not going with that privately, you will apply that thing with government. Okay, applied rule on this thing. Okay, then they will afraid of government. Okay, so ethical arguments, every species have an intrinsic value. Okay, its own value. We have a moral duty to care for their well-being. Okay, we have to preserve them, we have to save them. Okay, next, it is our moral duty to care for well-being of biodiversity and pass on our biological legacy in good order to the future generation. We have to say that to our future generation that tiger is there. Tiger zinda hai. Okay, tiger mara nahi hai, tiger zinda hai. We have to say that. If we do not say that, then how they will know that tiger जैसा कोई जानवर था? They only have to see that thing in the images. Okay, so there is different projects running in the world, that is save and protect the trees, fight for animal rights, like humans, animal feel pain too. Chipko movement, Chipko Andulan. Okay, speak up for those who cannot speak for themselves. Okay, pat that animals. Okay, at least that normal animals you can do. Okay, next is your biodiversity conservation. So we can conserve biodiversity. So there is two types of biodiversity conservation: in situ conservation on site, or ex situ conservation off site. Means in their natural habitat you are conserving them, then it is in situ, and artificially you are creating a condition in which that organism, either plant or animal, can live, then it is off site or ex situ. So how do we conserve biodiversity? So first we have in situ on site. Okay, so in which we have biosphere reserve, national parks, wildlife sanctuary, hotspots. Okay, that biospheres, all these come under that in situ conservation. So first we have national parks and wildlife sanctuaries. Okay, so in India, as according to September 2002 list, 90 national parks and 448 wildlife sanctuaries. Okay, Jim Corbett National Park, it is present in Uttarakhand. Okay, it is the first to be established in India in Nainital district of Uttarakhand. It was established in year 1936, and year Project Tiger was launched in 1973. The span of over 520 square feet kilometer and comprises of hill, marshy areas, river, pine beds, grassland, and large bank. This national park is present. Next, we have sanctuaries. Here, protection is given only to the animals. Sanctuary is for only animals. That national park is for animals and plant both. Okay, collection of timber, minor forest products, and private ownership are allowed so long as they do not harm the animals. You can build house, but the thing is that you do not have to interfere with the ecology of that atmosphere. Okay, example, Priya Wildlife Sanctuary in Kerala. Okay, These all are the images of Jim Corbett National Park and Pierre Wildlife Sanctuary. Okay. Next, we have some data, some national parks here listed. Jim Corbett National Park, Uttarakhand, where we preserve tiger. Then Rajaji National Park, Uttarakhand, we preserve tiger. It is a part of Jim Corbett National Park. Then Khana National Park, MP, for tigers. Then Sundarban National Park, West Bengal, for tigers. Then Bandipur National Park, Mysuru, Karnataka, tiger and elephants. Okay. Then Ranthambore National Park, Rajasthan, tiger. Then Desert National Park, Rajasthan, tiger and black duck. Okay. And Kilado National Park, Bharatpur, great Indian bustard and black buck. Kaziranga National Park, Assam, rhinoceros. Gir National Park, Gujarat, Kutch, Asiatic lion. Okay. Kashmir National Park for musk deer. Okay, Kashmir. So these all are different national park and sanctuaries present in India for the conservation of different organisms. Then we have biosphere reserves. Okay, it is protected area for wildlife. It has three zones: core zone, buffer zone, and manipulation zone. Okay, objective of biosphere to conserve diversity and integrity of biotic community, to provide area for ecological environmental research, to provide facility for educational training. In bioreserve, many educational training camps runs to study the atmosphere, to study the ecological availability of that organism particularly. Okay, number of biodiversity rivers, there is currently 714 biospheres reserved in 129 countries. In India, there is 14 bioreserves. Okay, this is the bioreserves in India. Okay, in Bombay, in Ghats, in Nilgiri, Hyderabad, Calcutta, Sundarban, New Delhi. Near the New Delhi area, you have a national park, that bioreserve in Uttarakhand, which occupies the area near the. This is Nilgiri bioreserve in Uttarakhand. Sorry, in Karnataka. Okay, this is the diagrammatic view of biosphere. Okay, this is the outer area which is called manipulation zone or transition zone. In this area, human population lives. Okay, human settlement is there. Then we have buffer zone. Only some people when can go inside there for the research purpose only. Okay, then we have core zone in which only government personnel can go, just like forest guards. Okay, that biosphere in charges, only these person can go. Human can, normal human, okay, cannot go in core area. Okay, and core area of biosphere having most endangered species of animals and plants, they live here. Okay, next is your biodiversity hotspots. Okay, extremely rich species with high degree of endism, means that organism should be of that particular ecosystem, it should be present from that ecosystem only. Biodiversity hotspots have been identified for maximum protection to the endemic and endangered species. At present, biodiversity hotspots cover more than 15.7% area of earth. Okay, in must these are the criteria to be area to be labeled as a hotspot. It must contain 1,500 species of vascular plant or more than 0.5% of the world total endemic species. Next, it has lost one third of 75% of its primary vegetation. Then it will become a hotspot. Okay, number of hotspots, there are 34 hotspots on the globe, means on earth we have 34 hotspots. Three are in India: Western Ghat, Indo-Burma region and Himalaya region. In the map you can see this is Himalayan region, first hotspot of India. Second, Indo-Burman region, the Seven Sister area, and the Western Ghat, in which your some part of Gujarat, coastal region of Maharashtra, coastal part of Karnataka, full Goa, and Kerala part will come. Okay. Next we have sacred forest. Okay. Put name of God in everything, you will survive. Okay. Just like elephant equals to Ganesha, monkey equals to Hanumanji, rat equals to Mushak, okay, dog equals to Kal Bhairav. Okay, so put that organism, people plant equals to Shanidev. Okay, put that plant to different religion, different god, then it will automatically save. Okay, so these are the undisturbed forests without any human intervention and a special religious importance to a particular culture. Okay, this area usually dedicated to a local deity, just like Kantara. Okay, in India, these sacred groups were brought under the Wildlife Protection Act in 2002. Such forests include a number of rare, endangered, and endemic species. Okay, Sacred Grove in India, Khasi and Jantar Hill in Meghalaya, Western Ghat region of Karnataka and Maharashtra, Aravalli Hills of Rajasthan, Sarj Grove, Chanda and Bastar area of MP. Okay, these all are sacred groves in India. Okay, Aravalli Hills of Rajasthan, it is famous for black buck, Indian great bustard, khejdi. Okay, this is sacred groups. Okay, in India, all these are the sacred group present in India. Okay, next one is ex situ conservation. Now you are not conserving that particular organism in their natural habitat. You are conserving that particular organism in an artificial condition made by human. Okay. So botanical garden, zoological park, arboretoriums are the conventional method of ex situ conservation. Cryopreservation is also an example of in ex situ conservation. India has total 122 botanical garden and 275 zoos, deer parks and aquarium and safari parks. Okay, these all are different examples of ex situ conservation. The semen containers, okay, that is your in that vegetative propagation. Okay, zoo, safari. Okay, in situ conservation, national park, wildlife sanctuary, national reserve, bioreserve, sacred groups. Ex situ conservation, botanical garden, capitative breeding, seed banks, aquarium, zoo. Okay, and at last we have convention of biodiversity, the Earth Summit and the World Summit. These two are the convention done on biodiversity to save biodiversity, to take action by government of world to fight against the loss of biodiversity. So first we have the Earth Summit. This is a historic convention on biodiversity held in Rio de Janeiro in 1992. It is called upon the national to take appropriate measure for conservation of biodiversity and sustainable utilization of benefits from biodiversity. Means use biodiversity but in a proper way. Do not over exploit. That thing has been tailed in this summit. Okay. Next, the World Summit or Sustainable Development. It was held in 2002 in Johannesburg, South Africa. 190 countries signed the commitment to achieve a significant reduction in the current rate of biodiversity loss at global region and the local level by 2010. Okay. In this treaty, they signed a treaty in which they will tell that we will use biodiversity in proper way, in sustainable way. We do not that over exploit that biodiversity at local level and global level and regional level. Okay, this is the images of these summits which will happen. It is in Rio de Janeiro, 13 to 14 June in 1992, and this is your Johannesburg Summit, that World Earth Summit Sustainable Development. Okay, in Johannesburg in 2002. Okay, so that is your end of the mega marathon 12th biology. Okay, I think you will all understand that thing thoroughly. Best of luck for your CBSE board exams. Okay, any type of board exam, either Karnataka board or CBSE board. Okay, and the one who are watching now, they can kindly subscribe us, like us, follow us, because if you like, if you follow, you will grow. Okay, and at last I want to say that बायोलॉजी समझ के फ्लावर समझा क्या? फ्लावर नहीं, फायर है अपन, फायर, फायर नहीं, वाइल्ड फायर है अपन. तो थैंक यू फॉर वॉचिंग. ऑल द बेस्ट.